Check, check. तो आया ऑडियो चेक. हेलो 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 हेलो.
diagnostics, there are multiple problems. वो भी क्या है कि कंपनीज आई है ना सभी तो वो मार्केटिंग का इतना ये लगा इसका मैम आपको ये कहाँ से हिम्मत मिलती है वो हम लोग फर्स्ट सेशन में थे ना चेयरपर्सन पर अच्छा
Good morning again. Welcome to the 9th annual conference of Molecular Pathology Association of India. You will be, I am rather happy to announce that this conference is live and more than 500 people are watching this conference online in online mode. There is one good news about this conference is if you are availing the membership of MPI Con with 3,000 rupees, this entire conference is free in the virtual mode. You can access the conference, so you can pass this message on to your colleagues and delegates. One more announcement is to be made that both the conference hall, our first conference hall A is on the ground floor and this is on the second floor. So some of you might want to listen few sessions of that hall as well and hear all. So the members who have already who are the members of MPI Con, so they can get the access of the session. All the sessions are recorded and that will be available for all the members for 1 month. For next 1 month. So rather you should if you want to still go again and watch listen to that talk in the hall, it's your choice, but this will be available on the online mode in the recorded version. So now we will move on to the third session of genetics, that is part 2. I would like to invite the chairpersons for this session, Dr. Shefali Desai, Executive Director, MyLab Discovery Solutions, and Dr. Anil Tarigopula, Senior Scientist and Clinical Coordinator, Laboratory Services, MGM Healthcare. Sir and ma'am, please. So we are running short of time, so I have one request to made here that we will be taking question and answers in the probably in the lunch break time. You can interact with the eminent speakers at the lunch break, so we can be on time at the end of the day. Right? We are running short of time, so that is my one. Ma'am and sir, I have just asked to put question and answer on the lunch break or that time. Right? Okay. So, shall we start? Yes. So, over to you, ma'am and sir. We can start this session.
So I would like to invite Dr. Parag. I don't think he would require actually an introduction. He's going to speak on molecular diagnostics in triplet repeat disorders. Dr. Parag, over to you.
At the outset, I would like to thank the organizers for inviting me and to the chairpersons for kind introduction. So we are dealing with triplet repeat diseases. So basically, as you know, the DNA is like a book. It consists of letters and there are around 6 billion letters. Some of the regions in the DNA are having repetitive DNA. So these repetitive sequences are in microsatellite regions and it accounts for about 30% of the human genome. And the main purpose of various lengths of repetitive DNA is to allow for evolutionary plasticity. However, when these nucleotides extend beyond a region, beyond a particular range, the expression of these segments may be expressed as an abnormal protein or they may be suppressed. So this leads to gain of function or loss of function type of mutations, and in certain cases, with every generation, the number of repeats increases, and this allows for a phenomenon called as anticipation. So if there is a disease in the family, it comes to more severe cases in the younger generation and at a younger age. So this is a dynamic mutation. Other than the point mutations or other mutations that we see, this mutation changes with generations. So depending upon where the repeat is, there are various disorders. So if you see the Fragile X disorder, there is in the 5' UTR region. The Friedreich's ataxia has a GAA repeat in the intron, first intron. In the coding regions, there are the polyglutamine ataxias, which are CAG repeats. SCA10 or spinocerebellar ataxia 10 is in an intronic area. It consists of 5 nucleotides. So it's not a trinucleotide, but a 5 pentanucleotide. And there are CTG repeats in the 3' UTR, especially the myotonic dystrophy gene, DMPK, and SCA8, spinocerebellar ataxia 8. So coming to the cases, so this is a family with 3 children with mental retardation. So it's one of the commonest causes of X-linked mental retardation in males, that is Fragile X. So you can see both of these, all 3 of these children have typical faces. They have elongated face, large ears, prominent jaws, and dysplastic dental dysplasia. They are hyperactive, they are autistic. The first test that was designed for Fragile X was a karyotype based test. So you can see there is a break at the X chromosome 27.3 locus. So this was expressed in folate deficient medium only. So that's if you add methotrexate to the medium, this will express. But this is not a reliable test. It's not positive in all the cells. So when they found the FMR1 gene in the 1990s, they developed a test which is called a Southern blot. So what was the basic pathophysiology is that the FMR1 gene in the 5' UTR region consists of CGG repeats. The normal repeats is less than 55. So it produces a FMRP protein, and then it's a normal phenotype. If there is a pre-mutation, then which means a mutation which is unstable, it is 55 to 200 in the repeat size of 55 to 200. So there is excessive mRNA production, and the excessive mRNA production leads to different diseases such as Fragile X tremor ataxia syndrome, Fragile X premature ovarian insufficiency, and Fragile X neuropsychiatric diseases. So all these 3 diseases occur because of gain of function mutation. Then if there is an excessive, more than 200 repeats, the whole CGG repeats becomes methylated, and the mRNA is not produced, and the FMRP production is absent. So this leads to Fragile X syndrome. So there is a typical syndrome which has developmental delay, intellectual disability, autism in 50 to 70%, and there is typical facies, macrocephaly, hypotonia, gastroesophageal reflux, strabismus, sleep disorders, seizures, macroorchidism, skeletal problems like joint laxity, pes planus, scoliosis, recurrent otitis. The adults also can have cardiac problems like mitral valve prolapse or aortic root dilatation. So this is not a recessive disease, it's an X-linked dominant disease, and even females can be affected. Although the incidence of Fragile X in males is 1 in 4,000, the incidence in females is half, 1 in 8,000. So what happens if there is a pre-mutation? So if pre-mutation carriers, especially females, they can lead to transmission of the disease in males. So the disease is transmitted to the transmitting female. So in the transmitting female, sometimes you may have Fragile X tremor ataxia syndrome, or even in males with pre-mutation, you may have this tremor ataxia syndrome. So this is, you get initially tremors, onset is around 60 to 65 years, not in the young age, it's in the old age. So you have ataxias, cognitive impairments, Parkinson's-like features. Even in the MRI, brain shows white matter lesions in the middle cerebral peduncle. And within 10 years of diagnosis, there is possibility that the patient might require assistance to walk. So this is because of toxicity to neurons because of the excessive mRNA. Similarly, the toxicity to the oocytes because of the excessive mRNA can lead to premature ovarian insufficiency in 20% of those of pre-mutation carriers. And this is a hypergonadotropic hypogonadism. And in less than 40 years, there may be amenorrhea. So the gold standard for diagnosis was initially Southern blot. So what do we do with Southern blot is that we digest the DNA with EcoR1 and BLN1. So EcoR1 gives a 5.2 kb site. So there are 2 alleles, right? One of them is methylated and another is unmethylated in the female. So in the normal female, you have 2 alleles. One is 5.2 and one is 2.8. In the normal male, you have 2.8 because it's the unmethylated allele. In pre-mutation, you will get one more band. In full mutation, you will have a band more than 5.2 kb, large band. In full mutation female, you will get a large band more than 5.2 kb. And in certain normal unmethylated full mutation alleles, you will get, this is unmethylated full mutation, you can get a smear between these 2 bands. And a mosaic, you will have a large band as well as a smear. So this is a gold standard, but it's quite laborious. You'll require micrograms of DNA to diagnose it. So a triple primer PCR was developed. So instead of 2 primers, we use 3 primers, one for the CGG repeats and other for the flanking regions. So this is how they appear. So in the normal you will get one band or two bands. In the intermediate you will get again a band. In pre-mutation you will get a ladder and it stops at the pre-mutation range, and in full mutation it goes on to the last where we see the full mutation allele. So this is typically the triple primer PCR on Asura Gen kit. So basically this is slowly replacing the southern blot. Okay, coming to the next disease, we have Huntington disease. You see this, it is an elderly patient. And you see that this is a chorea which is occurring in the patient. You see she has these movements, which is, you ask her to raise her hands and she gives a blessing. So this is called as blessing sign. Then you ask her to protrude the tongue, and this is Jack in the Box. So it comes out very rapidly, like just like Jack in the Box. So this is a disease which is a progressive disease, and it has motor, cognitive and psychiatric disturbances. Onset is around 35 to 45 years around, but it can occur even in juvenile age group if the repeats are more. The median survival, so it's a disease with decreased life expectancy, with survival time being 15 to 18 years after onset. So there is a CAG, it's a polyglutamine repeat in the HTT gene. Normal at 26 or fewer, pre-mutation is 27 to 35, and the full mutation is more than 35. So there are different phases of this disease. Basically, the neuropathology is in the basal ganglia. So there is affection of the encephalin spiny neurons and substance P neurons in the basal ganglia, which leads to the different defects. In juvenile HD, you have more than 60 repeats and the onset is less than 20 years. And there is anticipation. The mother was transmitting in the Fragile-X, and here it is the father who is giving of course both sexes can transmit, but it is the anticipation occurs because of paternal transmission. In the paternal transmission, the expansion occurs. So you can see here there is an increase in the ventricular size because of deficient in the basal ganglia. And the pathology, you can see loss of neurons and gliosis is present. So this is how it looks in the electropherogram. This is the normal, and in expanded you see two peaks. The larger peak is expanded. So there is treatment, of course, but it is not curative. You have, for chorea, you give tetrabenazine, and you have a newer drug called as pyridopidine, which is a dopamine stabilizer. It basically shows that there was an RCT, there was a systematic analysis from RCT for 1200 patients, which shows that the scale is lesser. Coming to next disease, myotonic dystrophy. So this is again in a 3' UTR region, and there are two types. The type 1 is the commonest, occurring for 90% of patients, and it's a DMPK gene. So here you can see the mother is transmitting most of for the congenital form. So there are three forms: classic form, juvenile form, and the congenital form. So you can see two children with congenital diseases and the mother. So there is a myotonia. You can see this myotonia. Myotonia means inability to relax the muscle. So you can see if you ask her to open the hands, initially she opens it very quickly, but you ask her to do it repeatedly, she will do it slowly. Very slowly she is doing it. So basically this is a clinical myotonia. You can also do electromyography, and you can see the myography showing myotonia. Again, this is a triple primer PCR. You can see the large repeats. However, in the other, we were able to see the exact repeat number size, like in full mutation. But here, we are not able to classify it more than 50 repeats. So even the juvenile will look like this, the classic will look like this, and congenital will look like this. So you have to do the southern blot if you want to do the exact sizing. Coming to hereditary ataxias, you have a whole list of them, or more than 50 to 60 of these ataxias. And what they have basically is wide-based staggering walk, and there is truncal instability. So there is problem in target maneuvers of the upper limbs, like dysmetria, in terminal tremors. There is dysdiadochokinesis, scanning speech, abnormal eye movements. So coming to very short, whether ataxia, you have DRPLA, which is a dominant disease, and this is more common in Japan. You have chorea, dementia, and myoclonus. You have SCA1, which in addition to ataxia has peripheral neuropathy. You have SCA2, which has slow saccadic eye movements and neuropathy. It reported in the large Cuban population. SCA3, which is called as Machado-Joseph disease, which is additional amyotrophy of fasciculations. You have SCA6, where you have sometimes episodic ataxia. Here anticipation is not seen. In some of them anticipation is not seen. So in most of these there is paternal transmission. So you have SCA7 with retinopathy. Here again it's a paternal transmission. Tax 18A is maternal transmission corresponding to in contrast to the others. And in SCA10 you have seizures in certain families. Then you have Friedreich's ataxia. So you have most of these as dominant. Most of the repeat disorders are dominant, but some of them rarely are recessive like this, which like Friedreich's ataxia, which is a recessive disease. Even spinobulbar atrophy, which is extremely recessive, is a recessive disorder. So this is usually a childhood onset disease. Then you have RFC1, which is called as canvas syndrome, which is cerebral ataxia with vestibular reflexia syndrome. Here, all these three diseases, Friedreich's ataxia, RFX, and TBP, and being one, we don't see an anticipation. So there is no increase in the number of repeats. So basically, you can clinically diagnose them, but you have to do the repeats. So you have a whole panel and then you have a whole repeats. In SCA, the difference is most of these are CAG repeats. These are polyglutamine changes. So these lead to accumulation over the age in the form of abnormal aggregates of this polyglutamine in the neurons, and this lead to damage. You have a pentanucleotide in the SCA10. So this is how it looks. So here there is no trail or the ladder that we were seeing in the myotonic dystrophy or in Fragile X, but we see an umbrella-like shape like this in the electrophotogram, because these are not in thousands, these are in 50s, 60s, 70s, like that. So Friedreich's ataxia, you have this typical photo. You have dysarthria, diabetes, cardiomyopathy, areflexia, abnormal ankle reflexes, then you have ataxia, and then you have, so basically this is occurring in juvenile age group. So this occurs in childhood, in contrast to the ataxia, which are mostly adult onset. And this is a recessive disease. So this is affecting the mitochondrial oxidation pathway, the iron pathway. Here you have to do dual primer as well as triple primer. In the dual primer, if there is a normal allele, you will get a good peak. And in the triple primer, you will not see the ladder size. But in the expanded primer, you will not see the dual primer showing because it's expanded. So there will be no product in the dual primer, but in the triple primer there will be a ladder. So this is how to distinguish. This is how you have to do both the primers, dual primer as well as triple primer for Huntington disease, for Fragile X disease. So in contrast to all the other diseases I have mentioned, facioscapular humeral dystrophy is in where the repeats becomes contracted. So there is a contraction of this. So it's linked. So you can see this is a chap with facioscapular. This is a paper which we published in 2010. You can see the polyhill sign. This looks like many hills, and this is a Popeye sign. This it has an arm like a Popeye, and this is a winging of the scapula. So this is one of the commonest muscular dystrophies, and it is 95% linked to 4q allele. So in the healthy patients, you have 100 to 11 repeats. In affected, you have less than 10 repeats. So it's contracted. So what pathophysiology they have found is that when there is normal more than 10 repeats, this region is having epigenetic silencing. So there is methylation of this region. But when there is less number of repeats, it's de-repressed. This region is de-repressed, and then you have expression of DUX4 protein. This DUX4 protein leads to apoptosis of the muscles. So you have basically a disease which causes which is occurring when there is contraction and formation of an abnormal protein. Again, Southern blot is the diagnostic method. So you have all the normal people having more than 35 kb band, and all the contracted bands, 17 kbs, 14 kbs, are all affected patients. In this, there is anticipation, but mind you, the number of repeats do not change with the generations. So there is same size allele with every generation, but there is phenomenon of anticipation. It is not explained why this occurs. Another disease, Gilbert syndrome. So this is a dinucleotide repeat. So in the Gilbert syndrome, what happens? Some of this, it's a very common disorder. These patients have jaundice throughout life, and you give phenobarbital, it responds very well. So what is Gilbert syndrome? There is a gene called as UGT1A1 which causes glucuronide conjugation of the bilirubin. So this glucuronide conjugation is affected, and what happens if there is 6 repeats, which is normal, it functions well. If it is gone to 7 repeats, the TA repeats, you can do it on Sanger sequencing, and you can see there is a homozygous TA repeats, then there is more, it's necessary but not sufficient. So many patients can have homozygous TA repeats but be normal. But in those who have Gilbert syndrome, we do this, and we found in almost all of them they have this expanded TA repeats. So this is like a HLA-B27, that more than, if you have HLA-B27, it's not necessary you have ankylosing spondylitis, but if you have ankylosing spondylitis, most of these patients have HLA-B27. So it's something like that. So most important in this is there is a scope for pre-symptomatic diagnosis, and it's a very challenging area because, of course, in India there is a whole lot of insurance problems because most of these patients will not have insurance. But in the West, where there is a lot of awareness, there is a lot of protection for these patients, where there are various acts which say that these results may not be leading to decreased potential for an insurance. So how does predictive testing or pre-symptomatic testing help in the patient's life? So it influences life decisions like how to plan your career, when to do what, and then basically it reduces the mortality and morbidity by doing a specific surveillance or intervention risk reduction, medical or surgical, and a lifestyle modifications. The family can have informed choices. So for example, if there is a child to be given birth to, they know that they don't want to transplant, they can avail pre-implantation or prenatal genetics. They will improve the society because it improves medical health for populations at risk, help prioritize use of medical resources, and facilitate development of molecular therapies. So in summary, these triplet repeat diseases are a unique type of inheritance pattern. So it's a non-Mendelian type of inheritance. Basically, it doesn't follow the Mendelian laws of inheritance. And it is many tests such as, there are laborious tests, southern blots still being the gold standard. Slowly these triple primer PCRs are slowly replacing these southern blots. And basically this unique phenomenon of anticipation which leads to earlier onset. And of course there are a lot of scope for a pre-symptomatic diagnosis, but there are ethical challenges. So basically this is a unique group of disorders where we can do a clinical as well as molecular diagnosis and give there is a scope for a lot of research in this area, how this disease is developed. It's still not evident how the molecular diagnosis of FSHD is there. So there's a lot of scope for this. Thank you.
Thank you, Dr. Parag, for the excellent talk.
Thank
It
you.
was a comprehensive overview of triplet repeat disorders. It is my pleasure to introduce the next speaker. In view of brevity of time, I think questions will be after the session. Or anybody wants to? One question we can probably take. Okay. Right. So we'll move on to the next topic. Ovarian insufficiency at molecular level. The speaker is Dr. Anu Peshambu. She is a senior group leader, reproductive developmental genetics and cellular reprogramming, Institut Pasteur, Paris. So, over to you, madam.
I think it's working now. Yeah. Okay. Is this any better? So how do I change the slide from here? Slide कैसे change करना है? Or pointer? Okay, so I would like to start by thanking the organizers and Usha ji for inviting me. And I would ask you not to go to sleep because I'd have to ask you for words in between. I'm recovering from COVID and I have serious brain fog. So please stay awake. I'm going to talk about ovarian insufficiency. And how do I get this thing? Okay, that's better. No? Okay. Okay, yeah, it's working. Right. So I'll just give you a brief overview of what you call infertility at a molecular level or at a definitional level. According to WHO, it's considered a disease of a male or female reproductive system, and it is defined by a failure to achieve pregnancy even after 12 months of or more of regulated and protected intercourse. It's a worldwide health issue. It affects over 186 million individuals globally and about 48 million couples. That makes it 1 in 7 married couples. It could be primary, it could be secondary. Primary means that a pregnancy has never been achieved, and secondary is when there is a failure to achieve pregnancy after one. It could be both male and female factor. So if you look at the number of people who are infertile in terms of couples, almost half of them are because of male factor infertility or unknown causes, and around half of them are female factor. Now for the male factors, you'll hear a lot about that from Deepak in the next talk. So I'll focus on the female infertility, where approximately half of them are because of structural defects, if this thing works. These are because of structural defects, and the other half are because of defects of ovulation. But you'll see that there is an overlap between these two phenotypes as well. I'll take you along that during my talk. The incidence and the increase in the frequency of female infertility, primary infertility, has been increasing globally, and it varies from year by year, from country to country, depending on your lifestyle, depending on your environmental exposure. So genetics still plays a role in there, but their genetic factors might predispose you more or prevent you based on your lifestyle and your environmental factors. So I came across this data about India recently, where this group analyzed the National Family and Health Survey from 1963 onwards till 2016. And what they found was that if you start from the first survey at the beginning, overall in India, there has been a trend in decrease in the mid-70s, 80s, a decrease of primary female infertility. That means there was an increase in fertility, or increase in number of children born. And in the last survey, it was shown that barring North India, there is a reduction. In North India, but there is an increase in primary infertility globally in India. Now, coming to female infertility, I'm still not coming specifically to the ovarian insufficiency, but globally, if you look at this pathology, it could have a multifaceted presentation. It could be at the level of fetal gonadal or reproductive system development. Again, in female, it manifests as the disorders or differences of sex determination, which is my core subject. It can present as the anomalies of Mullerian derivatives formation. Then the second aspect, there is a disruption of the endocrine support. Again, there is a big overlap. Okay, let's see. It's a big overlap between these three aspects. So endocrine disruptor, where the hypothalamic pituitary gonadal axis is disturbed, and that can manifest as either hypogonadotropic hypogonadism, amenorrhea, hypoplastic ovaries, polycystic ovarian syndrome, or precocious puberty. Then it is germ cell intrinsic, where there is insufficient production or function of the gamete, and that can present in a multiple forms. It could be at the level of the formation of oocytes. It could be the level of the development or division of oocytes, resulting in a reduced oocyte pool. It could be the arrest of maturation. It could be the arrest of maturation of oocytes. And it's combined with fertilization defects as well as the retention of pregnancy, resulting in early or premature termination of pregnancy. So coming to the tubular defects, because these are not classified under the ovarian insufficiency, nevertheless, they are a part of the spectrum of female infertility. And these are the anomalies of development of Mullerian derivatives. That would be the tubes, the uterus, the cervical development. And it could be malformation of either of these. It could be uterine fibroids or endometriosis. It's got a very high incidence. It's about 3% in live births. It could be the agenesis, atresia, or septation of either the tubes, uterus, cervix, or vagina. The structure of the uterine, according to the latest ESHRE guidelines, has been qualified what they call a normal uterus, which is a typical uterus in a healthy fertile female. That would be U0. And then different defects started from dysmorphia going down all the way to aplastic uterus is from U1 to U5. Either of these formation or a combination of these you can see in these syndromes, like the hand-foot genital syndrome, where there is a fusion of the uterus, there are malformations of the uterine structures, and this is caused by the deletions or the variants, mostly heterozygous, in a housekeeping gene called HOXA13. Or it is an expansion of the poly-A tail of this gene that is associated with hand-foot genital syndrome. This is a syndromic form. Another syndromic uterine malformation and associated with it are endometriosis, rare variants in other HOX genes called HOXA10 and HOXA11, as well as an aberrant expression of their antisense mRNA, which causes transcriptional and translational defects are associated with these malformations. Then one of the rather commoner syndromes would be Myers-Rokitansky syndrome. This has got, it's syndromic, it's got variable uterine malformations, and you can see anything from U1 to U0 in these patients in terms of uterine malformations. And there are variants in a number of genes, including LHX1, Wnt, different Wints, HNF1 beta that are associated with Myers-Rokitansky syndrome. Then there are other female reproductive tract malformations, and there is weaker evidence that there is a contribution of other housekeeping genes as well as chromodomain proteins or other transcription regulators like LHX, EMX, and PAX genes in the etiology of these diseases. Now, talking about the neuroendocrine disruption of neuroendocrine signaling, as you would know that hypothalamus secretes GnRH, which is gonadotropin-releasing hormone, which in turn stimulates the pituitary gland to secrete the gonadotropins, which are FSH and LH, which in turn stimulates the ovaries to secrete, see, I forgot the word, estrogen or progesterone, as well as activins and inhibins, which form a feed-forward loop between the hypothalamus, pituitary, and the ovary. Any disruption of that at a single level or multiple levels can result in female infertility. If there are variants in GnRH, it can result in hypogonadotropic hypogonadism, of which infertility presents as a phenotype, incomplete absence of puberty and infertility. The GnRH stimulates, as I said, the gonadotropins. In the gonadotropins, if there are variants in FSH beta, it can cause hypogonadotropic hypogonadism because of reduced production of FSH. However, if the variant is in the receptor of FSH, which is FSHR, that is associated with hypergonadotropic hypogonadism. And the severity of the phenotype is associated with where the variant is located in the protein. Similarly, for luteinizing hormone, the variants in LH beta that reduce the production of LH are associated with hypogonadotropic hypogonadism with infertility as an associated phenotype. And then variants in the receptor LHCGR are associated with luteinizing hormone resistance. So at the level of clinic, the overall presentation may be different, but female infertility comes as a part of these syndromes, and it's the disruption of the hypothalamic pituitary gonadotropic axis that is the reason for the infertility. Now, in the 40% of the ovulatory disorders that I said in the beginning, if you look at them, majority of them are PCOS, which is about 85%, which is polycystic ovarian syndrome. This is out of what is the classic definition of POI or primary ovarian insufficiency. These are the other which are included in the POI or ovarian insufficiency or primary ovarian insufficiency definition. So, if you look at the definition of female infertility, according to WHO, there is no definition. It is a number of conditions that are put together, and the only commonality is a failure to achieve pregnancy or arrest of ovarian function before 40 years. And POI is a major part of it, although if you look at it, it looks like a very small. So POI, again, by itself is very confusing. Now it's called primary ovarian insufficiency, and it's an umbrella term that is used to cover what was previously called ovarian insufficiency, premature ovarian failure, or premature ovarian insufficiency. Varied presentation, varied etiology. It is a very complex and a very poorly understood condition because, as I said, there is a range of etiologies. There are multiple systems involved, starting from hypothalamus, pituitary, gonad, and germ cells. But what is common here again, that all these conditions, they stem from the premature deprivation of estrogens and progesterone. And there is an arrest of ovarian development and function before the age of 40. Phenotypically, as I said, they are heterogeneous conditions. They could be infertility due to primary or secondary amenorrhea. There is decreased estrogens, elevated gonadotropins, and because of these individuals are at increased risk of osteoporosis and cardiovascular disorders. About 1 to 2% globally are affected, women globally are affected by POI. Again, the incidence varies between different countries and different, okay. So the global incidence is different, the country-wise incidence is different, and where you live, what environmental disruptors you are exposed to, incidence keeps on varying. It varies from year to year as well. Although there is no year-to-year data, but if you look at 5-yearly data, you see a variability in the incidence. And I could not find a single paper where I can come across all the incidences and then tell you that that's the variability, because it's changing constantly. Even though there is a range of heterogeneous conditions that are associated with POI, diagnostically they are similar because the patient present with hypogonadotropic hypogonadism, premature ovarian failure, and ovarian dysgenesis. Now, okay. So what can cause is that the POI, the intrinsic problems within the ovary, either development or function, that could be the cause of POI. It could be genetic and also iatrogenic causes, where the patient has been exposed to surgery or radiation after oncological treatments, and that causes POI. Severe presentation, I will come to it, which is a part again of gonadal dysgenesis, where there is a streak ovaries either due to malformation of the gonad at the time of sex determination, or streak ovaries due to germ cell depletion. At the level of genetics or at the level of molecular mechanisms, we can say that there is chromosomal anomalies that can cause POI. There is deficiency in DNA repair that causes all these associated conditions within POI. There is insufficient hormonal synthesis, and there are defects in pathways affecting folliculogenesis, oocyte maturation, and ovulation. I'll take these one by one. Coming to the defects of the ovarian development, these are the disorders or differences of sex determination or development called DSDs, where there is discordant development of chromosomal, phenotypic, and gonadal sex. You're going to hear a lot about this in the talk tomorrow by Ken McElreevy. I'm just going to give you just the beginning of what's pertinent to POI. So these are the conditions where there is anomaly of gonadal development, which are 46XY complete or partial gonadal dysgenesis, where there is formation of ovarian tissue in the XY individuals. Either there is complete ovarian formation or partial ovarian formation, and these are infertile individuals. On the other side of the coin, where there are anomalies of ovarian formation, or I call them anomalies of testes formation in XX individuals, because in XX individuals, testes are not supposed to form. Either there could be partial or complete formation of testes, and that would be testicular or over-testicular DSD. That can lead to infertility. And these conditions, although they are DSD, but they are grouped under the umbrella of POI. Coming to the genetics of DSD, so throughout my talk, I'm not going to take you along a list of genes. You can find that in very good reviews. I have referred to them throughout my talk. But the genes that contribute to sex determination at embryonic level are also contributing to POI, some of them directly, some of them indirectly. So in cases of XY DSD, if you look at the overall global data, we find that it's only about 50% of these individuals we can identify a genetic cause behind them. And these are the genes that one way or the other are associated with POI. Similarly, in individuals with XXDSD, the condition is a little bit more sad, where we can identify a molecular cause within only about 23% of the individuals. However, one of them I want to point out here is NR5A1 or SF1. This is a gene that is mother and father of all stereogenic factors in the human or in a mammalian body. And mutations in this gene are a major cause of DSDs. However, this is the only known monogenic cause of POI in females. So globally, between 1 to 2% of women presenting with POI will carry mutations in this gene, NR5A1, and it often overlaps with other reproductive pathologies, including DSD, male infertility, and others. In the previous talk, you have heard a lot about fMRI, so I'm not going to go into details of this, but I just wanted to highlight again the same that the previous speaker has said, that fMRI on XQ27.3, as you've heard, is an mRNA binding protein that regulates translation. It's expressed in neurons and granulosa cells, so it's affecting both the hypothalamic pituitary gonadal axis all the way along. And the inactivation causes Fragile X disability syndrome because of the DNA hypermethylation at the promoter or an expansion of the CTG repeat beyond 200 repeats. However, the pre-mutation allele shows elevated mRNA expression but reduced protein expression. There isn't enough protein. And that poses a significant risk from premature ovarian aging. And if I remember correctly, last speaker said that it's 20% of these individuals that present with premature ovarian insufficiency. However, for the full mutation allele, they do not have any ovarian dysfunction, and the full mutation allele, it arises with the repeat because of the expansion of these pre-mutation allele over generations because of the repeat length instability. So there's a very small window that has got a specific mechanism that can cause ovarian instability with the FMR1 mutation. But why I have shown you separately is I'm going to the other syndromic forms of POI. But why FMR1 is important, that if you look at all the syndromic forms, this is the one which has got the highest incidence, where 1 in 150 to 300 females globally that carry this pre-mutation, they present with POI. And it is high, as I said, it can vary by country, ethnicity, by where you live. In places like Colombia and Israel, it's even more frequent, 1 in 100 females. Other syndromes where POI presents as a part of the phenotype, the most common is the Turner, is the Turner variant, which is 45XO with ring X chromosomes, isochromosomes with mosaicism. These present infertility as a part of their presentation. There are other syndromes, some of them due to the genes that are involved in sex determination, others that are involved specifically in ovarian development or global housekeeping mechanisms within a body that can cause ovarian insufficiency. For the non-syndromic POI, as I said, there are genes that are involved in ovarian function like FSHR that I talked about and other genes, then ovarian development like NR5A1, SOX8, genes that are involved in the meiosis during oocyte development and maturation, and obviously the genes that are involved in the maintenance of the primordial germ cells even before it enters oogenesis. variants in any of these genes are associated with primary ovarian insufficiency. If we look at the site of these genes, by function we can divide them in the development as well as the function of the ovaries. If we look at their expression, again these genes are scattered all along the hypothalamic pituitary gonadal axis. So we have genes expressing in the pituitary, in the hypothalamus, in the pituitary, in the somatic cell component of the ovary here, and the genes that are involved in the germ cell component, and even going deeper, the genes that are involved even at the level of chromosomes within the germ cell component. So basically at every point, starting from the initiation of the ovarian development down to the function of it, there are multiple levels at which the genes could be hit and cause POI. Coming to the ovarian development, so the first is the depletion of the primary germ cell pool. So the genes like NOBOX and SOHL2 that are involved in the mitotic development or mitotic division of the primordial germ cells and maintaining the germ cell pool, mutations in these can cause POI. Then, once the oogenesis started, pre-puberty, the genes that are involved in synapses and cohesion as well as recombination during prophase 1. And then after diplotin, there is an dictyate arrest of prophase 1. The genes that are involved in the regulation of cell cycle at this point of time, and all these genes these are the genes that we know in terms of animal studies, but the ones that I have shown here are the ones that are involved in, that are implicated in POI. Then the meiosis is arrested unless the individual enters puberty. The prophase 1 oocyte grows, enters meiosis 1. There is the spindle genes involved in spindle formation, in the cytokine segregation and cell cycle regulation at these points are also implicated in POI. During meiosis, does anyone have a question? During meiosis, when there is the segregation of the chromosome, there's a formation of the synaptonemal complex, cohesion complex here, and the formation of the chiasma. Lots of genes that are at the checkpoints of all these functions are a part of POI. To exit meiosis, there are variants for the meiotic arrest that are associated with female infertility that could be involved in the maturation or at the... Have I gone 30 minutes? I think I have 10 more minutes. 10 minutes. Yeah. Okay. So, yeah. Thank you for destabilizing me. But let's go. Okay, yeah. Here I was talking about the genes that are involved in the meiotic arrest and the exit from meiosis of the oocyte, and all these genes are involved in female infertility or POI. Similarly, genes involved in segregation of the chromosome and spindle formation that I showed you at the beginning, again, are a target for variants that are associated with female infertility. I'll leave with that. So, if I summarize everything here and combine it with the phenotype, this is a very good review from Alexander Rakovic. So the genes that are involved in fetal ovarian development, once they are affected, they cause gonadal dysgenesis or DSDs. Then the genes that are involved in primordial follicle primordial germ cell division, they will cause the depletion of the ovarian reserve, so that is the depletion of the germ cell pool. Once oogenesis started, then it is affected by gonadotropins. Mutations in these genes will cause hypogonadotropic hypogonadism as well as depletion of germ cells. And as oocyte matures at every single step, there are genes that are required, and all these could be a target for primordial for POI. So I'll just summarize that as in the last slide, you saw that ovarian development and function are regulated by a large number of genes, and each of them has a specialist function in a specialist cell, but there is a complex intra and intracellular interactions going on where these genes form a part of a network, talk to each other, and work together along the hypothalamic pituitary gonadal axis. If you look at the animal studies, there are several thousand genes that have been identified that potentially cause infertility in animals, but in human, there are only about 200 genes that are implicated definitely in POI. Of course, these numbers are increasing with the advances in exome and genome sequencing, and combined with functional studies and very good phenotypic characterization of the patients, and these numbers will continue to increase. And finally, something that my lab is actively doing is in vitro derivation of the somatic cells of the gonad. So we have done in vitro derivation of the Sertoli cells and testicular tubules. Ken might talk about it tomorrow. But we're getting granulosa cells starting from the pluripotent cells and then getting PGCs from that pluripotent cells. Seeing the interaction of these two will give us more insight into the mechanisms that are involved in primary ovarian insufficiency in human, and in future possibly therapeutic potential. And in the end, some of the work that I've shown from our lab and other groups, all the people in the lab, past and present, our collaborators, and people who give us the money to do the work. And I'll stop here, and I'll take questions.
So thank you very much, Doctor, to walk through us with a comprehensive knowledge about ovarian insufficiency. Anyone has any questions about? Or maybe we will take all the questions at the break time. Thank you so much. I'd like to invite Dr. Deepak Modi, and he would be speaking about Y chromosome testing in infertile males. He is scientific officer and head, Molecular and Cellular Biology Laboratory, ICMR, National Institute of Research and Reproductive and Child Health, Mumbai. Over to you, Doctor.
Very good afternoon to everyone, and at the outset I thank the organizers, especially Usha ma'am for getting me invited here. Just calling up one fine afternoon and said, Deepak, there is a meeting in Ahmedabad and you have to come. So obviously I can't even ask her that, okay, what meeting it is and what am I supposed to talk, and that's it. So that's how it works, and I'm sure that worked for you, Anu, as well.
She writes one point and says, come.
So thank you, ma'am, for inviting. It's a wonderful meeting. My lab at Mumbai, it's a National Institute of Research in Reproductive and Child Health. It's graduating from IRR to NIRRCH. And my lab is looking at two broad aspects. One is understanding how embryo implants into the endometrium and how things go wrong in women with infertility, recurrent implantation failure, recurrent pregnancy losses. And we also got interested in how placenta is developed and how placentation failures can cause pregnancy-related conditions. The other part, which is more closer to my heart, but that's not the major part of the lab, is understanding molecular basis of gonad development and how things go wrong there to cause infertility. And I have a bunch of enthusiastic students who helped me to do that work, and I really thank them. For today's work, the work is being done by, originally started with Shadan, eventually taken up by Sanjukta, and now majority of the work has been taken over by postdoc Stacy, and I acknowledge all the hard work that they've done. I'm just standing here talking about all the hard work they've done there, so thank you to them. So Anu has made my life much simpler because she's already explained what is infertility. I don't have to explain this audience. Just to tell you again, reiterate the same fact that infertility is increasing, and in India at least 1 in 5 couples have some problem of conceiving. So therefore, it is one of the growing problems. We think that India is a populous country and we don't need research in infertility. We are wrong. The infertility population is increasing. We have heard earlier this year that we are now, our birth and death ratios are now equal, and at some point our death ratios will surpass the birth ratios, and therefore infertility is going to be on rise. And unfortunately, we don't know the causes. And once we know the causes, we also need to develop diagnostics. And today what I'm going to tell you is a story about one such diagnostic assay which we have developed. So unlike female infertility, which is terribly complex, of course, females are complex. Male is a little easier. We have much fewer causes, much fewer genes, which I'm sure Harsh will correct me that there are not much fewer genes, but at least you will agree that there are fewer than the females. And we currently worry about just one chromosome, the Y chromosome, which half of us are here and who has been terribly ignored in most conditions. So let's hear of the story and stand here to defend the honor of the Y chromosome. So all the Y chromosomes here should be a little more happier. So infertility, male infertility is caused by a variety of factors, where the variety of presentations, like the female infertility, but all of them have an underlying genetic cause. And identifying these genetic causes become crucial. As of today, we have karyotypic abnormalities, the very classical ones. Then we have Y chromosome microdeletions, and now the world is moving towards identifying newer mutations, somatic mutations, which contribute to male infertility. So when I say male infertility is easier, is because we have just not looked at it. The females are far more ahead in there. They've looked at multiple things. We are still focused on the Y chromosome, but in time to come we will have more data. For today's presentation, I'm only going to focus on Y chromosome microdeletions as one of the major causes of male infertility. So the poor little Y chromosome, terribly ignored over centuries. So if you really Google up and say that, okay, what are genetic association studies to different chromosomes? Every single chromosome in the human genome is decorated with some association with some disease, except the Y chromosome. I'm not showing you the picture, but you will see it's one of those empty regions, largely because it is believed that there are no genes on the Y chromosome, or the only gene there is important is SRY. And once SRY determines the testis, the Y has no business, and everything is now left to the autosomes. But I'll tell you that's not true, because in the last few years, a number of genes have been identified on the long arm of the Y chromosome. These genes are there, and they play a very important role in spermatogenesis. One such gene which we had looked several years ago was RBMY, which is expressed in spermatogonia. It starts into spermatocytes, goes into packetines, and it even continues when there is elongated sperm, meaning ejaculated sperm has RBMY, telling us that the Y chromosome not just plays a role in deciding spermatogenesis, but also probably extending post-fertilization. This story I'll tell you a little later. So anyways, over the years, several groups across the world had started looking at what are the genetic causes of male infertility. And based on a lot of molecular analysis and cytogenetic analysis, 3 distinct regions have been identified on the Y chromosome, called as AZFA, AZFB, and AZFC. Deletions of these regions, that means chunks of genes are missing out there, is a cause of infertility. And this was some work which we had started several years ago, where we wanted to determine, okay, so if you lose one of these loci singularly or in combination, is associated with male infertility. And there is a little more smaller locus which is in the AZFC, which is also, if deleted, can cause male infertility. So with the primary questions that I started this project around 2005 or 6 was a simple question: what is the contribution of Y chromosome microdeletions to infertility in the Indian population? World over, we had a lot of data. There was very little data coming from India. I think Dr. Tangraj's lab was the only one at that time. Looking at it, we thought, okay, let's look at it in the Maharashtra population. So we looked at about 403 individuals, started very simple PCRs that time, and started looking for AZF deletions, and we found that whatever the presentation was, whether it was azoospermia, that is no sperm in the semen, oligozoospermia, few sperm in the semen, or oligoasthenia teratozoospermia, where the sperm don't move or the abnormal shapes and sizes, had Y chromosome microdeletions. And almost every locus was deleted, so AZFA, AZFB, AZFC, most of these locuses were deleted. So there was no unique phenotype to which these deletions were correlating to. So there was very little phenotype to genotype association. We were saying that, okay, fine, whatever the semen parameters are, one should start doing Y chromosome microdeletions. And our overall frequency in our cohort was about 4.9%. I want you to remember this number because this is going to become crucial at the later part of my talk. Then we started looking at the smaller deletions, the AZFC subdeletions or GRGR deletions, what we talk about. And we find that unlike the European population, I don't remember, but unlike the American population where it does not associate with male infertility, in the Indian population, although some fertile men will have GRGR deletions, the odds ratio of having a GRGR deletion is azoospermia and oligospermia is almost to the tune of 4. That means one is 4 times likely to be infertile if they are carrying a GRGR deletion, and therefore in the Indian population, the GRGR deletions are associated with male infertility. Not just that, even amongst the fertile men with GRGR deletions had sperm count much lower than the one without GRGR, telling us that in the Indian population for sure, GRGR has to be one of the diagnostic markers for male infertility. Now we started looking at the global population. So Stacey started a project with this idea to look at what is the prevalence of Y chromosome microdeletions across the world. And the cumulative frequency came up to be 6.7%, which was close to what we had discovered, about 4.9%. However, when we look at the shades, you realize that there is a wide variation in different parts of the world. Some countries, it was, except for 2 African countries and one of the European, I don't remember which country it was, had much low frequency. But most of the countries were between the tune of 9 to 12%. Whereas India was somewhere about half of it, 6.5%. So we were wondering why India has such a low frequency. And we thought, oh, we are genetically different, we are ethnically different. And things became more complex when we looked at pan-India data. Different labs that by that time started contributing to Y chromosome microdeletion in the country. And we realized that some parts in the country had about 7.6%. To my surprise, Gujarat had a record of about 24%, which I was wondering why it is, but we had no idea. But there were some places which are around 5.5%. So Maharashtra, we had 4.9%, and rest of it we had 5.5%. We were wondering why this is happening, and we were trying to associate with some genetic factors, some mutations, some polymorphisms, blah, 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 only to realize at some point that there was some problem with the diagnostic markers that we used. Now, detecting these Y chromosome microdeletions requires to do a PCR using a combination of markers, which are called as STS markers. And there are about 90 STS markers across these 3 regions on the Y chromosome. Of this 90, the European Union, the European Andrology Association had recommended only 6 markers, which they said was diagnostic in 98% of the population. And most of the labs across the world eventually adapted using the 6 markers. What we observed was that in India, whenever the frequency was around 5%, most of them had used 6 STS markers. But when the frequency was higher, most of them had used more than 10 STS markers. So you're wondering that whether we are using the wrong sets of STS markers to diagnose in the Indian population. And in fact, that was true, that if we were using only 6 STS markers, we were missing out almost 50% of the men. So we were misdiagnosing 50% of the men just because we chose the wrong set of markers which are to be used for diagnosis. In the European population, they worked very well, but in the Indian population, we were missing out half of our cases and we were misdiagnosing. So it is important that when we start developing assays, we start looking at what are the right kind of markers in our population, and then we should move ahead. Anyway, so then we thought that there is a need to determine an India-specific panel which should be useful for diagnosis in the Indian population. And Stacey took up the job, so we forgot our basic science where we were trying to wonder what does RBMY do and what does all those genes do. We said, forget all that, this is important. India needs the right kind of panel. The Indian infertile men need the right kind of panel. So what Stacey did was she started with systematic review. She picked up 45 studies, which was giving very consistent data. And she did a meta-analysis of more than 90 markers in all these studies. She completed and she applied some statistical algorithms to figure out that there were 14 STS markers which she thought were diagnostic in the Indian population. And she said, okay, I'll develop a diagnostic PCR for them. Let's do a diagnostic PCR for them. And we would do a commercial or a lab validation first. Now, doing a multiplex PCR for any molecular biology lab seems to be a very trivial job. Okay, a summer intern can even do that. And it was not a big job till we realized that getting reproducible results each time with a multiplex PCR was far more challenging. For our lab, assay fail ho gaya, it's okay, फिर से लगा लेंगे. But in a diagnostic laboratory, you can't afford that, and therefore we needed one, a reproducible assay, and two, you didn't want your STS markers for AZF, A, B, C running in different lanes. You know, then it became very difficult. So when we show our gels to people who have never seen these markers before, they could never diagnose them at one go. So we thought, number one, it has to be a simple assay, easy to interpret, reproducible, and very robust. And it took 2 years for Stacey to actually get to that best conditions which would work. So I really put my fingers on my ears that when I told her that this is the simplest project I would ever do, it was the most complex assay which we were developing. Anyways, we finally developed this assay, very beautiful. We have about 4 or 5 markers for AZF-A, some markers for B, some markers for C. And what you simply have to do is count the number of bands, and you can figure out whether it's an intact AZF or a deleted AZF. And I'll show you an example here. You can see that in this one of the lanes, in the normal, you should have 2 bands, but if you just see 1 band there, you know it's a GR deletion. So if you know your lane number and number of bands expected, you can say straight yes, no, whether it's a deletion or not. You can see that in one of the patients, you need 4 bands, and you see only 3 bands here. And you see one of the entire lane is empty, telling that it's a much larger deletion. So simply by counting the number of bands in each lane, you can decide which it is. So it was very reproducible, and when we started giving off to people who had never used these assays before, they could very easily diagnose. So we're very happy with it. And we now think that the 48% or the 50% that was missing in the Indian population should be validated by this assay. And I'm very glad to inform you that ICMR has picked up this technology, and it was transferred to APS LifeTech earlier this year. And this PCR assay will be commercially available sometimes at the end of this year, which will be validated. But before that, we wanted to validate it pan-country. So currently, this assay is being validated about 9 centers in the country. Ahmedabad being one of them. Thank you, Dr. Harsh, for being a part of this assay. And Dipanjana is here. You can see that you are also in the team. We really are looking forward for outputs on this assay, that how it's performing, how well it's diagnosing. And one of the underlying things we also want to know is what is the ultimate frequency of Y chromosome microdeletions in the Indian population. Some of the centers have really crossed the 400 mark. Some of the centers are catching up. But yes, probably by the end of this year, early next year, we should have the pan-India data with the best kind of markers which are diagnostic in our population. Now the question arises why this is important. Obviously there's an infertile male, and if you diagnose it, the doctors can decide whether to treat that male or not, because for oligozoospermia, sperm in hand, you can give hormones or certain molecules to increase sperm count, but if there's a Y chromosome microdeletion, sperm count cannot be increased because it's a genetic defect, and therefore the clinician can decide whether to give a treatment or not. But it's a very small fraction. Most of these men who are infertile would obviously go to an IVF clinic. And many of these azoospermic, oligospermic men do have focal spermatogenesis in their testis, because testis is much bigger an organ. It's a continuous process. So somewhere some spermatogenesis can happen. You can extract the sperm by a process called as TISA or a micro-TISA, testicular sperm extraction. Even if you get 2 or 3 sperm, you can inject it into the oocytes, do an ICSI, intracytoplasmic sperm injection, and he can have embryos. Once he has embryos, they can be transferred to the mother, and they can have babies, as many as they wish. So then this becomes a futile exercise. That, okay, why do you want to ever detect it? Just because you want to stop therapy, so some of them. But the problem is, all these couples have to be told that if the father has a Y chromosome microdeletion, all the sons who will be conceived with these deletions will also be infertile. So you're perpetuating infertility in the family, and with every male child born to such a father, you're doubling the frequency of Y chromosome microdeletions in the population. So IVF clinics and the patients need to be told that. So that's one important thing for counseling. But then the problem starts is that often when I speak about this in IVF meetings, the doctors and the patients, both of them say, Dr. Saab, आप अभी मुझे बच्चा दे रहे हो, तो मेरे लड़के को भी आप बच्चा दे दोगे after 20 years. And technology would have improved, it's okay, aap aage kar lijiye. So we say, yes, that's also right. That how does finally testing this in a clinical setup will matter? Obviously it matters because, remember I told you, ouch, Remember I told you that the Y chromosome genes are expressed in the sperm. They are also expressed in early embryos. As early as 4-cell embryos to 8-cell embryos, the Y chromosome genes are expressed. And if any one of these are deleted, obviously embryonic development has to be compromised. And yes, men with Y chromosome microdeletions, if they are conceived after ICSI, if they're fertilized after ICSI, there are large numbers of grade 3 embryos. This is also then we have completed a systematic review and we are writing up this paper where we show that there's reduced fertilization rates and poor embryo quality. So finally we said, okay, there is data out there all scattered. How can we help the patient and the clinician in an effective decision making? So then what we did was, we collected all the data which was available across the world, which was published, some clinics contributed to the data, and we said that, okay, let us start looking at or developing a software where one can input the deletion that they've identified or the STS markers that they have known, and can the doctors or the clinicians get an information of what is the likelihood of a successful pregnancy. So Abhishek Sengupta and Priyanka Narad at Amity University have developed this software, which we call it as Fertility Predictor. This is a software which allows you to predict in men with Y chromosome microdeletions what is the likelihood of IVF success rates. And this is copyrighted, and the copyright is protected now. So here, this is a kind of the opening page which you would get, where you can enter the type of deletion that you have got in your patient, or the STS markers, or the DYS markers, whatever the assay that you have used. You can enter this information, you can upload your gel picture, and you will get a report like this, where you will have the patient name, gel picture, signatures, and here I'll expand this for you. It will tell you that, okay, there's a man with AZFA deletion. His chance of success, the chance of retrieving a sperm in the testis is only 20%, which is typically much higher if there is no deletion. So that means now the clinician can take a decision that, okay, should I even go for an TISA or not? But it's an invasive procedure. Even if sperm is retrieved, the chance of fertilization is only half. If the woman is given 10 oocytes, only 5 of them will ever fertilize. Predicted clinical pregnancy rate and predicted live birth rate is even lower. Typically, in the best of the IVF clinics, it's now around 50%. It is just 10 to 13%. If it's a C deletion, the scenario is much better. The patient can get sperm out of it, but even if the sperms are there, the fertilization rate and the clinical pregnancy rates are still lower. So this software now allows an effective clinical decision making. It doesn't. This test allows you to diagnose it, but this software allows you to take an effective clinical decision making for the detection of Y chromosome microdeletions. Just last 2 slides. That it's not, Y chromosome is not just related to male infertility. Now things are being discovered that Y chromosome has far more implications. The genes in the AZF locus are not just expressed in the testis. Originally we thought they're just expressed in the testis, they express nowhere else. But you see these are tissues here, all human tissues, and these are gene expression patterns, and you can see almost every freaking tissue in the XY conditions express some of the AZFBN genes. Of course, many of these are just expressed in the testis. Telling us that if now it's deletions of these genes can not just affect the testis, but affect the other parts of the organs. And now there is data available which tells us that almost 20 to 30% of the Y-linked genes are expressed in different parts of the somatic tissues, 25% in the brain and 25% in the heart as well. And we now know from literature that if you have spontaneous losses of Y chromosome or genes within the Y chromosome, these men are predisposed to a variety of cancers. But what we don't have data is what happens to these men with Y chromosome microdeletions. Are they having a higher frequency of cancers? We don't know. Not just cancers, there is now evidence from a number of studies which show that, okay, I told you 25% of the Y-linked genes are expressed in the brain, and men who have Y chromosome microdeletions, about 21% of them have some form of neuropsychiatric disorders as they age, because a number of genes are expressed in the brain. And this is one case report where we had a man with a smaller deletion. He conceived and he got a baby with a larger deletion, and this baby had mental retardation and developmental delay. All the other chromosomes were normal. Exome has not been done there, but CMVs have been looked at it and they are all normal, indicating that yes, there is a possible association of Y chromosome microdeletions or Y chromosome in general with health sequences like tumors, like cancers, like mental retardation, And there was a report last month which came that if you spontaneously lose Y chromosome from your blood cells, there is an increased likelihood of developing cardiac fibrosis. That means that the Y chromosome has implications way beyond infertility, and these men at very young age will only come to the infertility clinic. The only thing that will be done to them is their testis is looked at like a sperm producing machine. Kuch usme se nikal lo, dal do, bachcha de do, and the man is sent home. But it is quite likely that these men would eventually develop a bunch of conditions. So now it's important that we evolve a field, what I call it as androgenetics. It's time that we start looking at infertile men more seriously, because the Y chromosome has roles way beyond its fertility to a range from early embryonic development in adulthood, where they could be predisposed to many of the diseases. So just a take-home message, Y chromosome microdeletions contribute to about 10% of cases of infertile males, but this number needs to be revised, especially in the Indian context. As I told you, that you need to use the right kind of markers to detect the right kind of conditions, and such patients will not benefit from hormone therapies. There will be 100% vertical transmission from father to sons. Males with Y chromosome microdeletions are probably increased risk of neurological disorders, probably cancers, and probably certain other conditions. Therefore, it becomes mandatory to test this Y chromosome microdeletions in infertile males at least. And ICMR has released a policy brief where we are now saying that yes, this has to be implemented in most infertility clinics. With that, I'll close. I thank my earlier collaborators who have really helped me to collect these samples and patient material, and Abhishek, Priyanka, and the network of labs who are validating these assays. Thank you, and I'll be happy to take questions.
Dr. Deepak, may I ask?
Yes.
So, Dr. Deepak, there are already available Y chromosome microdeletion kits.
Yes.
So, how it is different from the other available?
Wonderful question. The Y chromosome microdeletion kits which are currently commercially available are using, some of them, like for example, the Creagen kit doesn't use STS markers, it uses STR markers, if I correctly remember. Some of them use a DYS markers. Some of them use a random set of markers which they have done. Here, this is an India-specific panel. So that's point number one. Point number two, here when you look at these tests, when you do a PCR, you have to look at all the lanes and then diagnose your deletion, whereas here it's very straightforward, one-on-one. So it's much simpler assay, and obviously cost-wise, because it's indigenously developed, it will be much cheaper than the available assays.
Thank you, Doctor.
I have a question, Deepak, here. It's me here.
Oh,
Hi.
sorry.
So, it's just a very general question about the very first part about the incidence. So, you know, the WHO criteria is constantly being revised for the sperm parameters.
Yes.
So, what used to be oligozoospermic 15 years ago is now normospermic with 15 million per ml.
Yes.
So, I was wondering, in the light of that, all that, is there any data there which revises the contribution of the Y deletions to oligozoospermia, because now they'd be classified as normal?
Yes. So that's our major worry, that when we had done this, these Y chromosome microdeletions, the oligozoospermia was with an earlier definition. But what we have observed is that time used to call it a severe oligozoospermia, which has now become oligo, and many of these are severe oligozoospermia as per the older definition. So I think we are getting there, but yet we have not come to that point where we want to reclassify these
No, not
contributions.
just for your data, but globally, just like there are all these studies are including.
I think this is one of the most burnt out fields where people have lost interest in doing anything further. But we will now revise it and show that it's important.
WHO is also changing after 2-3 years. They are changing.
Yeah,
Yeah, so.
hi Deepak, I'm here on your left.
Hi.
Very high energy talk, always like
Thank you.
what you deliver. So Deepak, you mentioned about the Y chromosome loss in the peripheral blood.
Yes.
Very intriguing and interesting concept, and the consequence associated with it. Do we also know, do we have any understanding what leads to specifically the Y chromosome loss in the blood cells?
We have no idea. It's just an observation in a bunch of cancers. What they have shown is that in the blood there is a loss, plus there is a loss in the tumor tissue itself. Why it is lost, we don't know, but probably being the smallest chromosome, it just loses in mitosis is one hypothesis. We don't know why it happens.
Because that's not a very mitotically active cell either as well.
True. But the cardiac cells are. The tumor cells are. The blood cells are not, but maybe in the bone marrow it happens, we don't know. Or these could be mosaics to begin with, XOXY mosaics and one of the particular line has expanded over time. We really don't know.
Is there any mouse model, any animal model for this?
So unfortunately, the mouse doesn't carry any of these AZF locus or the genes there, so we can't use that. The XO mouse really doesn't develop anything. I think that's what it is. Right? They're like happy-go-lucky and fertile.
Thanks.
Yes.
Deepak? Yeah. So I was going to ask a question, but I can actually address the previous question as well, because we published a paper in the Lancet about 20 years ago to show that AZF deleted Y chromosomes are unstable, and that those are the chromosomes that are lost from the genome. So that fits in well with more recent studies that show that Y chromosome is lost with predisposition to cancers and so on. So I think people have forgotten about that paper where AZF deleted Y chromosomes are preferentially eliminated from the genome, or they're unstable. So we were able to show that. So it was about 2002, 2003, so 20 years ago we published that. The second question, a point I want to ask is that there's something here that's missing a lot from the field of infertility, which is a big problem, which is the lack of studies, and maybe you can tell me if this is taking place now, of sequencing the genome or exome of normospermic individuals. Everyone is sequencing infertile individuals. The problem is that the publicly available databases don't filter out people who are infertile. So every time you look at your data against a public database, the database is full of variants that cause infertility, and you don't know it. So what we need is a database or set of data saying, this is normospermic individuals, this is a variation that's going on, so we know if Y chromosome microdeletions occur, do they occur in normospermic? If other variants occur in infertile genes, they're present in normospermic. There's a lack of a normospermic foundation for all of these studies.
True. So for our database, we have looked at least about 500 normospermic individuals, and they're not found, except for the GRGR, which is found in a small subset of them. Regarding other locus, I think Dr. Tangaraj had done some work, and now with Harsh, we are doing a much bigger study where we are trying to look for differences between normospermic and azospermic. So in time to come, we should have the data.
I think we are running one session late, so most of the questions can be taken up afterwards. Yes. Thank you very much to all the speakers for really enlightening us for really important, I think, questions or answers, I would say, in our life. Thank you very much.
So I would like to thank the chairpersons for chairing this session. So audiences and my delegates, I would have to love to have your opinion on this, because we are running one session late. I am means with your all concerns and your permission, can we go ahead with this session, because it is already lunch time. But if you allow us, then I will start the session. Is it okay?
How is the other hall?
Other hall is also. So I right now do not have live information what is going on in the other hall. They are also running late. I don't know like how now we have catch up, but if with your all permission we can go ahead, because.
They have a question to
Yes.
announcement.
So it's okay. If they're not eating, we are fine. They're not missing out. Absolutely.
Okay, so we are going ahead with the next session.
Ahead with no questions and ask them to finish in time, properly 18 minutes.
Okay, so I hope all the speakers also have heard this final sentence, like no questions will be asked in this session. All the questions will be taken care in the lunch time. You can meet these respective speakers. So now we are moving into this fourth session of today's, that is Translational Research Part 1. And I would like to invite the chairpersons for this session, Dr. Amrit Kaur Kaler, MBBS MD Pathology. She has done his fellowship in molecular biology and genomics from SCG Cancer Care. She is genetics and molecular medicine, laboratory medicine from Kokilaben Institute of Mumbai. Unfortunately, we are missing the other chairperson, Dr. Himal Shah. So, ma'am, can you do this alone? Okay. Okay. So, If anyone can join Dr. Dhanlakshmi, ma'am. So Dr. Dhanlakshmi Sethi will be joining the chair for this session. So ma'am, over to you. Yes.
So welcome delegates for this next session based on the cancer, back to the cancer genomics here. So we have seen that FGFR alterations have really been established in cholangiocarcinomas and in urothelial carcinomas. And let's see their significance and their impact on the lung cancers. And with that, I'll invite Dr. Amit Dutt, a very renowned researcher, and he's working as a principal investigator at Tata Medical Center, Trek, Mumbai. Over to you, sir.
Thank you, organizers, for having me. Yes, so thank you. Yeah, so a very good afternoon to everyone. Thank you, organizers, for having me. Thank you, Ravita. Thank you, Shane. So I decided today that today I would deviate a little bit from the topic that was initially that I was wanting to talk about. With the permission of the chair, I mean, I'm not going to emphasize too much upon the FGFR today. Rather, I would talk about more of the EGFR and more of the recent work that has primarily been done, all the lung cancer done, along with my good friend and senior medical oncologist, Dr. Kumar Prabhash group at the Tata Memorial Hospital. And the graduate student, Asim, from my laboratory, and a postdoc, Dr. Ashwin Butle. So, without going into the much of like details of the background of lung cancer or cancer in general, I would just in one slide introduce you that lung cancer, as compared to the other different malignancies, it's one of the most prevalent and most common malignancy that you tend to get to see globally and as well as in India among men. And you could see that how deadly it is, that even though the incidence is such high, 11.6, the mortality associated is way too much significant. So that kind of like underlies that this is one of the very deadly disease. And similarly, the trend that what we tend to get to see in India as well, the 5-year survival of people who have been diagnosed with lung cancer is significantly low. Just one brief introduction to you. There are different subtypes that introduce of lung cancer. Primarily, it is the non-small subtype, which is the most predominant one, and then there is a small cell. This is based on the histological classification. And then there are other different subtypes, adenocarcinoma. So my laboratory in general, along with Dr. Kumar Prabhash, we have been focusing on all of these subtypes, and we have been making on trying to understand the genomic alterations and the underpinnings that leads to so that we would be able to identify the deficiencies that might be therapeutically relevant. However, today I wanted to talk about something slightly different and something more recent and very clinically relevant. Here's an old slide. This slide is with regards to the EGFR mutation in lung cancer. Now, EGFR mutation is something very therapeutically relevant in lung cancer. Today, across the globe, including here in Mumbai as well, every lung cancer patient who is walking into the clinics, something that the clinicians would definitely want to know is, with a bunch of genes, whether the person is EGFR mutation positive or not. And the reason why would you want to know is because there are FDA-approved therapy of if the patient is EGFR mutation positive and the response is drastic. So, but this EGFR mutation frequency, it varies by ethnicity. So we were among the first ones to establish one of the well-cited paper that even though it varies in the Caucasian and the East Asian population, our frequency is somewhere intermediate in 23, and that has a clinical implications. We went on to kind of like show primarily the work of Dr. Kumar Prabhash and his group that those patients who were harboring EGFR mutation, if they are given a single pill, so single pill orally at an advanced stage, they survive, we were able to increase their survival. So, and at the same time, it's not just the EGFR, we went on to kind of like show that even the other mutations, such as in KRAS, the Indian population is something where the mutation frequency is distinct than what you tend to get to see in the Caucasian population. Just to summarize, in the Caucasian population, the EGFR mutation frequency is 10 to 15%. In our case, it is about 25 to 30%. In the East Asian population, it is much higher, from 30 to 50%. But in the KRAS, our mutation frequency is lower than the EGFR as compared to the Caucasian population. So in general, this is our observation in the laboratory with all the different, we have now probed in across different tumor types of Indian origin. What we have seen is that the major player tends to remain the same. However, they are altered at a very distinct frequency. More so, we were interested in understanding the resistance. Even though these patients who are receiving EGFR-directed therapy, they more often than not, they tend to come back with the progression of the disease because they develop a resistance. And these resistance we wanted to understand is that what are the underlying mechanisms. There are two different kind of resistance. One is an intrinsic, where the patients are refractory right from the beginning. Even after 2-4 months, you do not see that there is a CT scan and does not see any remission over here. And otherwise, there could be an initial response, but then soon after, the patients, as what I mentioned, they would eventually develop resistance. Now these resistance are directed to the therapy, the patients that have received those pills, that is the tyrosine kinase inhibitor, that drives the resistance to come. There are different mechanisms which is already known in literature. Again, that is not the focus of the talk, but just to enumerate those. Those are that there could be an alternative pathway that can get activated, and there could be an alternative pathway different parallel pathway which can get activated, or the same pathway with an underlying alteration. There could be phenotypic switch. One of the most important and the relevant one is a secondary mutation in the EGFR molecule itself. This is called as the T790M mutation, and this is about 50% of those which are developing mutations are T790M mutations. So this is taking you back to the basics. Now this is so much practiced across the globe that this has really gone to the textbooks. So taking you back onto the textbooks to the T790M, and what recently what we have found something is interesting, and that again will be able to relate it to these clients, and that's why I wanted to introduce you to that. So having a T790M mutation in these patients is not a bad news anymore, because there is a second line of treatment which is already available, which is called as osimertinib. And if these patients, they have an EGFR mutation, activating mutation in the first line, they receive first generation TKI, then they develop a T790M, which is again a kind of good news, because now they can use another inhibitor, where there is a very significant response. But the problem is that there is a bit of a nuance that even though this is an approved treatment, the problem is that there is a bit of a nuance trying to understand other different mechanisms, and also with regards to the T790M mutation, I will introduce to that. So one of the projects that what we recently started with Asim Joshi as the graduate student along with Dr. Kumar Prabhash, what we did is that we got the samples from the baseline. These are adenocarcinoma patients, and they were positive for EGFR mutation, and they were given EGFR TKI, and they responded to it. And then, after some time, the same patient, longitudinally, we collected the rebiopsy samples from these patients, and because we wanted to understand mechanisms which is beyond T790M. In the earlier slide, I showed you that T790M is to the range of 50%. So what we did is that we purposely went for samples which were T790M mutation negative, so that we could look for beyond. And we performed whole exome sequencing and did the standard mutation analysis, and the hope was to find additional mechanisms. What we found was something surprising, and that's what I wanted to enumerate over here. So here are the baseline samples, and here are the rebiopsy, the samples which had already progressed. And in the baseline samples, these were primarily, they were supposed to be T790M negative, as what I said. That was one of the inclusion criteria, that they should be negative for T790M. However, that was done in the clinics using routine technology such as Sanger-based sequencing or a real-time based assay, primarily the work of like Dr. Anuradha Chokle. However, when we threw these samples onto the NGS, what we found that those samples which were initially T790M, which were found to be T790M negative, which is not very surprising given that the higher sensitivity of the T790M. What was striking to us in the rebiopsy sample, there was a significantly higher. These were also supposed to be T790M negative because they had after progression, we had chosen those samples which were T790M. But we found that there was significantly good number of patients, that was like 25% good number of patients, which were still T790M positive. When Asim looked into the details of it, he found that why we really lost detecting them initially is because they had a lower allele fraction. By allele fraction, what you would mean is that of all the different EGFR molecules, what is the number of molecule, what is the percent of molecule which is mutated. So he found that here the allele fraction was about 5% and lower. Now, which is not surprising. We knew that now, that's the reason why we missed out onto the Sanger sequencing or real-time PCR. But the question becomes far more intense and far more concernsome for the reason that I showed earlier, because this was supposed to be a good news. If these patients were harboring T790M, should they have received the osimertinib? Based on this initial results, they were not provided T790M because they were categorized as negative for T790M. So we thought, we asked the question, that is this clinically even relevant? I mean, they are such low allele, this is less than 5%. Would that still respond to osimertinib or not? And did we really miss out these folks to give them the osimertinib treatment? So what we did is that as follows. First of all, if you are wanting to ask these kind of questions, you would want to have the right reagents in your hands. So what Asim does, he takes a lung cancer cell line, which is EGFR mutation positive, and which is sensitive to the TKI. And then there is a protocol called as dose escalation protocol. Over a period of a year or longer, he exposes the cell with a constant dose of an EGFR TKI at a low dose. And essentially at the end of a year, what he finds is the into the same inhibitor. And now you have a system where you can kind of probe for what went wrong in these cells. So this is just an MTT that shows you that this cell was sensitive, and after a year, this cell had become resistant. One of the questions that you would ask, that what if that this was a different cell? I mean, there was during a year of passaging, maybe it got contaminated. So you need to have those kind of controls. So we perform an SGR profiling, and also based on analysis of the exome sequencing, we were able to show that the PC9 resistant cells, what were derived, were indeed derived from the original parent cell. Now here comes the interesting part. There were different clones which were established. Even though they were resistant to the EGFR TKI, when we performed the Sanger sequencing at the T790M loci, what you found that they were wild type. So in this clone, it was wild type, which was suggestive that maybe there might be some other mechanism in there. But hold on. When we performed an NGS on this sample, in this clone, there were different clones established. In this clone, what it was found that the resistant cell was actually positive for the T790M, albeit at a low frequency, at a low mutation allele frequency. Now, this reminds us that this is a model now that has been generated in the laboratory, which recapitulates what happens in the primary tumors. So here is a cell line which is resistant to the first generation TKI and is negative for T790M based on a low sensitive technology such as Sanger sequencing or real-time PCR. However, it is positive based on NGS. Let's go, and this is how we also kind of like develop some other technologies such as allele-specific PCRs and other, and just to show that this mutant allele was indeed positive, and that could be reproduced even beyond by NGS by performing a very specific allele-specific PCR in a real-time based. So now the question that kind of comes around in your mind is that why did these cells were having a low allele fraction? There could be two reasons for it. A, there are a group of cells, there is an heterogeneity. There are a group of cells, of the total number of cells, 5% of those or less of them are mutant and rest of them are wild type. So now what you would do, what Asim does is that he takes and he performs a single cell dilution from these these specific clones. He makes about like 60 to 70 subclones of individual single cell. Now, if it is 60 of those, at least 5% of those should be mutant and rest of them should be wild type. But what then he found is like this is a representation of 20 different clones. He found that all of these clones, the 20 clones that were derived from that cell, were indeed they were still positive for the T790M mutation, and all of them at a comparable frequency of like 5% and lower. That wasn't me. Okay. So what he found that it was which kind of like gives us then alternative. Then what could be the reason? It looks like that, and then when you look at the copy number of the EGFR, so what is happening actually, the nuance is more at the EGFR chromosomal loci. So at the chromosomal loci, there are multiple copies of EGFR, and it's a stable cell. And of all the different EGFR molecule at that loci, 5% of them are mutant and 95% of them are wild type. So even if you keep on making single cell clones of those would still be identical. So that's great. Now let us go ahead and ask the question that whether these guys would be sensitive to the or would respond to the osimertinib. You can show a number of things in vitro. There would be like several different questions whether it would be something that would hold true also in an in vivo settings. But then we did not have a orthotopic lung cancer mouse model. Now that's a very sophisticated technology to kind of like make. So Dr. Ashwin Butle, the postdoc in the laboratory, went ahead and developed a technology in the laboratory where what he essentially does, he directly injects the cells into the intercostal muscle and try and see that if the lung could develop the tumor, and then of course he stitches it up, and if the cells, if the mice is viable, and if you would be able to. So initially he tries that by doing with the dye. He just injects a dye over here, and then you could see in the lungs the dye is there. Then what they do is that they go ahead and they tag the cell, the PC9R cells, with the luciferase gene. So that allows them to do a live imaging of these without even sacrificing. So this is how it was injected, and if you would see, and if you would follow the days, you would see that there was more and more, the intensity was very linearly, it was kind of growing, and when you cut it open, you found that indeed the tumors were formed. And this would, all the pathologists would be kind of like agreeing to this, all the surgeons as well, that the tumors that were formed in the lung were actually recapitulating, or that was mimicking what you tend to get to see in the human. And these mice, and not only these, even when you're taking out the pleural effusions and all, you could see that these pleural effusions were positive for the luciferase gene, also histologically. So what this tells us that now we have been able to successfully establish a lung cancer orthotopic mouse model in the laboratory. Now we are all set to ask the question that what we were wanting to ask. So here's the answer. So when you take the wild type cell, the PC9 cell, and when you make the lung tumors orthotopically implanted, you would see that the tumor, this is how the tumor grows. And when these guys are, and when you take the PC9R cells, the resistant derivative of this, again, they form the tumors. When these folks, they are given erlotinib, that is the first generation EGFR TKI, you see that there is a strong response to it. However, in the resistant cells, you see that the response was not as what you want to see in the in vitro conditions. That was also in the in vitro settings as well. Coming to the next generation, the wonder drug osimertinib, which otherwise is a good news if the patient is T790M positive. What you see is that indeed the piece of osimertinib that is also the parental cell is very highly sensitive to osimertinib. And when you take the PC9R cells, which are at a low tumor mutant allele fraction of T790M, they were mutant as well. They are response sensitive to that as well, which means that we did miss those patients they could have otherwise qualified for an osimertinib treatment. So what it kind of like tells us is that the time has really come. We are all set that bringing these next generation or advanced sequencing technologies to the clinics where all the rebiopsy samples, don't do it with Sanger's, don't do it with the real time. Otherwise, very likely you are bound to miss about 25% of these patients who would otherwise qualify for it. I hope I'm left with like few minutes. I would, or 2 minutes? Okay. So the next question that we wanted to ask, I won't take you into the detail of it, again in collaboration with Dr. Kumar Prabhash and Dr. Vanita Norona, we asked that, okay, now that we have shown that osimertinib is a viable option for patients who are not positive for a T790M, which could not be detected by Sanger's, but then you are expanding it to additional 25%. How about the treatment response? I mean, it's so expensive. I mean, can we really afford it? So, and also there was an ADORA trial. I won't go into the details of it because basically, essentially, this kind of like tells you that osimertinib, when given in an adjuvant settings, if given every day to the patients, the relapse could be prolonged. I mean, there was a very effective clinical trial that was shown. What Ashwin Butle, along with Dr. Vikram Gota, a clinical pharmacologist, we found that osimertinib has a significant half-life as compared to the erlotinib. In the blood, you could see it even after 21 days. So this asks the question, is that, and now that osimertinib, which is an approved treatment, do we really need to give it every day? So again, this could only be confirmed by establishing a mouse model. Won't take into the detail. This was a different mouse model that was made by Ashwin Butle. It's the tail vein mouse model. Essentially, what you today, if you inject the cells into the tail, tomorrow, if the cells are tagged with the luminescence, you would see that these cells directly go and home to the lungs. Now, this is something similar to the settings when the cells would kind of relapse, and if you would be able to detect that. So, taking that. And cutting a long story short, I would show you, cutting a long story short, what I essentially want to kind of like show to you is that, maybe I'll just skip this. So if you would take these cells and they would home to the lungs in just a day, and this is how in different day, till day 18 that we follow, and then we cut it open, it does form tumors. When you give an erlotinib, that is the first generation TKI, on a daily basis, you see that it is still effective. However, and when you give the osimertinib, it is also effective as well, which is a second generation and the third generation that is expected. What happens when you change the therapeutic regimen, when you take the duration of this regimen? When you give on a weekly basis with the first generation TKI, that is an EGF, that is erlotinib, you see that the efficacy is lost. The cells still very happily home to the lung cells, to the lung, and that means this is not a viable treatment with erlotinib giving on a weekly basis. However, when you do onto the osimertinib, you see that even in a weekly dosing of osimertinib was effective and the cells could not reach up onto the lungs. So osimertinib, whether you give daily, or that's what the ADORA trial emphasizes upon, or you give it on a weekly basis, the response appeared to be like similar under in vivo condition. This has great ramification, is that now you can essentially lower down the cost by 1/7 if it would be possible to give the patient just by like 1 week. And this is a long-term treatment. I mean, it would be for several years. Yes, so the cost comes down from 16 lakhs to 2 lakhs in 3 years. That's the summary. So if this would be, this of course needs to, I mean, Dr. Kumar Prabhash is leading this effort along with Dr. Vikram Gota. We would be kind of like need to do a clinical trial to see what we have seen in the mice could also be. But then with the osimertinib, wanted to say that osimertinib is a wonder drug, and that could, if we do an NGS, if we can include 25% additional of people, and then if we could lower down the price, lower down the dosage, it would be something. We have done like other different lung cancers as well. Primarily, I just want to like thanks everyone, my group students who have primarily been working on lung cancer, along with Dr. Kumar Prabhash and Dr. Anuradha Chokle, one of the most important person in these. She's here. So along with them that we have been working, we have been very generously funded by the Tata Memorial Center, the Department of Biotechnology, Wellcome Trust, DBTP, NCI, TST, SORB, and the Terry Fox Foundation. Sorry for exceeding the time, and thank you for your attention.
in the lunchtime. So with that, we will go for the next speaker.
Yes, in the interest of time, I think you can, interact with Dr. Amit later. Thank you for an excellent talk. Our next speaker is Dr. R.V. Saji, Professor, Department of Hematology and Adjunct Scientist, Center for Stem Cell Research, CMC, Vellore. And his talk is interesting. It is on inherited bone marrow failure syndromes.
I start with thanking the organizers to invite me for this conference. And this is, there's a slight difference in the title, and I just mentioned only bone marrow failure disease, and to be specific, I added laboratory diagnosis of inherited bone marrow failure diseases, specifically Fanconi anemia and Diamond-Blackfan anemia. And just to start with the Fanconi anemia, because of a lot of research that we did in establishing molecular diagnosis for this particular inherited bone marrow failure disease. And it's a most common inherited bone marrow failure disease. And all the textbooks you can see is it is so rare that you don't see it very frequently. But however, in certain populations, the frequency is very, very high. And in CMC, being a tertiary center, we see at least 40 patients per year. And there's a lot of importance for differential diagnosis of Fanconi anemia with other types of cytopenia diseases. The clinical features, because it's a hematology disease and it's a progressive bone marrow failure, it's the most common phenotype seen in these patients. And there's a lot of congenital malformations that can be seen depending on the age of these patients. And these patients have increased risk of malignancies observed, and that's seen in a large number of patients, around 30% of patients. And most of these patients with the malignancies, have AML or MDS, two common hematological malignancies. But the most common the phenotype is that all these patients are sensitive to DNA cross-linking agents, including the commonly given drugs to treat the patients. When you do the molecular diagnosis and when you try to correlate what you see in the clinical side and what happens in the lab side, what's important to remember is that there is a large number of patients with the mosaicism. And it's around 15% of the patients, they have normal hematopoietic cells. And that depends on when you see the patient. If you see a little older patient, it's possible that the patient has developed mosaicism with normal hematopoietic cells. And that is very, very common. It's because even as one or two cells in the hematopoietic system gets the mutation corrected, because these corrected cells can outcompete and grow better than the mutant cells. And usually, by 4 to 5 years, the patients can have normal hematopoietic cells. And it's called as natural gene therapy. And this is an important information when you do the diagnosis for the patients with Fanconi anemia. And this has been successfully used for the recent gene therapy trials for Fanconi anemia. And in the biology of Fanconi anemia, what we have to remember is that the interstrand cross-linking agents which make the interstrand cross-links in the DNA are usually they're successfully removed by the normal Fanconi anemia pathway. But in the patients with the defective Fanconi anemia pathway, the interstrand cross-links stay between the two DNA strands, and that causes a lot of problem with the DNA replication, and cells go through replicative stress, and that finally leads to accumulation of mutation in the hematopoietic cells. And that is the basis of Fanconi anemia. And the most susceptible cells are the hematopoietic stem cells, and they need a particular type of cell division. And this interstrand crosslinks which are sitting in the DNA interfere with the DNA replication process. And this is to tell you it's a rare disease with more than 22 genes involved in the FA pathway. And you can see here the names of the genes, and they're involved in the different steps in the ICL interstrand crosslink removal. And any one of the 22 genes, when it's mutated, patients can get Fanconi anemia. And why is it important to do the accurate molecular diagnosis for Fanconi anemia? It's because 25% of the Fanconi patients, they do not have the typical Fanconi anemia characteristic physical abnormality. So it's very difficult to differentiate the idiopathic aplastic anemia and Fanconi anemia just based on cytopenia alone. And it's important for clinicians to know whether it's idiopathic aplastic anemia or Fanconi anemia to decide to take the patient for transplantation or for taking the patient for ATG treatment. And it's also stated in the Fanconi anemia guidelines that it's important to ask the patients, especially the children, and ask the family members whether there's a history of MDS or solid tumors. Then the patient has to be looked for underlying Fanconi anemia. That is mainly because when these patients are treated with the usual dose of chemotherapy drugs, they create intestine crosslinks, and intestine crosslinks will not be removed because of the defective FA pathway, and the patients, you'll see that they just can't take this treatment at any the usual dose of that's given to this patients with the CML or MDS. And also that it's important to remember every time when you do the Fanconi anemia diagnosis is that 15% of the patients have somatic mosaicism, as I stated earlier, but the hematopoietic system may look normal, they may not have cytopenia, but they may have FA features and solid tumors. It's important to see if the patient has physical abnormalities. Then it's important to suspect that the patient may be having somatic reversion and hematopoietic system may be looking normal. But when you start treating, the drugs go all over the body and the patient cannot take the usual dose of all the chemotherapy drugs. And the last important part is that the prenatal diagnosis becomes important, and it's important to screen the siblings because this usually the median age of disease manifestations occur is around 7 years, and the family may be prepared to know whether siblings have Fanconi anemia or not. And I'm just going to take you through how we establish the laboratory diagnosis of Fanconi anemia. As I said, we get more than 40 patients every year, and it's important to differentiate the Fanconi anemia patients from the usual idiopathic aplastic anemia patients. And you can see the chromosome pictures that you see is a usual test that is used for the diagnosis of chromosome aberrations. And when the triradials or gaps or breaks which are seen in the peripheral blood stimulated T cells, and one can identify and say the patient has Fanconi anemia or not. But there are a lot of problems with that. I'll come to that slowly. One of the other tests that we established in the lab is a Western blot. It's called a FANG-D2 Western blot. And most of the genes that's involved in the FA pathway, they are all focused in converting the FANG-D2 protein to get ubiquitinated. So when you do a Western blot, you can see two bands. That means that patient is normal, and give a point to it. And the two bands that you see, and that actually shows that it's a normal pattern. And you can see in the patient's fibroblasts and T cells, only one band is seen. The FANG-D2 is not ubiquitinated, therefore the patient has Fanconi anemia. And this is to say the CBS scores, chromosome breakage scores that we calculate from these chromosomes. And you can see in the Fanconi anemia patients have more of these breaks that are seen in the peripheral blood. And what we did was, we did the chromosome break scores, and we did a comparison with this Western blot, and also we did NGS, and I'll show you some of this data in the subsequent slides, and compare to see which is the most important test for identifying and differentiating Fanconi anemia from other diseases. And in this slide, we see that we recruited 144 patients for this study. And these patients, after doing the chromosome breakage analysis and Western blot, we also took them for exome sequencing, and we did robust bioinformatics analysis in collaboration with MedGenome Bangalore. And there's a lot of discussions that we did, and they helped us to establish this protocol. And also that finally, when we had all these missense mutations, those who were doing the bioinformatics analysis, you know that you get a lot of variants which are called variants of unknown significance. And we applied a recent criteria, this is called Eve score, to identify the mutations which are really pathogenic. And this Eve scores really helped us to identify the benign, to differentiate benign and pathogenic mutations whenever you see variants of unknown significance of bioinformatics data. This is to say that for 95% of the patients we could do the genotyping. These are types of mutations here: homozygous, compound heterozygous states, and you can see here FANK is the most defective mutation, followed by FANK-L. This is an observation that KEM, NIH also had here that FANK-L is the second most mutation that is defective, and followed by FANK-G. There's a slight disturbance in the frequency of genes that are defective in Fanconi anemia, although FANK-A is the one that everyone has observed in all the populations. And all other genes, mutations are very rare. And in bioinformatics analysis, we also use for identifying copy number variations. You know, bioinformatics can successfully identify SNPs. And with the help of Medgenome, we did the copy number variations, and subsequently these copy number variations were analyzed by MLPA. And we did for a large number of patients, we could successfully see this copy number variations, which are confirmed by MLPA. You can see here a lot of patients we did MLPA for the deletions, which were identified by the bioinformatics analysis, and MLPA and bioinformatics correlated, MLPA and CMV analysis, bioinformatics analysis correlated. As I said earlier, we get a large number of sometimes the VUS. And what we did was, for some of those important cases, we knew that the patient had Fanconi anemia, but the mutations that we're seeing were VUS. So we did complementation analysis by cloning these genes in lentiviral vectors and transducing into fibroblast. If the patient had a FANK-A VUS, we took the fibroblast and transduced with a lentiviral vector expressing FANK-A. Then we looked at whether there's phenotype correction in the fibroblast. And subsequently, we did Western blot and checked whether the FANG-D2 ubiquitination is restored or not. And we confirmed that the 10 pathogenic and 10 VUS missense variants were analyzed with complementation analysis. And so far what we see, there's 100% correlation with Eve scores. And this is the details of the FANK-L mutation, which is the most common mutation found in the Fanconi patients. And this is around 20%, and it's exactly what NIH KEM did. We did this analysis and confirmed that there is exome skipping that occurs in these patients. Additionally, we did complementation analysis by transducing the fibroblast with the FANK-L lentiviral vector, and after transduction, there is FANG-D2 ubiquitination, confirming that this mutation is pathogenic. And after doing chromosome breakage analysis, Western blot for FANG-D2, and also performing the exome sequencing for a large number of patients, the major observation was FANK-A, FANK-L, FANG-G. These three genes were most defective. What we did was to make the test faster and easier and cheaper so that most patients can afford. What we did was we did long amplicon PCRs to amplify amplify FANK-A gene and FANG-G genes, and pooled them as one tube and did the sequencing by NGS. And this is much cheaper, and it could be run with spiking in exome sequencing samples. It worked out to be only 1,700 rupees to find out the mutations in the FANG-G and FANK-A exonic region. And it's important because most patients can't afford doing a whole exome sequencing and robust bioinformatics to be applied to identify these mutations. And there's one test which is usually used when you read the literature that cell cycle analysis can be used. We had applied for a few patients. You can see here the patient 1 and patient 2. There is G2M arrest here that occurs the last peak here. And in the normal control, when we add mitomycin, we do not see cell cycle arrest there. And we are not doing routinely because it's time consuming to make LCLs. However, the routine step what we do is chromosome breakage analysis and Western blot. And some cases, when it's required, we do long amplicon LC. I'll come to the conclusions there. So what are the important findings after doing this 144 patients? This data is generated from a Wellcome DBT grant for looking at different objectives. One of the objectives was to do the genotype-phenotype correlation. This is outcome and spin-off from that particular objective. And one important finding what we had was, in most of the labs use a particular protocol deciding the CBS score of 40. And we see that that is wrong, and the CBS score has to be changed to 15. And this is backed up by the FANG-D2 Western blot and exome sequencing. The second important information that's important for the diagnosis is that using robust bioinformatics analysis, it's possible to genotype more than 95% of Fanconi patients. And Western blot, in most cases, is more sensitive than chromosome breakage analysis. And we could do in a large number of patients the differential diagnosis between aplastic anemia and Fanconi anemia, especially children. And the other part is, as I said earlier, because we could see a large number of patients with the FANC-A and FANC-L mutations and FANC gene, we developed this protocol, and we can see that within 10 days we can do the molecular diagnosis in less than 4,000 rupees or so. And it's like if it's taken exome sequencing 3 weeks, a huge data will come, it'll occupy a lot of space in your computer, then you have to do bioinformatics analysis and subsequently do, it takes more than 30 days to get meaningful data. And we also performed prenatal diagnosis. You can see here that in the chorionic villus samples, we cultured them and added mitomycin C to create the intestinal crosslinks. And you can see here in the chorionic villus sample is not affected. And in addition to using this Western blot to analyze and detect these mutations, to identify the FANCD tuberculation status of severe tissues. So far for 7 patients, we did mutation analysis and performed the prenatal diagnosis. And this is part of a big study using induced pluripotent stem cells to study the disease mechanisms and all. And what I presented so far is about the objective 1 on genotype phenotype correlation. And I'll just, do I have 2 more minutes? Okay. And I just go to the second part of the Diamond-Blackfan anemia. I'm not going into the details here. And this is a disease that affects only the erythroid lineage. And although it's called as a bone marrow failure disease, and congenital abnormalities may be seen in 50% cases. However, the most predominant phenotype is severe anemia and erythroid hyperplasia when you check the bone marrow of these patients. And what is important, again, in the like Fanconi anemia, is there is a hematological remission that can occur in patients with the Diamond-Blackfan anemia, and they may become therapy independent. And that is because the recent studies have shown that in the hematopoietic stem cells, additional mutations can occur, and they can give a survival advantage, clonal survival advantage, and the patients will be out of the disease. And this is give you genetics of DBA, and most of the mutations occur in the ribosome biogenesis genes. And this is the data from several publications showing that RPS19 is the most commonly mutated gene. And recent studies have shown, in addition to ribosome biogenesis genes, and mutations can occur, few others which are not involved in the ribosome biogenesis, but it's important for the translation process in the erythroid cells. And it's getting more complicated with the classical DBA, DBA-like diseases, and mutations that occur in GATA1 gene that has a slightly different phenotype. This is to say the recent publication showing that these patients can have somatic reversion by having mutations that. This again a part of a big project that we're doing it for the mutations with the patients with the different phenotypes, mutations in the RPS genes and RPL genes. And we're looking at the different phenotypes in the early age and following of these patients to study what exactly is causing this erythroid-specific phenotype in the DBA. And I'm presenting only the diagnosis part here. And this is the current strategy. I've explained most of these techniques like long amplicon NGS that for common mutations, that's much cheaper than doing exome sequencing. Then MLPA is important, and if they do not give the mutations, do the exome sequencing. This is the data that we have from 78 patients that we recruited so far, and we have large number of patients that we did not find the mutations, and we are doing a lot of studies to see what exactly has caused the erythroid-specific disease phenotype. One important assay, which is not usually used for the routine diagnosis, is you can perform ex vivo erythropoiesis DBA. You can take peripheral blood cells and add erythroid culture medium and monitor the erythropoiesis kinetics, looking at the expression of CD71, 235, 2 different markers to look at the progression. And you can see here that the controls differentiate better than the patients with the DBA. And this is a confirmatory test. That is what is expected to happen in the patients there. And in monitoring these cells for 15 days, you get a confirmation that the patient really has DBA, is not immune PRCA. and can look at the cell numbers, get a confirmation that the patient has real DBA, is not any other disease there. I thank all the people who helped me, and I have a wonderful collaborator. Biju George is always very excited, just like a newly joined PhD student in the lab, and others include Dr. Alok Srivastava, Vikram, and Dr. Mamachandi, who always inspired me to work in these areas. And a lot of help we got from MedGenome, and partially the initial exome sequencing was funded by MedGenome, and funding from India Alliance and Department of Biotechnology. Thank you.
Thank you, Dr. Saji, and excellent insight into FA. Questions can be taken, over lunch. We move to the next speaker.
Thank you, sir. I do have a question on PARP inhibitors, and Franken is being here later. Yeah. So our next talk is from Dr. Nidhan Biswas. His topic is a genomic narrative of the progression from pre-cancer to frank oral cancers. Dr. Nidhan is an Associate Professor, Department of Biotechnology, Government of India, National Institute of Biomedical Genomics, Kolkata. Welcome, sir.
Yeah. Hello. Thanks, organizer, for inviting me, and especially to Dr. Neeraj Arora for asking me to deliver this talk. So I slightly changed my title. Can you project? Yeah. Mapping molecular progression paths of oral leukoplakia to frank oral cancer. So my research interest is primarily in the cancer genomics and genome data informatics. So I'm from National Institute of Biomedical Genomics. So we generally do basic research, on the clinical data samples. So as I was saying, with the avalanche of data, there is requirement for developing these omics pipelines and parallel processing, high throughput analysis. So part of my research is concentrated on these areas, but the other part of our team actually works on cancer genomics, primarily oral cancer, and slowly we are moving into the other cancers too. And if you look into the right side of this plot from Globocan data, in terms of the oral cancer, as you see in India, it is like the most prevalent among all other world countries. And with the same, in 2010, a group led by Professor Partha Majumdar and team, and we actually participated understanding the genomic landscape of oral cancer. In 2013, we identified 10 cancer drivers in oral tumors, specifically in oral gingival cancer. And there are 5 novel genes we identified at that time. And as all of you know, that this is primarily driven by, from the habit perspective, tobacco chewing, pan with silicate lime, smoking, and arecanata. This is the combo that primarily work on that. So afterwards, we carried on our work in the oral cancer front, and after 2013 work, we actually looked into the epigenomic front and identified markers that are driving oral cancer, specifically in Indian oral cancer. So we looked into the different subgroups of patients who have different survival outcomes, and we got to see somatic mutations in arachidonic acid metabolism pathway to be one important factor that gives additional survival outcome. Afterwards, we looked into another phenotype, a subset of individuals. They proceed to lymph node metastasis even during presentation, surgical presentation. So we asked what sort of molecular markers can we differentiate these two groups of individuals, and we identified DNA repair defects, and specifically there are specific genomic signature that are enriched in lymph node positive oral tumors. So all of these studies, then we thought of putting it into our database so that the clinicians can mine it and can identify these genomic markers that are present in the Indian oral tumors, and that was available in this DB Genovac website, which was recently published in Database Journal. So So today I'm going to talk about another work that we are doing in terms of the understanding tumor progression from the precancerous patches that the oral cancer patients they have. And as you know, in India, we do see a lot of leukoplakia cases. These are precancer non-malignant oral lesions. And about 0.2 to 5.2% of these cases from the past studies, they know that this is the kind of prevalence. And once clinicians, they detect it through these white thickened patches, the lesions as hyperkeratinizations and mild to high dysplasia. And if you look into the data that these precancer patches in tumors, they actually form malignant tumors in about 5 to 10 years, and their malignant transformation rate is at about 9%. But most interestingly, leukoplakia is observed with or without presence of tumor in the oral cavity. So in some patients, you only see leukoplakia. In some patients, you might see leukoplakia in one area, and in another area you do see oral tumor. And generally with leukoplakia mostly untreated and kept under observations, and you ask patients to come for a repeat visit. So we ask, can we identify the early molecular changes in leukoplakia state and what drives malignant transformation in leukoplakia? So, because as I said, the transformation takes to 5 to 10 year time. So for us, at this moment, the best study design was to sample those individuals where in the same individual I have, leukoplakia patch and also oral tumor. So we sampled 4 biospecimen blood for control from the same individual, and then tumor, and then leukoplakia, and then adjacent normal from the same individual. To study, paired DNA sequencing and paired RNA sequencing. We did at 100x depth, and we kind of generated 100 million reads. And to have comparison to these, we also recruited a set of individuals, a small number, only 11, who only have leukoplakia but do not have any tumor in their oral cavity. And we kind of did a multi-omics analysis, understood the whole genome-wide or rather exome-wide somatic mutation profiles, germline mutations profiles, somatic copy number alteration burden, and looked at the mutational signatures, transcriptomic perturbations, and immune contexture from deconvolution analysis of the RNA-seq data. So overall, as you see here, for somatic mutation, it's not only a single variant caller, because as we see, there are pipeline-specific effects for individual variant calling algorithms. So we actually used 4 variant callers just to take such calls which are kind of identified by 4 different pipelines, and then we looked into the polymorphic and germline, rare germline variants. So we identified somatic copy number variation from both chip data, array data, and also from the WS data. Of course, the genomoid chip data is a little bit, that has a higher sensitivity to retake copy number alterations. But on the WS data also, we see that there are a bit of overlap between both data sets. So then we looked into the both extrinsic and extrinsic mutational processes that are operative in the tumor tissues using a deconvolution algorithm that uses genome-wide exome data to identify mutation signatures, which are well documented now in Wellcome Trust Cosmic site. And looked into the gene expression differences between the leukoplakia and tumor with the adjacent normal tissue, and then used that data to identify infiltrating immune cells by deconvoluting the whole transcriptome data. So the questions that we had to start with, that what are the differences in genomic features in leukoplakia and tumor for the same individual, and we looked into the somatic mutation burden, mutational signatures, somatic mutations in cancer driver genes, somatic copy number alteration status. So here, primary focus to start with in the somatic mutation landscape, and then I'll also talk about a little bit on the germline. So if we look into the leukoplakia without tumor in oral cavity in those n equal to 111 individuals, we see 0.39 mutations per MB. But when we look into the concomitant leukoplakia for individuals where we have both leukoplakia and tumor from the same oral cavity, what you see general trend in higher somatic mutation rate from 1.21 MB in the leukoplakia patch to the oral tumor patch. And what we see is that in these individuals, in the leukoplakia compartment versus your oral tumor compartment, there is a higher proportion of frame shift deletions. You see a lot of red bars in the tumor site, but on the same individual's leukoplakia patch, you do not have those many somatic frame shift deletions. So later on, we asked that what are the mutational processes are operating on that, and as tumor age, there are age-related signatures, there are DNA repair defect-related signatures, there are internal enzymes like ApoBac, they act on the tumor DNA, and these signatures are now available in Cosmic database, and these are named as signature 1, 2, 3, 2 dot dot signature 60. So what we see that these ApoBac signatures already we knew that it's very active in many cancer types and also in oral cancer. So we asked in these tumors, concomitant tumors, what is the effect of ApoBac signatures, and what we see that in 42% of the tumors had higher ApoBac mutation signature in the tumor. And that is, of course, it's not that much detectable in the same precancer areas, lesions, and this is kind of validated from the RNA-seq data. So next we asked that if you look into these precancer patch and the tumor in the same oral cavity, they are always occur at a distance. So in cases they are very nearby, and in some cases they are away from the tumor, almost in the opposite cheek. And so we actually estimated the angular distance between this tumor and then asked that what portion of these somatic mutations that we have identified, how many of these are common between the precancer patch and also the tumor of the same individual, how many are unique to the precancer patch, and that is the leukoplakia, and how many of these unique to the tumor. So the common ones are the yellow ones, and the unique to the tumor is highlighted in here in red. And we'll use this color scheme for the next couple of slides. So as you see, as you go from over like 80-degree angular distance, these are the tumors where we don't see any shared mutations. But these are tumors where we do see, you see those yellow bars. So these are the same mutations that you see in both in the precancer patch and also in the oral tumor. So then we asked that, okay, we know the set of driver genes for the oral cancer from our past studies and also from this study, how it looks from the same individual's background. So if we look at patient number 23 here, what we see in the leukoplakia is in the line above. If you look at, this is the tumor profile, and the below is leukoplakia profile. So for patient number 23, 27, what you see for this set of individuals, you get the same caspase-8 mutation to be in the precancer patch, and the same mutation is also present in the tumor patch. And for some of these patients, you do see only in the tumor patch and not in the leukoplakia. For this specific individual, you do see one specific mutation in the tumor patch, but not that same mutation. It's a different mutation in the leukoplakia patch. So now if you ask, and caspase-8 mutation was found to be shared across leukoplakia tumor in most of these individuals. And if you actually look into their occurrence on the domain, these are mostly located towards the peptidase domain, which from our prior studies, what we have seen in general ICGC cohort, the caspase mutations are kind of present throughout the whole protein. But patients where we do see concomitant mutations, this is a new finding that these are primarily located towards the peptidase domain. And we did RNA-seq analysis with patients for which the caspase mutation is present versus absent. We do see the signals for intraepithelial cell migration and survival for caspase mutated tumors. So we kind of looked at all across the other genes, known head and neck drivers, oral drivers specifically, and we do see TP53 mutations to be present only in the tumor. There are specific Notch1, specific leukoplakia, specific mutations are present, and then in FBXW7 mutations found only in the tumor, not in the leukoplakia. Then we asked what is happening at the CNV level. We do see EGFR amplifications detected only in the tumor, so these are kind of a late event, and Notch1 deletions present both in leukoplakia and the tumor. So these are kind of early event that's driving these precancerous patches to the leukoplakia, to the tumor. So we also next asked that what is the profile of rare germline mutations in the DNA repair genes, and do we see any signatures there, and what do we see that base excision repair and homologous recombination pathway mutations to be highly prevalent in this cohort in comparison to the healthy population cohort of say Genome Asia 100K. So we then asked that if you look into the genes like TP53, Notch1, HRAS, what is happening in terms of the hotspot and pathogenic mutations. So for the same patients, what we see that the second hit, it's getting a second hit in the same genes twice or thrice in some of these patients, and always the second is in most of the times are hotspot or frameshift deletions or stop gain mutation. So this is a little bit busy slides, but all it says that the variant allele frequencies for these driver genes, if you look at in the leukoplakia tissue for the same mutations, when you track that to the tumor, it's going high, and in most cases these driver genes, the alleles, they are actually sitting at a very high variant allele frequency. So these are actually driving these tumor, and this shows that there are clonal growth of these selected mutations from leukoplakia to the tumor. So then we actually gone ahead and build the individual evolutionary tree for individual tumors, and kind of we drawn this graph for each tumor and asked what are the common mutations and how their variant allele frequency looks like, and if you look into the tumor specific what are the genes that were mutated and how their variant allele frequencies are expanding. And so to cut down again the long story, that there are heterogeneous evolutionary histories of oral tumors for each individual. So as you see in the right hand side, for each arm, you do see there are different mutations and happening in driver genes, and they are having different clonal advantage. So we gone ahead and looked into the copy number alteration profiles, and it is kind of known earlier, and it's kind of replicated here also, that specific chromosomal arms are getting amplified in the leukoplakia in the oral tumor than the leukoplakia of the same individuals, and there is DNA repair genes getting deleted in tumor significantly more often than the leukoplakia lesions. So finally we did the immune deconvolution analysis, and the major findings are that there are these anti-tumor cells like CD8 plus T cells. These are lower in tumor than the precancerous lesions, and things like there are pro-inflammatory cytokines and interleukins. There are angiogenetic factors like VEGF-C. The anti-inflammatory signals are going high from the leukoplakia to the tumor compartment. So overall, if you get a snapshot, if you kind of try to draw a snapshot of the entire progression map, so early on there are caspase mutations, alterations are happening in these tumors, and then you do started seeing accumulations of pathogenic mutations in key driver genes. There are increased activity of the APOBAC activity followed by clonal expansion. And lately, there are EGFR amplifications and deletions of the DNA repair genes that kind of sum up the story at this moment from this little study. So, of course, we are expanding sample sizes because it's based on the small data. We are expanding that so as to get a validation code out of it and get a final story. What is happening. So we recently published this in Journal of Pathology. We are very happy to get a cover art of also on the journal, and this has been led by our colleagues Arnob and Chitrapita, and there is a big team behind this study, and Professor Arindam Moitra and Professor Partha Majumdar has kindly helped. The study was funded by CIMEC project and Department of Biotechnology. Thank you. So I'll be very happy to take any questions if you have.
Thank you for your time. Any questions from the audience? I think we can have a break from lunch, but I'm sure the mutational signatures which you have introduced here is a big topic to be discussed. So later we can do that. Sure. Thank you so much.
Thank you, chairpersons, and thank you, speakers. Hello. Okay. Thank you, chairpersons, and thank you, speakers. I would like to make announcement here that we are taking only 30 minutes of lunch break as we have to match up with the timings. And I would like the chairpersons of the next sessions to be available exactly sharp by 2:50. So it is right now 2:20, and we will start the next session after 30 minutes of lunch break at 2:50. Thank you, everyone.
सबने हमें देना है
Okay, I mean. Okay.
मैं शेयर कर रहा
એટલે તો કશું કરવું બેટરી કાઢો ખોટી બેટરી બધાઈ કરતા આપણો સીધો પાવર ઉપર જ છે હું ટેન્શન અરે પેલું અચાનક પાવર બંધ થઈ જાય તો તારા કેમેરો બંધ ના થાય ટ્રાન્સફર થઈ જાય ભાઈ હમણાં કેવું થઈ ગયું ખ્યાલ છે ને હમણાં ઓલવાઈ ગયો આ સ્વિચ કોઈ બંધ કરી નાખ્યું ને તો કેમેરો પર મને ખબર જ
So loving.
Just met for the first time and we came down.
Thank you.
Very happy.
કાં તો પેલી બાજુ લાઈન નહીં આગળ આવી જાઓ તમે ત્યાં બેસો આગળ આવી જાઓ ના ના બસ થોડા પાછળ બરાબર ના આટલું બરાબર લાગતું તો બેસ બેસ હું તો અહીંયાથી નીકળતા હા હું પાછળ જતી રહું એક કહેતા ને એક ખસી ગયું પાણી જોઈએ જ કોઈ હા અહીંયા પાણી જોઈએ જ કોઈ ના સારામાં ખરાબ નહીં સારું ખરાબ નહીં કશો વાંધો જ્યાં જોવાનું ત્યાં જૂઠું જ ઓર્ગેનિક ઓર્ગેનિકમાં જૂઠ કશું ચાલી રહ્યું છે હા સારું હા હા સારું મને લાગે ત્યાં જ બેસવું પડશે પણ ખબર વચ્ચે ભરાય હેરાન કરવાનો છે તો ખબર જ હતી આપણે પક્કો કરીને આવ્યા એટલે એવું જ ને આપણે કરવા લાગ્યું શું અમે સવારે બતાવીને આવ્યા કે ભાઈ આમાં આવું નથી કર્યું તો પછી એ તો અત્યારે મંડી જ પડે ને અને પાછું એવું કીધું હતું કે હું તમને વિડીયો કોલ કરી કોણ કે કરી છે એટલે પેલો મજૂર અચ્છા હા તો કઈ નહીં તું તારે પતાયા પછી કોલ કર જોઈ લે સીધો વિડિયો કોલ
બબીના 3 અને ટોટલ 6 પોઈન્ટ મલ્ટી
કેટલું સારું
હવે એ લોકોને અલ્ટિમેટ
પેમેન્ટ લગાડી ક્વિઝના પોઈન્ટ ઝીરોલોજી
એની મેથી આગળ
અચ્છા
ઝીરોલોજી મેથી
બધા ખુશી છે બરાબર છે સાચી વાત છે
હવે બધા દોડે
ઇલેક્ટ્રિક પોઈન્ટ તારા પર જ છોડી દો
હા મેં કીધું નશી મોડું થશે બધું કહેવાનું ભૂલી ગઈ હા બસ હા કેમ કરું જીન તો ગઈ હા મળી નહીં હા પણ હું કેતન નહીં બધું સાથે
ફૂલ નાફેટ કરनी है मतलब पार्शियल एफेक्ट करनी है
લેમને પડે હું દેવેન્દ્ર નાખી
okay so yeah you put all of it just to know that they're little heartbroken because 31-year-old no so anyway I think we have to do some work because but does it look like that કાલવાળા બેન તો સવારના મેં જોયા હતા આપણે મારી બાજુમાં બેઠા હતા ને પછી જોયા જ નથી એમને કંઈક બધા ગ્રુપ વાળા 2-3 જણા આયા ને બાર ચલો ઇધર ને બેઠે ચલો બાર no you can actually release it if you're urgent you can not an issue just because it's a very કાલે મને ಅದು ಒಂದದ ಕೊಂಚ so let me I'll have a look but anyway if you want to get I'll ask Ramya Pernahari the patient is going to be કાલે ઉપાય દે ને પછી લેબ વિઝિટ હતી એટલે લેબમાં મન કે નતા ને મન કે મેડમ મોટા જાવ ને ડેલિગેટ્સ
ಅದು
મૂકવા ગઈ ને તો કે બહુ મેં કીધું 5 વાગે તો ઓછું જ હોય ને 5 વાગે હતા એટલે પેલો ટ્રાફિક ને વેરી નાઇસ ક્વાઇટ પણ દિલ્હી મુંબઈ વાળા હતા 2 જણા એટલે એ લોકોના પ્રમાણમાં તો ખબર એટલે એમાં કેવું કે બધા લોકો ગાડી લઈને એમ આવતા ને એટલે એમને એમ લાગે કે આવું કરી શકાય મુંબઈ વાળાને તો ખાસ લાગે દિલ્હી વાળા તો કદાચ ગાડીઓમાં એમને તો પેલું પડી જાય શોભામાં શું કહેવાય ગાડી વિના દિલ્હીમાં હા સ્ટેટસ હા ગાડીમાં મુંબઈ વાળાને એવું નહીં
have started
ये लोगों
therapy.
तो कार पेलो गाड़ी आ ट्रेन
Maybe
જ
it's not
બહુ
epithelial that much
એટલે
issue.
ફટાફટ જવાય.
Only thing is whether
કે ગાડી વપરાય જ
Sign
અહીંયા
not even, only
શ્રેયસ નહીં
cytoplasmic
લોકોને ઘરે
lesion.
ગાડી છે તો નીચે પાર્કિંગમાં
It's not
મૂકી
nuclear.
રાખે છે પાર્કિંગમાં એ કેટલી દૂર થઈ જાય તારે તો પાછા સાફ બાકી જ કાઢતા હોય એક લીધી ને વેચી દીધી વળી પાછી બીજી લીધી કે ના લઈએ
ನಮ್ಮ
લઈએ
ಇದರಲ್ಲಿ ಇದರಲ್ಲಿ ನಮ್ಮ ವಿಧೇಯದ ಪಣಿಟ್ಟುಕೋಮ we are not able to push
What is
further.
this?
They called me for this chairing and also moderating this molecular pathology conference here in Ahmedabad. While coming back, I'm just thinking maybe the whole conference. First talks in a workshop. I will come and discuss with you. But yeah, we'll come and discuss. Okay. આપણે નીકળાય ને પછી છેલ્લું જ લેક્ચર હશે પછી નીકળી ગયા
હા ખાલી થઈ ગયું છે અહીંયા છે ને બધા કામ કરવામાં બધા એન્થુ લોકો છે બરાબર
Hello. I'm Dr. Sandhya from Ramchandra Medical College, Chennai.
Chennai. I am Dr. Varsha from Unipath. Transplant
Unipath.
immunology.
Oh, okay, right. And
Transplant
what-
immunology.
Sumita Vausani from Transplant Immunology, Unipath.
Oh, wow, you're on the Unipath group, is it?
No, these guys are like
We are from IKDRC.
Acha, IKDRC.
Me and Dr. Varsha, when we are working
Acha.
together.
Yeah, y'all are from Gujarat?
Yeah.
Gujarat only, local.
Acha, local. Wow, okay, good. You know, great conference and very nicely organized.
Very
And
nicely.
Unipath has taken the forefront role,
Forefront,
right?
yes, but it's look like people are working, really working.
Yeah.
So right now I'm going to talk about the transplant immunology. This is going to be like a straightforward. Since
No,
morning
but that
I
is
was
very important. See,
very important.
Yeah,
Yeah,
we just
But
need what is happening on ground.
till now it's like lost and found. We did not found this, we found this. We did not
Okay,
find
that
this,
is
lost
also,
and found.
it's your experience.
That's also
yeah.
science,
Of course,
right?
Of course, everything is.
Yeah, but
And somebody is going to talk on prostate cancer genomics.
prostate
कोई और बात
like
है? आपका
somebody
पहला
after.
है
No, I'm going to talk
Talk
about
on
the
transplant.
role of transplant immunology lab.
मुंबई से थे मुंबई से थे डॉक्टर का नाम
हाँ,
था
कोई
डॉक्टर अपने नाम से
हाँ जी.
हाँ जी, आइए.
So who will introduce you?
You
Okay.
and only, you don't have choice.
I don't mind it also.
No, I think I'm co-chairing with him. He's
You?
Dr.
Okay,
Yeah,
so
they
हम
put me
लोग
as
ही
well.
बोलने
हाँ,
वाले
we
और
both
वो
are
ये.
co-chairs.
अच्छा, okay, fine.
So I am Dr. Sandhya from Ramchandra Medical College,
I see.
Chennai. I'm
Chennai,
the head.
yeah.
You are pathologist.
I'm a pathologist.
Working on what? Specialization?
So my specialty is GU pathology and breast pathology.
So gene
And
therapy
also,
you-
yes, and I do molecular pathology in my college.
Okay.
Yeah, I'm there for many years.
So gene therapy, you are doing like a looking at
Not gene therapy really, but of course a lot of oncology.
lot of
Yeah, lot of
oncology. Yeah, it's all about oncology.
Yeah.
Everything has been developed in the oncology only.
Yeah, because I think
So
that's where
major
the,
is oncology. There may be the other field, but this is
But
like
यहाँ पे projection
very
है।
prominent.
वो तो है।
So fine, I am Dr. Mandot and I am a nephrologist.
Okay. Oh, you're a
And
nephrologist, sir?
we had worked
Oh,
together
wonderful,
at
sir.
Kidney Institute.
I
That
can see
also in the
that.
initial years where everything
I know,
was
sir.
evolving
Yeah,
and
nephrology
developing.
and
Yeah.
pathology, I think we all cannot do without pathology and we cannot do without clinical input.
It's very important to interact with the. See, problem वही आता है कि जब when we are not interacting.
Yeah.
So if we are interacting with each other, then
So we run
can
a
get like a good
nephropathology
solution.
fellowship, Ramchandra. We run a 6 month. It's quite popular. So we have.
Who is a nephrologist in the?
So nephrologists we have plenty. We have.
Like somebody from our institute is there.
Really? Dr. Jay Kumar is a very well known person.
No.
He is there. Then Dr. Indumati is there. Maybe somebody was there. You know. Or Dr. Shroff, are you talking about? They keep coming in.
I think बीच में चेन्नई
हाँ चेन्नई में कोई
नारायण जाता था
नारायण
But I'm
जाता
not-
था ना
he probably institute I'm not remembering.
ओके नारायण का बीच में फोन आया था एक बार
वेलू भी मिला था
हाँ वेलू इज डूइंग लाइक पूरा वन मैन शो कर रहा है हाँ एनेस्टोमोसिस ही टोल्ड मी आई डू एवरीथिंग मैडम आई डू एवरीथिंग
ये वही करता है सर्जन का तो नाम रखा है
फिर ही स्टार्टेड फ्रॉम द डायलिसिस एनेस्टोमोसिस वो पिछला बनाना ये करना वहाँ से स्टार्ट किया उनने नाउ ही हैज रीच्ड टू द सर्जरी आई डोंट नो हाउ ही इज मैनेजिंग
I'm not trained. Even though I developed the skills,
Skills,
that doesn't
yes.
mean I can do it.
इवन इन दोज डेज डॉक्टर सूर्यकांत करते थे बेबी यूज्ड टू से दैट यू नो दिस हॉस्पिटल तो पीछे था बोलते थे
वो ठीक है
पर उस जमाने में कोई था नहीं बट वी वर लाइक ओह जनरल सर्जन कैसे कर सकते हैं
दूसरी बात कर R&D मेडिकल कॉलेज उदयपुर वाला उनके जो फर्स्ट प्रिंसिपल थे ही वाज बेसिकली कार्डियक सर्जन सब कुछ ऐसा बट इफ यू आस्क फॉर द डिग्री ही वाज MBBS एंड देन जो छोटी-मोटी फेलोशिप्स करने के बाद वो डूइंग कार्डियक सर्जरी
राइट
ये सब तो ठीक है उस जमाने में
उस जमाने में हाँ सर वो व्हेन माय सिस्टर वाज डूइंग द लाइक MBBS के बाद कोई बढ़ता नहीं देन उनका PHC का जब भी वो पोस्टिंग आता था तो शॉर्ट एनेस्थीसिया उनको देने का सर्जरी उनको करने का स्टिच करने तो उस टाइम कर लेते थे अभी इंटर्न को कि कुछ नहीं आता है
नहीं आई वाज असिस्टेंट माइसेल्फ ऑल दिस एक दिन फर्स्ट डे तैयार शी भाई तो कहते राज क्या करेंगे तुम हेल्प कराएगा छोटी मोटी मैंने कहा ठीक है मैं करा देता हूँ सर
वी लर्न अ लॉट
है
काफ़ी सारी चीज़ें सीखी
नहीं वो है करने के बाद में कहते कि राज तेरा तो हाथ बहुत अच्छा है मैंने पहले भी बहुत असिस्ट करा है बट मैं बोलता नहीं हूँ मेरे को थोड़ा ही काम करना है वो तो ठीक है यू कैन लर्न बट यू कांट प्रैक्टिस
वी हैव टू हैव लिमिटेशन ये चीज़ अभी ये ज़माना आ गया है कि कंसंट्रेट ऑन वन थिंग तो जब ये शुरू करते हैं पेरिफेरी में कोई आने को नहीं होता है यू डोंट हैव सपोर्ट देन यू हैव टू डू दैट थिंग्स
कोयम्बटूर ऐसा नहीं है इट्स यू कैन से बड़ौदा राजकोट से सीखते
ही जी ने
नहीं पौंडीचेरी में नारायण वेलू कोयम्बटूर
कोयम्बतूर तो बड़ा है कोयम्बतूर आई वाज अंडर इंप्रेशन ही वाज इन सम पेरिफेरी
नारायण भी पेरिफेरी में नहीं पौंडीचेरी में
कोयम्बतूर में तो लैब सब कुछ है
बट स्टिल वो बताया कि सी दिस दैट स्मॉल हॉस्पिटल उसमें सब कुछ करेगा और स्मॉल में ही कर सकता बड़े में नहीं कर सकता
कोयम्बतूर इज अ बिगर प्लेस वहाँ तो है दूसरे सेंटर भी है आपका कहाँ है
हमने सब कुछ लीज पे दे के है जॉइंट शेयर
शर्बी यू हैव जॉइंड शेयर ओह
2 साल के लिए
गुरुजी के साथ होंगे
वही सबसे बड़ा प्रॉब्लम है
गुरुजी
मेरे को
के साथ
पता
होंगे,
नहीं मेरे को पता ही नहीं था
नहीं पता था आपको
आई वाज नॉट
गुरुजी है
ठीक है अब चल रहा है
वो
आई वांटेड अ ब्रेक
बहुत किया अभी हाँ यू एट सम पॉइंट यू हैव टू स्टे क्योंकि
देखो
पर वहाँ इंफ्रास्ट्रक्चर है सब ट्रांसप्लांट किया तो देखो विल लुक आफ्टर वो चाहिए अभी रेजिडेंस की तरह नहीं कर सकते
रेजिडेंट की तरह भी नहीं है एंड देन फाइनेंशियली जब इश्यूज ही नहीं रहे कुछ भी तो वही वही पूछा मेरे को वही छोड़ना पड़ा हमारे को बड़ा फोर्स करके छोड़ा है हॉस्पिटल
Your wife is a radiologist, right? where she is working?
She's sunflower.
Sunflower. Mayflower और sunflower दोनों अलग हो गए हैं. Long back. So capital. वो चारों लीलाबेन के स्टूडेंट थे. गायनेकोलॉजिस्ट संजय भाई और जाडेजा और सब. All four मेरे elder sisters. उनके स्टूडेंट थे. अच्छा बनाया था हॉस्पिटल when they like inauguration हुआ ना तो लाइक वैसा गायने का कोई नहीं था उस टाइम पे नहीं था. अभी तो सब जगह पे क्या पैसे दो और 5 स्टार में रहो.
आपने इनको ज्वाइन कर लिया क्या करो?
करती है ना डिपार्टमेंट पूरा डेवलप करती है. इट्स ऑलरेडी
आईकेडी में कौन?
आईकेडी में मैं हूँ ना. ये लोग दोनों आईकेडी में भी है. हिमांशु भाई साइन करते हैं. हाँ वो बेचारे फंस गए अभी.
वो सब जगह स्टॉप गैप की जैसे यूज कर लेते हैं आप लोग.
अभी they are going to move to the like a new hospital. बनाया है बहुत बनाया है उसको अच्छे से डेवलप करना चाहिए. रिसर्च चाहिए. 9 crore.
तो वो तो फिर दिन आप
7th ना थी. 9
टोटल हॉस्पिटल है
टोटल 9 છે પણ આપણી લેબ તો 7th.
આપણી લેબ 7 ઉપર ઓટી છે.
આપણી नहीं होगा તમે.
ના ના આપણી તમે શરૂ કરી છે એટલે
हाँ.
देखो टेक्नीशियन लोन विल देता वो चीज आईकेडी को समझिए.
अभी तो क्लिनिशियन भी नहीं चला पाते.
नहीं आपने क्लिनिशियन कैन डेवलप. मैंने चलाने की बात की. चलाने के लिए आई वुड से कि मैनेजमेंट का बंदा अलग से ले आओ चल जाएगा. तो जो सिस्टम अभी चल रहा है उसमें नहीं चल.
Everybody believes that. Like a पूरे हॉस्पिटल वाले मानते हैं. This way we cannot.
कितनी फालतू की बातें आती हैं आप बताइए.
Why to waste time in all other things? Development में आपका दिमाग लगाओ.
अपने डेवलपमेंट में लगा रहे हैं दिमाग.
हाँ डेवलपमेंट लगा रहे हैं पर कौन सा डेवलपमेंट राइट. फुल टाइम आप ज्वाइन करती है सर्दी में.
अभी क्या है 2 साल 10 to 5 काम करना है फिर बाद में से
नहीं करना है. फिर और वहाँ तो क्या है कि अब भले बकरी भाई है बकरी इज देयर. हाँ कुछ फर्क नहीं पड़ता है. आपको तो क्लिनिकल कोई नहीं. आपके नाम के पेशेंट आएगा आपको और डॉक्टर शिमलानी भी है.
और कोई नहीं. पंकज भाई बाकी सब निकल गए.
शिमलानी भी निकल गए. अब तक तो था वो
सब निकल गए. पंकज भाई को लिया. मेरे को आइडिया नहीं था. इन्होंने मेरे से बात की. मैंने मना कर दिया.
अच्छा.
फिर हमने लीव पे देने की हॉस्पिटल किया.
हाँ.
तो कीर्तिपाल विसाना साहब उनके वहाँ पे गए. तो डॉक्टर विसाना ने उनको बोला. फिर मेरे को बोला कहता क्या करना है. कुछ नहीं करना है. आराम से बैठ. सर ने वहाँ पे बोला था फिर
नहीं पर he is not going to like वो आगे भी नहीं जाएंगे पीछे भी नहीं जाएंगे. आपको कुछ बोलेंगे भी. हाँ
वो कभी बहुत आपको तो पता ही है. अपने को पता है. अभी लोगों को नहीं पता है तो
नहीं सब मैं तो बहुत अच्छे से जानती. वो रेसिडेंट थे तब से है ना.
यही बोल रहा हूँ ना. वो अब उसका
वो गेरा सर से तो अभी भी सब बातें होती है. पंकज भाई तो हम लोग बाजू बाजू में रहते थे ना. तो गाड़ी नहीं लाते थे तो तभी जाते जाते सब मैंने आपने इतना क्लिनिकल काम क्यों छोड़ दिया? Why? How can you ask me a question? मैंने अब फिर थोड़ा अच्छा नहीं है. You need to be in a clinical.
पर अब क्या है कि मैं ऐसा मानता हूँ, मेरे से पूछा, मैंने कहा यार आपका चॉइस गलत था. वंस ही इज रिटायर्ड फ्रॉम एवरीवेयर, तो आपको लेना ही नहीं चाहिए. तो आप मेरे से डिस्कस कर रहे हो, आप ये हमारे इश्यूज हो रहे हैं, डिपार्टमेंट को तुम बैठ गए, तो आपने चॉइस ही रखी है, उसका कुछ नहीं कर सकते.
अभी बहुत वो तो मुझे तो ऐसा बोल रहा था कि वी वांट टू ट्रांसप्लांट सेंटर बनाने वाले हैं, मल्टीपल ट्रांसप्लांट मींस नॉट
मेरा
ओनली किडनी.
नहीं, वो तो मैंने ड्रीम दिया इसलिए कर
अच्छा
रहे हैं
आपने दिया है.
इसलिए मेरे को लिया. आई हैव लॉट ऑफ लिबर्टीज इन अदर थिंग्स.
So that's okay. That will be like हर जगह पे थोड़े लोग ऐसे तो
चलता कुछ भी नहीं. आई स्टार्टेड डेवलपिंग दिन का लोड़ा से इस मंथ. सो उसके बाद में वी डिसाइडेड टू टेक सूरत.
Oh.
So we'll take one by one. मैंने बोला प्रोजेक्ट में ये न्यूसेंस वैल्यू रहेगी, ये सबसे बड़ी न्यूसेंस वैल्यू है. करते कुछ नहीं,
अब बहुत
बोलते
भागोगे
रहते
तो थोड़ा ब्रेक लगाएंगे ना. आप आराम करने गए थोड़ा ब्रेक लगाएंगे.
वो तो सही.
ब्लेसिंग
नहीं वो
मैं बोल रही हूँ.
नहीं मैं तो उन सब में पढ़ता ही नहीं. दे हैव गिवन मी काफी राइट्स एंड पैरेलल सिस्टम मेरे लिए खड़ा किया हुआ, उसको यूज करते रहते हैं.
कुछ न कुछ तो करना है, नया तो करना है. अपना नहीं है पढ़ने. You have to have desire. तो अभी तो एक्सपीरियंस यूज करना है. At certain level you have to use your experience. इसलिए मैंने ये enjoy किया सब कि इसलिए किया कि there was a new thing, वो चैलेंज भी था कि नया करना है. और the guys
बहुत आगे भाई.
मैं इंटरनेट करना चाहती हूँ. शी इज इन एलजी. किसकी? संदीप सुप्रटेक.
नहीं आप किसमें ज्वाइन किया?
मैंने यूनिपेट.
यूनिपेट में फिर यूनिपेट से आपने सुप्रटेक बोला तो कंफ्यूज हो गया.
नहीं सुप्रटेक में तो वो उसको भी जानते हैं. संदीप को और उसकी वाइफ तो मीन्स शी वाज लाइक इंडिविजुअल.
यूनिपेट का पोटेंशियल ज़्यादा है सुप्रटेक.
अभी तो मैंने तो उसी टाइम पे बोला था कि भाई अभी तो
नहीं आज प्लानिंग टू कम अप विद वो कैसे उसको ज्वाइन करेंगे वो तो है श्योर. बट इंटास इज प्लानिंग टू कम अप विद 1000 euros. So if that counts, they have to join all the jobs.
They have taken a big lap in molecular. Means they have bought like 5 engineers. First latest.
You put it how did you join all the मेरे हिसाब से they should join all the jobs. I was with Intas. I know it.
Oh, you were acting out.
I was the medical advisor.
अच्छा. Okay.
So I know everything and I know how actually the first part was developed by me for.
Okay.
So उनका लैम्बडा भी है. मेरे को हमने कुछ चीज़ें ऐसी की जो बहुत कम लोगों को पता, मेरे को पता है. कभी बार फोन किया तो कहते हैं सर आपके में ये बाकी लोग भी हमने भी उनके तरह किया. वही कह रहे हैं कि they have to join all the jobs.
Dr. Raj Mandor is here.
Yeah. I'm here.
Dr. Sandhya.
બોલવા વાળા છે બધા અહીંયા. સાંભળવા વાળા જ જમવાના છે.
ચાલો.
I think you can make an announcement. Lunch area में कि the session is going to start shortly.
Yeah, somebody should make an announcement.
A session is
The session has just started.
नहीं वो वहाँ पे नीचे बोलोगे तो लोग बाहर आएंगे.
So we will just wait for next, I think 5 minutes, and then we will start the sessions. I have already asked to make an announcement in the exhibition area and lunch area, so that all the delegates who wants to listen will join us.
અમારી બેનપણીઓ આવી ગઈ છે બાકી બધા શોપિંગમાં જવાનું.
So actually we will be trying to match up with the delays. So with that, all the speakers will be given sharp 20 minutes of time. Okay, so that's great. Chairpersons has to take care of these things, right? So that 15 minutes bell will be there. If you want, I will ask them to add, one additional 3 minutes. I mean, after 18 minutes also they can press a bell. So you will have 18 minutes. We will try to finish if all the speakers will allow, because 3 minutes delay if all the speakers, what I have observed from the morning, 2 to 3 minutes everyone is taking, and in general like 21 presentations are there, so 21 into 363 minutes are already extra. Question answer is already we have skipped, so we will be taking, I cannot now say like it will be taken in the next tea break, because tea break is again overlapped. So you can take the tea from there and you can come back and join us here. So we will not be taking a tea break now. So this after the session, which will be soon starting, there will be no breaks till the evening, because we will have the general body meeting at the end of all the sessions. So we will not have time for that. So that's why we are a little bit running up. So welcome everyone for the 5th session on transplant research part 2, and now I invite the chairperson Dr. Raj Mandot, consultant nephrologist and transplant physician, Selby Multi-Speciality Hospitals, Dr. Sandhya Sundaram, professor and head pathology at SRMC. So please come on stage and raise the chairs. Sir and ma'am, over to you. So I have already requested all the speakers to finish your talks within stipulated time. So let's start this session. So over to you, sir and ma'am.
A very good afternoon, and I hope the hall slowly fills up. Otherwise, it's just for the chairpersons, I think. I'm sure they're coming over. So welcome, and it's my great pleasure to be here, and thank you to Unipath and to MPI for this invitation. May I request the first speaker to take the stage, and sir, if you have a couple of...
Good afternoon, everybody. I'll be very short. All I can say, I had the honor to work with Dr. Varsha Trivedi at IKDRC, and those were the initial days for transplant program, and we did develop together everything. So, over to the speaker.
Thank you for the I think I'm audible, right? Yeah. Okay. So I can take a liberty. Thank you very much for the invitation for this talk. And today's my topic is role of transplant immunology laboratory in transplantation. So,
बंटी
okay, so everyone in this world know that Lord Ganesha has a successful xenotransplantation. And now to start with the subject is what is transplant? The transfer of the tissue or the organ from one part of the body to the another part, or the transfer of the tissue from the tissue or organ from one individual to the another individual. So the types of the transplant organs are the autograft, isograft, allograft, and xenograft. So autografts is in the same individual from one side to the another side. Isograft is two identical.
Shall I start again?
So to start with the autograft, isograft, allograft and xenograft. Autograft is like a self-tissue transfer from one side to the another side. Isograft is a transfer of the tissue from one identical two individual, and allograft is from the same species but not the genetically identical. Xenograft is from the two different species. Now this is the schematic diagram of the all types of the graft. Nowadays, like today, the kidney, many organs are being transplanted successfully, like kidney, pancreas, heart, lung, liver, bone marrow, hand, intestine, face, cornea. These transplantations are done on the regular basis with the increasing frequency and success. So, the role of transplant immunology, what is the role of how a transplant immunology lab can help to the transplantation? Transplantation itself is like multiple teams are working together in a coordination. So the role of HLA typing lab is to do the HLA typing of patient and donor, detecting the post and pre-transplant, from circulating HLA antibodies, pre and post-transplant immune monitoring to guide the requirement of immunosuppressive medication. So how the transplant program come into the India? So in 2004, the review committee was set up to regulate the transplant program. And the law was introduced, the guidelines was introduced to the government, and every transplant center will follow those guidelines. In this very important part is that you can have the living donor only from the family, not the unrelated donor you can use. There are some conditions, there was an amendment, and we will talk later on about that. Here you can see the waiting list about so lakhs and lakhs of people are waiting for the organs in the world and in India also. So we need to increase the donor pool. That is the angle. There are challenges in the transplant program. That's there is a severe scarcity of the donor pool. There is a challenge into the other area also, how to maintain the immunosuppressive. We don't have any tool to say yes or no. We have to have different kind of data and analyze them, and how to use those information in a clinical side. So how many types of the donor? You can have a living donor or you can have a diseased donor. Like in the living donor, you have to pass through the review board committee. If they give you as a donor permission that your donor is medically fit and related to the patient, then and then only they give the permission, and then you can go ahead. So the medical legal aspect is also involved in the transplantation. So the first modality to increase the transplant pool is ABO incompatible, which is now done in the regular basis. Initially it was not a so good success, but now it's successfully done. You need to bring down mismatch ABO antibodies, bring down at the acceptable level, and you can go ahead with the transplantation. The second modality is a swab transplant. So one pair of patient donor is incompatible, either ABO or sensitized patient. Another pair has the same problem with the patient and donor. Then you can swap the donor if they are the compatible pair, and you can go ahead with the transplantation. This donor pool kind of swap transplant can be make a cluster up to the hundreds to find a good match, compatible donor, and age compatible also. You can have multiple factors considering the find a donor and recipient pair, and you can have the better outcome if you have this kind of the donor and patient compatibility. Third modality is if the patient is sensitized and have a low level of antibody, or if you treat them medically, and the patient is responding, and if you can bring down the HLA antibodies up to the level that they are acceptable for the transplantation, then you can go ahead with the transplantation and do the transplant successfully. The fourth modality is to increase the donor pool, is to enroll, sensitize all the people who are willing to donate their organs if something happens to them. That will also increase the donor pool. Now coming a little bit about the transplant immunology. So HLA plays a very important role into that. Actually it's a protective, antigens are protecting to us, we don't get infections with that. And here HLA stands for the human leukocyte antigen. Bunch of the genes on chromosome 6 on MHC complex are called the HLA antigens. Expression of these HLA antigens are different on all the nucleated cells. As for example, the T cells has a maximum level of the expression for the class 1, whereas the class 2 expression is less. So forth, so on. Here in this diagram you can see that chromosome 6, and here you can see that this is the chromosome 6 centromere, and here this is the MHC complex. This MHC complex is divided into the 3 groups, class 1, class 2 and class 3. HLA A, B and C for the class 1 and HLA DR, DQ and DP for the class 2 are relevant into the transplantation. Coming to the molecular structure of the class 1 antigens. Class 1 antigen has heavy chains like alpha 1, alpha 2 and aspartate, which is supported by the beta 2 methylglobulin. And class 3, alpha 3 domain is attached to the CD8 cells, and here you can see the peptide binding site of this antigen. Class 2 has alpha 1, alpha 2 and beta 1, beta 2 chains, and alpha 2 and beta 2 chains are binding to the CD4 cells. Here also this notch is also the peptide binding site where the antigen is binding. So here the top view of the antigen binding site. The polymorphism is focused around the peptide binding region. Every individual's HLA haplotypes allows the presentation of the different repertoires of the peptide T cells. So polymorphism. The HLA antigens are highly polymorphic. They are in thousands. Alleles are being identified. Also the proteins are being identified. This has made HLA typing more complicated and challenging. The World Bank data has to come up with a nomenclature, because thousands of how to name these thousands of the HLA antigens. So here is the guidelines. Here you can see the HLA is a prefix. Here A is a gene. This 2 digit is a field 1, which will show the allele group, and field 2 is a specific for HLA protein. Allele field 3 is used to show the synonymous DNA substitution within the coding region. Field 4 is used to show the differences into the non-coding region. Now coming to the transplantation, the patient and donor. If the patient HLA is A1, A3, B8, B52, and donor is A1, A2, B7, B8. In post-transplant scenario, there are chances that the patient will develop the donor-specific antibodies against A2 and B7. But here in this figure, you can see that along with the A2, B7, we see many more antibodies due to the shared epitopes with the donor HLA. So what is triggering this kind of immune response? This is called the sensitization. Why the sensitization occurring to the patients? Because of the pregnancy in the female patient, blood transfusions, transplantation, bacterial and viral infections are causing the sensitization. This kind of immune response is highly specific recognition of the foreign antigens. Transplant rejection occurs as a result of the anti-MHC immune response. Mechanism for the elimination of the microbes bearing such antigens. A vast universe of the distinct antigenic species and immunologic memory and tolerance. This kind of immune response is a very robust immune response we will find. And in this kind of immune response, there will be a different type of the cells taking part. Antigen presenting cell, B cell and antibodies, T cells and other cells like NK cells, T cells, express NK cell, monocyte and macrophages. Now the antigen presentation. Antigen presentation by the antigen presenting cell is done directly or indirectly to the CD4 and CD8 cells for the class 1 and class 2. If the antigen is presented to the B lymphocyte, then there will be a antibody response. And this antibody response, if it is a primary antibody response, then there will be a low level of antibody and slow response. But when there is a second time immune response exposure to that same antigen, then there will be a secondary response, prompt and high level of the antibody will found. And in the transplantation, this will lead to the hyperacute rejection, which can occur in the post-transplant scenario in minutes or hours. Acute rejection can occur in days, and chronic rejection case takes more time. So why the chronic rejection? Chronic rejection over the period of time, like 20% of the patient lose their graft, transplanted graft, in the 5 years post-transplant because of the chronic rejection. And 35 to 40% of the patient lose their graft after 10 years. So we need, this is a severe problem, how to protect those graft. So this chronic rejection is why it's causing. Because of the previous episode of the acute rejections, there is a high number of the HLA mismatch antigens with the recipient, and the patient on inadequate immunosuppression and primary disease of the host. So coming to the medical management, in the post-transplant scenario, medical management is done by the clinicians using the immunosuppressive different kind of drugs. I'm not going to go into the detail. Now the transplant immunology lab workflow. So when the donor gets a permission to donate the organs, they are being referred to the transplant immunology lab, and we do the HLA genotyping of the patient and donor and antibody screening. So the recommendation is to do the HLA typing when they first come to transplant immunology lab. HLA typing of patient and donor, LCM, flow cross-match and single antigen test to be done. In post-transplant scenario, maybe the donor is if available, then you can do the flow cytometric cross-match. Otherwise, in single antigen, only you need is a patient serum. Renal function test, complete blood count, and if needed, then the biopsy is performed. If the patient is enrolled for the cadaveric donor, then at the time of the enrollment, HLA typing is done, panel reactive antibodies is being done, and then the antibody screening every 6 months till they get the transplantation. At the time of transplantation, donor HLA typing is done, lymphocyte cross-match and flow cytometric cross-match is done. In post-transplant scenario, the patient has to be monitored for the HLA antibody to prevent the, to pick up the early rejection. So in the first week, then the 2-week interval, 4-week interval, 8-week interval, so on has to be monitored according to if the patient is stable, you need not to screen them so frequently. Why the antibody screening is important into the post-transplant scenario? Because this is the first indicator you can see. This is the gray area where we don't have right now any test in the market, but people are working in that. Soon we will have that test. Here you can see the first indicator is the donor-specific antibody. So you can pick up the rejection early if the donor-specific antibody, there is a alert that the patient may suffer rejection. So you can address that problem or monitor those patients closely. Now coming to the HLA typing method, serology, cellular and molecular method. The molecular method is more accepted in most of the lab right now. So the serology lab typing has become absolute right now. And you can see here the molecular typing, SSP, SSOP, RSSO and sequencing. Sequencing is done mostly into the patients who are going to work for the bone marrow transplant. But for the solid organ transplant, 4-digit medium intermediate typing is okay. If you have 4-digit typing, you can go with the virtual cross-match also. So it's okay to have medium resolution typing. Here by the Luminex HLA SSO typing, here you get the 4-digit up to 6-digit also you can get the typing in Luminex platform. This is a sequence-based typing. You can compare the sequence database with your testing. This is a known phenomenon that's HLA matched transplant has a better outcome than the mismatched. So now the antibody screening. In 1969, Dr. Terasaki found that, published a paper that antibodies are responsible for the hyperacute rejection. Subsequently, many more methods are being introduced for the antibody detection testing. Those are the CDC cross-match and flow cytometry cross-match are the real cell-based assays. So we need a living cell in those, donor's living cell in those things. But here in the other area, you need just a serum and you use as an antigen, you use coated beads. There will be, if you are doing this many tests, there will be this kind of scenario, and you need to know how the test is working and what the information you are getting from the test, whatever you have advised. And these tests are being analyzed according to sensitivity and specificity of the test. So in the serology, with the density gradient, we separate the lymphocyte incubated with the serum and then followed by the complement addition, and then after incubation staining is done. And scoring is done under phase contrast microscope. Guidelines followed by the ASHI, that's a live and dead cells are scored and test is analyzed. In flow cytometry cross-match, lymphocytes are incubated with the serum, and then the incubation and followed by the washes, and then IgG, FITC, CD19, CD3 is added, followed by the incubation and wash, and then the test is ready to read under the flow cytometric cross-match. And these are the gating of the T cells and B cell, and the test is being analyzed considering the negative and positive control, and mean channel shift is found into the test. Those patients who had flow cytometric cross-match positive, they are more prone to suffer a post-transplant rejection. Now coming to the Luminex platform. Luminex platform has changed the scenario for the approach for the transplantation into from the lab. There are different tests available on the Luminex platform. It's the antibody screening, then PRA, then you can have the HLA antibodies with the MFI and specificity. So here you can see that this is a MFI and this is the specificity of the test. Here you can see that the D 9, 14, 30, 53, 9 and 7. They are the mismatch for the donor, mismatch to the patient. This is a donor typing. So this is a mismatch, and here you see those antibodies has developed in the post-transplant scenario. These are called the donor-specific antibody, and they are harmful to the transplanted graft. Here, this data, HLA typing and antibody profile, you can use into the post-transplant. whenever you are doing the virtual cross-match. In that, you are just analyzing the data, whatever the information you have. You need not to do the real cross-match or real thing. If the patient has a HLA antibody and donor has a HLA antigen, HLA-A2 antigen, this is called a positive virtual cross-match. So you can predict the flow cytometry, predict the cross-match. So the advantage of the virtual cross-matches are eliminates the physical cross-match, saves 6 to 8 hours to cut the donor cold ischemia time, no sample is required, reduce the laboratory workload and transplant organ cost, transplant program cost, and precision to the actual cross-match. CDC and flow cross-match are the predictable, and DSA identification is there, improves the allocation efficacy and increase the rate of transplantation for sensitized patient. Risk of the memory response can be accounted because you never know the second transplant or third person or the pregnancy, low level of antibody may not, you may neglect that part. Also, in the patient who are having absence of the donor specific antibody and still histopathology shows that there is a rejection, then one should look for the non-HLA antibodies. For the second. Now review literature. Here you can see that those patients who are responding to the desensitization protocol has a better survival than those who are not responding to the treatment. High level of MFI or high level of the antibodies are detrimental to the graft outcome. Complement fixing antibodies are also detrimental to the graft outcome. Now in conclusion, I would like to say that solid phase and flow cytometric single antigen bead assays offer a greater sensitivity and specificity for the HLA antibody detection. These high resolution tests allow the patients to be listed for the transplant by the virtual cross-match, thereby increasing the donor pool. The solid phase C1q binding assay further distinguishes the HLA antibodies that can bind the first component of the complement and may further develop to expand the donor pool by the identifying the most pathogenic antibodies. Thank you very much for the attention. I will be happy to take questions.
Thank you, madam, for that very nice presentation. I think we have a time. Yeah. We'll go on to the next one. Thank you, madam. We'll take the questions during tea time. Thank you once again. I think may I invite the next speaker on stage. Yeah, Dr. Akshita Gujar. So, Dr. Akshita is Senior Scientist at Novartis Institute of Biomedical Research, Cambridge, USA. And I think she's coming online and she's talking about gene therapy for retinal dystrophy, a success story. Can we hear her? Yeah,
so this session is virtual. Session is virtual, so we will start it again from the start. So you can start.
Advances in gene therapy. I'm a senior scientist in the gene therapy group at Novartis Institutes for Biomedical Research in Cambridge, USA. And I'm very happy to be a part of this conference. Thank you to the organizing committee for this opportunity. I just want to go over the evolution of gene therapy. So I just want to mention specifically about the adeno-associated viruses, and in a few slides I will explain more about those. They were discovered in 1965, and ever since a lot of people have tried experimenting with gene therapy, and in the 1990s, during one of the clinical trials, an 18-year-old boy lost his life, and this was a huge setback for gene therapy, and it slowed down gene therapy research a lot. But the scientists continued researching different new ways to treat genetic diseases, and finally, in 2012, the first gene therapy was recommended for approval by the European Medicines Agency. And in 2017, a few more gene therapy drugs were approved in the US. So, what is gene therapy? So, the definition of gene therapy is an innovative treatment approach for monogenic diseases or overexpression of a single gene. So, it basically introduces genetic material into a target cell that has a defective gene, and it helps in treatment by preventing the genetic disease through an expression of a functional protein, so a functional normal protein. And there are different approaches to gene therapy. So one is the basic gene transfer, where the function of a dysfunctional or the missing gene is replaced with a functional transgene. And this can be applied to a lot of monogenic diseases like sickle cell disease, a lot of retinal dystrophy disease, neurodegenerative diseases, Huntington's disease. And another approach is the gene augmentation, where there is an addition of a therapeutic gene, and this often is used to supplement a targeted therapy. And this can be applied to different infectious diseases. Gene inhibition is another approach where silencing the expression of a mutant gene is achieved by that codes for a toxic protein, and this can be applied to a lot of cancer therapies. Gene editing is another approach where the patient's genome is edited to correct a mutant gene that promotes disease, and this can be applied to various monogenic diseases and cancers. And CRISPR-Cas9 editing is very popularly used for this method. So there are different types of viral vectors utilized for gene therapy delivery. So why viral vectors? So viruses are known to naturally infect cells and transfer their genetic material to the cells. And so these are like the best vehicles for a treatment. And there can be like they are in two different categories. So there is an integrating vectors and non-integrating vectors, which means that the integrating vectors, when they transfer their genetic material, the genetic material becomes a part of the host genome's material. So that way it's carried on from one cell to another when it divides. And examples for these viruses are the retroviruses and the lentiviruses. And since the dividing cells carry it, the main advantage for these viruses is that you have a sustained gene transfer. But at the same time, there could be the limitation is that because it's integrated into the host genome, in some applications, and depending on the disease and the target cells, you can have oncogenesis in some cases. For the non-integrating vectors, the examples that have been used in the market is the HSV1 virus and the adenovirus. So these have large packaging capacities, which is a huge advantage of these viruses, and they're very efficient in transduction of most tissues. The main limitation here, though, is that they induce a very potent inflammatory response. And then another non-integrating vector is AAV, which is adeno-associated virus. And this virus was actually accidentally discovered as a contaminant in an adenovirus sample. And these are designed to be non-pathogenic, so they do not cause any disease in the humans. And they're replication defective, so they can't replicate on their own. They need either a helper virus or an adenovirus in order to replicate. And they don't induce very strong immune response. The limitation for this virus is that they have a very small packaging capacity, and this limits the range of therapeutic genes and thereby limiting the diseases that you can target. So an AAV vector capsid sequence is the one that determines the serotype of AAV, and there are several serotypes that were discovered, ranging from 1 all the way to AAV9, AAV10 even. And in the recent years, actually, people have been working on generating their own capsid sequences by library reconstruction and designing and cloning different components of the capsid in order to target specific cells. And so these different serotypes target different parts of the body. So there are ones that are specific for the eyes, liver, the muscle, heart, and brain. So depending on your disease and your target cell, you can choose the serotype of AAV to work with. And here, I'm going to talk a little bit more about retinitis punctata albicans. This was my first gene therapy project in Novartis, so it's very close to my heart. And this disease is an autosomal recessive orphan disease, and there were only a few hundred patients, 100 to 200 patients identified so far. And this is caused by a dysfunction in cellular retinaldehyde binding protein, termed as CRAB, due to mutations in the RLBP1 gene. CRAB is expressed in the RPE cells, so the retinal pigment epithelium, and it helps to produce the visual pigment for the rods. And rods help us to see in the dark. So when CRAB is dysfunctional, it leads to night blindness in these patients at an early age, at 3 to 4 years, and followed by progressive loss of cone function, leading to macular degeneration and legal blindness by adulthood. And the schematic that you see up here is the fundus image of a patient, and they show the classic punctate white dots throughout their fundus. So for this disease, we decided to use the AAV as a gene delivery mechanism, and this schematic shows the different components of an AAV. So it has inverted terminal repeats, termed as ITR, on either ends of the AAV that come together and form the capsid. And it also has, you can have an enhancer for your promoter, and the promoter drives the transgene expression, which in this case is the RLBP1 cDNA. And this is followed by the poly A sequence that helps in transcription at the RNA level. So for this project, we tested multiple serotypes for AAV, and composing of different promoter sequences, different genome conformations, and capsid serotypes. And finally, the AAV8 capsid was chosen with an RLBP1 promoter expressing the human RLBP1 gene. And this was subretinally injected in mice that are knocked out for the RLBP1 gene. And we saw an improvement in the rate of dark adaptation based on ERGs, which are electroretinograms, which are recordings of visual activity in the eye. And we saw this effect. We saw that this effect was present in the mice for even after 1 year, which is a long time. If you relatively look at the age of a mice, and we were very happy to see this. And this is being currently tested in the clinic. So we started working on this project in 2010. And the optimization and characterization of the AAV happened by around 2013, and when this drug went into the clinic in 2016. This is a subretinal injection where the drug is introduced between the RPE and the photoreceptor cells, because our target cells are RPE. And patients have noted subjective improvements in their symptoms and activities in daily living. And we hope that this is going to be a one-time therapy, and the patients are being monitored long-term. So typically what happens when gene therapy is applied to the eye is that only one eye is treated at a time, and the worst eye is treated first. And once we see an improvement, then the second eye is treated. So we hope that someday this drug will be in the market and will be very beneficial for all the patients out there. Another gene therapy drug that I want to talk about is for RPE65 associated retinal dystrophy. And this treats patients with biallelic RPE65 mutation. This drug is called Luxturna, and this is an AAV2 vector that expresses the human RPE65 gene. So here patients experience a progressive loss of vision due to night blindness, due to several actually impairments, including night blindness, loss of light sensitivity, impaired dark adaptation, or loss of clarity of vision. And this happens very early in their age. So, and the way Luxturna works is that it expresses a working RPE65 gene. So RPE65 is involved in the visual cycle, and it basically switches on the visual cycle in the light and switches off the visual cycle in the darkness. So in this drug, this is packaged, RPE65 is packaged into an AAV2 vector, and this is also given as a subretinal injection in the patients. And what was noted was that the patients had improved photoreceptor function, and this further improved their vision. Another gene therapy drug that Novartis was working on is for the spinal muscular atrophy, SMA. And this is caused by biallelic mutations of the survival motor neuron, SMN1 gene. And this affects children less than 2 years old. And it's a genetic disorder that starts in the central nervous system and affects all the muscles in the body. So these children, by the time they reach 2 years, they don't actually reach 2 to 3 years go beyond 2 to 3 years of age. And they have really poor motor skills and muscle movement. And this drug is given as an infusion for babies as little as 1 month old. And it's an infusion for over 60 minutes. And the drug provides a working copy of the SMN gene, which targets the motor neuron cells in the body. And there have been several testimonials, several videos even, if you're interested, that you can find on this drug, Zolgensma, where children are able to not only survive beyond 2 years old, but they also are able to do a lot of activities independently, like eating on their own. They play on they are able to play and jump and run and do all the things that a 2-year-old is expected to. So we are hoping that this drug lasts for a long time, and we hope to see improvement in these kids. They are also being monitored currently, and this was approved in 2019. And it's actually termed as a miracle drug because it's saving little kids. All right, so no gene therapy talk is complete without talking about cell therapy. So I'm just going to touch up a little bit on the CAR T cell therapy, where CAR stands for chimeric antigen receptor. So basically here patients so blood is collected from the patients, and T cells are isolated, and these are reprogrammed either using a lentivirus or a retrovirus that carries the CAR, the chimeric antigen receptor. And CAR is made for a specific cancer's antigen, so it's made specifically according to the patient's needs. So it is very personalized. And once these T cells are treated, they now carry the CAR, which you see in red in the schematic here. And these cells are multiplied and scaled up in the lab, and then they are injected back into the patient. And then these T cells carrying CAR go and attack the tumor cells, and that's how they reduce or prevent the cancer. So this technology has been approved for several different cancers, and lentiviral vector or retroviral vector is used for this therapy. And this is four of these have already been approved, 2 in 2017, 1 in 2020, and 1 in 2021. So it has shown amazing response in the patients. So in short, the future of gene therapy is really bright, and there are several known rare diseases, like 7,000 plus, and 80% of them are genetic. And so if we can diagnose these early and provide genetic counseling, then that definitely helps with the treatment. And I just want to end saying with an exciting note of how many clinical trials are ongoing in gene therapy. There are over 1,000, and there are over 2,000 clinical trials ongoing in cell therapy. So we hope to help all the patients out there with gene therapy. I just want to say a big thank you to my group shown here in the picture. I've learned a lot from this group, and it's been amazing working with them. I couldn't be here personally at the conference, but if you have any questions, please feel free to email me. And this is just a picture of the lab that I work in Cambridge. And thanks a lot for your time and attention. Thank you.
So, ma'am, can you please move on with the third session?
I think, yeah, that was a very nice overview of gene therapy. We'll move on to the third talk in the session, and that is on prostate cancer genomics, and this is by Dr. Bushra, Jolly Hill Chair Professor, Senior Fellow, DBT Wellcome Trust India Alliance, Principal Investigator, Molecular Oncology Laboratory, Department of Biological Sciences and Bioengineering, the Mehta Family Center for Engineering and Medicine, Indian Institute of Technology, Kanpur. I think it's quite a lot, and the stage is all yours. Yes.
First of all, thanks to organizer for having me here, and especially Dr. Neeraj for the invitation, for giving me this opportunity. So I have a disclaimer, though my slide says prostate cancer genomics, but I'm not going to talk about that. And this is some recent study from the lab that I'm going to focus on. So coming to the prostate cancer, like this walnut-shaped gland is located at the base of urinary bladder, and you can see there are like 4 different zones in this gland, and each zone decide whether it will get cancer or not. And if you see mostly peripheral zone is having more incidences of tumor, prostate cancer tumor, followed by transition zone. And pathologically, if you look at the gland, there are several such foci, and this is one foci which has multiple cells. You can see these luminal cells which is lining the lumina, and then you also have basal cells, few neuroendocrine cells, and with neoplastic trigger in these luminal cells, they start proliferating, giving rise to adenocarcinoma. Since androgen signaling play a very critical role in gland development as well as in the prostate cancer, mainly all these patients are put on androgen deprivation therapy, but after some few years, these patients, they develop resistance, and they are known as castration-resistant prostate cancer patient. And eventually, because of the failed treatment, some of these patients, they even develop neuroendocrine prostate cancer, either as a resistance mechanism to ADT, or sometimes it's like de novo, which is also overt neuroendocrine prostate cancer. So there are multiple molecular reasons why these phases occur. Like for example, genetic rearrangement, and one such genetic rearrangement is between TMPRSS2 and ER, and there are several mutations and so on and so forth. So I'm not going to go in detail on this alteration, but what we thought, coming from the cancer field and then also looking at the normal development, many genes that play critical role during the development, they also have a role to play in oncogenesis. So if you look at this diagram, this is a developing epithelia, and in this particular case, you see there's not many genes which are involved in differentiation, but once the epithelium is developed, you see increase in the expression of differentiation specific gene. And similar to this region here, also in prostate cancer or any cancer, you see expression of some of the homeobox genes which are earlier here. So there's a huge list like Hox gene family, NKX and DLX1. So I'm going to focus on DLX1 family. And the gene that I'm going to talk about is DLX1, which is like shown here in this embryonic stage, and they play a critical role in cranial development as well as in the interneuron correction of the GABAergic neuron. Now, like this gene that I'm going to talk about, DLX1, has been used as a ligand diagnostic marker in the urine sediments or urine exosomes, and this test has been developed by Select MDX. So if you see in this panel, this is one of such genes. So what we thought, like if this gene is being detected by this company, Select DX, what is the role of this gene? Is it expressed in the localized and metastatic tissue or not? How it's regulated in cancer disease, in prostate cancer, and what is the functional significance of this gene? So what we did, we examined like a localized disease, like localized prostate cancer, around 144 patient specimen we got. And what we did, we just did RNA in situ hybridization, and we found like 60% of these prostate cancer patients, they have overexpression of DLX1. And when we looked into ERG genetic alteration that I told you, it's like recurrent in 50% of the prostate cancer patient, we found majority of the patient who have this TMPRSS2 ERG genetic regimen and also show overexpression of ERG oncogene are positive for either high or medium DLX1 expression, whereas in ERG negative cases there was DLX1 but not as high as in ERG positive. So since I told you like androgen signaling play a crucial role in prostate cancer, we also examined the expression of androgen receptor in these cases, and we found equal distribution. So we found all the cases where we see TMPRSS2 ERG fusion, they were positive for AR signaling, whereas like some of the cases they show 50% AR and 50% no ERG expression, whereas all the cases where ERG was there were positive for expression. Now, correlating with the disease progression from clinical pathological point of view, what we did, we characterized this patient on the basis of like high, medium, low DLX1 expression and the increase in Gleason score, which is a pathological grading, many of you know. So we see like in advanced Gleason score, the expression of DLX1 was high or medium, and similarly when we go at the advanced disease, like here you can see like the high and the medium DLX1 expression was more at the advanced pathological disease condition. Second, to understand like what is the status of DLX1 in metastatic castration-resistant prostate cancer, what we did, we got these multiple metastatic sites, and when we stained this for DLX1 expression, we got like quite a surprising number. About 64% of these total metastatic site they show like DLX1 expression, and of these, highest expression of DLX1 was found in bone, followed by liver and other tissues. So in the lab, like to understand the role of DLX1, what exactly is it doing, we know like it's highly expressed, and now exactly what it's doing, what we did, we took this benign cell line, overexpressed DLX1 in these cells, and examined this cell line for oncogenic properties, and you can see here with overexpression of DLX1 in the benign line, we see increase in cell proliferation as well as foci and invasive potential. Likewise, when we took a cell line which is positive for DLX1 and performed CRISPR-Cas based knockout, and what we got here, like we see significant decrease in the oncogenic potential of this cell line, which is highly cancerous or highly oncogenic. So to understand what are the biological pathways which are involved in DLX1 mediated prostate cancer, what we did, we performed the gene expression profiling of these cell lines that we created with the CRISPR-Cas9 knockout. And when we performed this David analysis, we found within the knockdown cells, there was a significant decrease in these all oncogenic pathways such as proliferation and migration, whereas we see increase in the apoptosis and other pathways such as differentiation and the mitotic cell regulation. Likewise, just to validate this, when we looked for the genes which are involved in epithelial to mesenchymal transition, we found that at both transcript level as well as protein level, we see significant decrease in the expression of the gene which are involved in mesenchymal phenotypes such as vimentin, and increase in the expression of E-cadherin, which is more of a epithelial mark. Likewise, we also see increase in ABG2, which is ATP binding cassette receptor, also CD44, which play critical role in stemness. You see there's a significant decrease when we knock down DLX1 in this line, 2 to RA1 and VCAP. And likewise, in RWP, benign line, when we overexpress DLX1, we see marginal increase in these two cell surface receptors, which confirm its role in EMT as well as stemness. Now coming to the mouse model, what we did, the cell line we generated with the CRISPR-Cas knockout, we implanted in immunodeficient mice and let them grow, and we can see significant reduction in the tumor burden in the group where we implanted DLX1 knockout cells. Likewise, when we, at the end of the study, we collected bone marrow and lungs to see how many these cells metastasize to these organs, and this was characterized by performing allo-specific PCR, which will detect only allo sequences in the human cancer cells, and we see significant decrease in the metastasis in the mice where we implanted DLX1 knockout cells. Since I told you, like in CRPC human patient sample, we found high DLX1 in the bone, so we performed this experiment, which is experimental bone metastasis, and here we implanted DLX1 wild type cells, that is 22RE1, and the knockout clones in the intermedullary tibia, and let them grow for a month, followed by this micro CT horizontal and the ventricle imaging. And you can see here that the mice where we implanted the cells, the wild type cells where we had high DLX1 show more bone loss and low bone tumor volume as well as bone surface area and the trabecular unit, compared to the cells where we have a low DLX1 or knockout of DLX1. So again, it confirms the DLX1 has some role to play in bone metastasis and disease progression. Now, since I told you TMPRSS2 play crucial role in in 50% of the prostate cancer patient, and we also found that all the TMPRSS2 positive patient, they have DLX1. So we hypothesized, could it be that TMPRSS2 is kind of driving the expression of DLX1 in prostate cancer patient, and maybe AR is also cooperating with this. So to examine this, what we did, we scanned the DLX1 promoter region. We found a few ERK binding site, which was confirmed by performing chip PCR. So you can see here in VCAP cells on both the ERK binding site, that is P1 and P2, we found in this chip PCR data that there was an increased ERK recruitment, and PLAW was used as a positive control, which was also confirmed by increasing the H3K9 acetylation mark, which is an activation mark, and also enrichment of RNA pol. So again, it confirms that ERK get recruited on the DLX1 promoter and drives the expression of DLX1. Now, the question was like how it's working in the cases where there's no TMPRSS ERK fusion, because I told you in 50% of the patient which have higher DLX1 have no TMPRSS2 ERK genetic arrangement. So that data I'm not showing in interest of time, but what we have shown in the absence of ERK, AR and ARV7, which is variant 7, along with FOXA1, drives the expression of DLX1, which was confirmed by a sequential chip PCR. So you can see here, CULT L2 was used as a control and DLX1 here. So what we did, we pulled down the immune complex by using ERK antibody. We see enrichment, and then followed by AR pull down. So we see the co-localization of both ERK and AR on this particular ERK binding site. So this data was also validated when we analyzed the publicly available Chia PET data for VCAP cells for RNA pol. And what we did, we integrated this Chia PET data along with the chip PCR data for AR and FOXA1, and we could see there's a significant peaks in these regions, and also there's a looping. So it suggests that the ERK binds on the DLX1 promoter, and it interacts like in an enhancer promoter interaction, which leads to DLX1 overexpression in ERK positive patients. So since bromodomain and extraterminal protein are known to be play critical role in the gene regulation, especially they are known to bind on the lysine of the histone, and that's how they drive the recruitment of some of the transcription factor as well as RNA pol, and drive the expression of some of the genes such as ERK and AR. So you can so we hypothesize, like if bromodomain protein, they're driving the expression of ERK and AR, could we use the bromodomain inhibitor to perturb this angle? And then that's what we did. We used the VCAP cells, which is translocation TMPRSS2 are positive and also express a higher expression of DLX1. So when we treated these cells with JQ, it's a BET domain inhibitor, and we see significant reduction in the ERK as well as DLX1. Likewise, in functional assays, treatment of these cells with ENZA, that is anti-androgen, and enzalutamide, JQ, or a combination resulted in a significant decrease in the cell proliferation as well as migration of these cells. So same thing we recapitulated in the mice model where we implanted 2,2-ROV1 cells in immunodeficient mice and randomized these mice into 4 groups and treated them with either enzalutamide or a BET inhibitor or a combination of JQ and enzalutamide. And what we found that there was a significant reduction in the tumor burden in both in the mice treated with JQ as well as a combination. So there was no additive effect with ENZA and JQ, and at the end of the study these mice were euthanized and the bone marrow and lung was collected to analyze how many cells have been metastasized. So you can appreciate here, again we see significant decrease in the micrometastasis in bone marrow as well as lung in the mice who were treated with either with JQ or a combination of JQ and ENZA both. So coming to the model that we have proposed, so we kind of showed the how DLX1 is regulated in TMPRSS2 fusion positive cases as well as in fusion negative cases. So you can see here in a fusion positive cases it's more of a ERK dependent, and what happened ERK get recruited on the promoter region of the DLX1 on the ERK binding domain, and it kind of loops out with the AR and the FOXA1 which get recruited on the FOX exon in this gene and drives the DLX1 expression. Whereas in ERK-independent cases, the AR and FOXA1, they drive, this complex drives the expression of DLX1, and what we have shown that this DLX1 being a homeobox transcription factor also bind to these 2 genes, that is LDH1A and HNF1A, which both of these genes, they play a critical role in stemness in the disease progression. So this data I have not shown in favor of time, but these are the 2 DLX1 target gene, and combine this together, it play a crucial role in promoting the tumorigenesis of prostate cancer. These genes play a crucial role in cell cycle, in stemness as well as metastasis and decrease in the apoptosis. So we kind of shown that ERK binds to the DLX1 promoter and interacts with AR and FOXA1 via short-range promoter enhancer interaction, which is leading to increase in DLX1. And in these cases, inhibitors such as JQ or a BET inhibitor could be used to disrupt this ERK and AR transcription circuitry, which will result in the decrease in the ERK expression and AR expression, and subsequently resulting in decrease in DLX1 expression and DLX1 mediated oncogenesis. So finally the acknowledgement slide. So this is the my clinical collaborators within India as well as abroad. So thanks to them, without their support this would not possible to finish this work, and all these talented students from my group. This is the old picture, this is a recent group, and thanks to the funding agency for supporting this work. Thank you.
Thank you so much, Dr. Bushra. That was very nice, fine work. I think you were also coordinating with Dr. Nalla there, am I right?
Yes, yes. Yeah, Nalla is my long-time collaborator.
I'm calling from here, Chairperson.
Oh, okay, sorry.
Yeah. So thank you so much, sir. Would you like to just close the session?
Yeah. Basically, I would like to ask you one question. That no doubt there is a lot of things are going around in prostate genomics. And are they at the experimental stage, or they have come clinical trials, or they have come into the market for treatment also?
In India, no. In India, definitely no. But in US and European countries, yes. So JQ inhibitor, not JQ, but other advanced BET inhibitor has been in clinical trial. And even in one study, they've even shown some resistance to a BET inhibitor also. So, yeah.
So basically, what you want to say, DLX1 inhibitor, the
so
DLX1 gene is the...
I want to say there's a diagnostic test which is available in the field. We are not doing in the India. I don't know Tata they have this on panel or not. But people in select MDX, they are detecting DLX1. So if we categorize patients who are positive for DLX1, who are positive for ERK, so these subset of patients may benefit with BET inhibitors. Because anti-androgen may work, but after some time they'll develop resistance and become CRPC. So maybe as a combinatorial therapy will work.
It is irrespective of the grade of prostate cancer.
Yes, it's irrespective. So the first data I showed 60% in localized disease, and then CRPC have shown more patients who have more DLX1, they have more bone metastasis.
But basically, DLX1 is one of the components,
right,
probably, and there are much more.
So what I have shown that all the ERK positive patient, which are for 50%, in India we have shown, my group has shown 50% of the patient from northern part of India, they're positive for TMPRSS2 ERK. So they're all positive for DLX1. But DLX1 could not only be with the ERK positive, it could be ERK independent also, it should be like, so total 60% of the cases they have DLX1 overexpression. And since it's a transcription factor, it may have many more other roles that I have shown that also regulate the expression of two other genes which play critical role in stemness and cell migration. So that could be one of the reasons.
So if the audience want to ask any question. No, then we would like to thank all the three speakers. It was a nice session, and all I would like to comment the last one. Basically, she is the right. She has said rightly that this is some therapy which is available abroad. And it needs to come to India because no doubt there are few patients who will not respond to castration alone or androgen therapies alone, resistance. Yeah. So in that cases, DLX1 and the ERK transcription plays a major role, almost 50%, and 50% of the patients can still be treated, all we can say.
Right.
Okay, thank you very much.
Thanks. Yeah.
I would like to thank the chairpersons and the eminent speakers for this interesting session. So now we will be proceeding for the 6th session, that is the technology part 1. Thank you, sir. Thank you, ma'am. So for this session, I would like to invite Dr. Gopal Parikh, consultant histopathologist, Green Cross Pathology Laboratory and Molecular Laboratory, Ahmedabad, and Dr. Umang Patel, Director, Green Cross Laboratory Genetics, Ahmedabad, to chair the session. I would like to make few announcements. This complete session is going live, entire conference is going live, and more than 500 viewers are there who are watching this online. In online mode, there are questions which are flashed after each session, but I would like, because these sessions are not on their time, so you can log in, whoever is answering those questions, whoever wants to attempt the answer to those questions, they will be flashed for 20 seconds, and at the end of the entire conference, they will be given prizes. The top scorer will be given prizes. So now, sir, with your all consent, we can start the session.
So, very good afternoon, everybody. I would like to invite Dr. Feroz Ahmed. He is section head in molecular pathology, Department of Molecular Genomics at SRL. I know he handles a wonderful team there, looking after the NGS portfolio there. Welcome, Dr. Feroz. His topic will be understanding the clinical utility of NGS in identifying chromosomal anomalies associated with first trimester pregnancy loss. What does the data say?
Hello. Good afternoon, everyone. First of all, I would like to thank the organizing committee for giving me this opportunity to share some of the interesting findings on relatively rare kind of things on an NGS platform. So, if you see, miscarriages and pregnancy loss are not uncommon. It's quite common, and it usually results in spontaneous loss of pregnancy, mostly before 20th week of pregnancy. And if you see frequency-wise, it occurs almost 10 to 20% of all clinically confirmed pregnancies. Now, if you look what are the different etiology and the frequency which are associated with this pregnancy loss, so around 10 to 15% of those pregnancy loss is because of certain anatomical defects which are seen in an individual. Roughly 1 to 5% of the patient may have certain infection which results in loss of pregnancy. Around 15 to 20% more than that is associated with hormonal or autoimmune disease wherein there is a recurrent pregnancy loss. And 1 to 5% of the cases will have certain epigenetics and other unknown factors which are not yet elucidated. But what is important to note over here is that almost 50 to 60% of these pregnancy loss in the first trimester happens because of certain embryonic chromosomal abnormalities. And this frequency is relatively which is quite high. So if you see, the most common chromosomal anomalies which are seen in the fetus in early pregnancies are numerical abnormalities. Almost 90% of them are numerical. It could be either trisomies, or it could be monosomies or polyploidies. And structural abnormalities are relatively less, maybe around 5 to 10%. And most of the structural abnormalities are deletions, loss or gain or translocations. Now, if you see what are the different available methods which are being used to detect these abnormalities, of course, conventional karyotype remains the gold standard, and it is there since years. It has its own advantage and disadvantage. The biggest advantage is that it screens the entire genome. The resolution is 5 to 10 MB. The beauty is that it can also detect balanced rearrangement translocations. Though mosaicism is relatively less, if you count 20 or 30 or 40 cells, you may get 1 or 2 mosaic cells also. So it is not completely no, but I will say compared to other methodology, it is less. Failure rate, of course, because it needs certain cultures, so failure rate is relatively high if you compare to other. And throughput wise, it is low because it is manual. And cost wise, definitely it is less as compared to any other advanced methodology. Next comes the FISH. FISH, again, it is a targeted based because you need to know which target you are looking at. The resolution is beautiful, 50 to 1 MB size. Balanced rearrangement can be detected. Mosaicism, yes. Failure is relatively less as compared to cytogenetics and low throughput, but again, if you compare with the conventional karyotyping, the cost is relatively high. This is one most important thing that a lot of people now are being used by chromosomal microarray. Now, the beauty of chromosomal microarray is that it screens the entire genome with a very, very high resolution. So resolution-wise, I think it is the best in the industry. But again, it cannot detect any balanced rearrangement translocations, but mosaicism can be detected. It has, of course, low failure rate, but it needs a high throughput instrument as well as little bit of more expertise as compared to FISH and cytogenetics. Now, what is changing is that apart from these technologies, what we see is that can NGS, which is being now used in wide range of clinical applications, whether it is cancer, whether it is reproductive genomics, clinical genomics, can NGS also play a role in identifying these chromosomal anomalies? The answer is yes. And the basic idea is to extract the DNA from the fetal product of conception. Then you prepare a whole genome, low pass whole genome amplification, and then you prepare a library, and then you do sequencing, and the analysis is done. So the advantage is that, of course, the coverage is good because it screens the entire genome. Resolution wise, it is around 2 to 5 MB with modified workflow. Of course, reliability is very high because you get a mosaicism called very, very high. Success rate is very high, very low failure rate. And again, one major advantage is that the multiplexing of up to 16 to 96 samples can be done. So cost wise, it's become very economical. And of course, except for the library preparation, rest everything is fully automated. You don't need to have a manual intervention. So it is less prone to undergo any kind of any error. So once you do the sequencing, the analysis depends upon the platform which you are using. Right now we are using on Iron Torrent platform. So it's a cloud-based platform wherein you can upload the run files on the system and then select the workflow which will screen the aneuploidies and then launch the analysis. There is a provision to make your own copy number variant CNV analysis also if in case required. Otherwise, the workflow is already set and it's an automatic workflow. There is no need to do anything extra. Now this is how the data looks like. This is a apparently normal euploid. You can see very nicely the entire, these are the chromosome 1, 2, 3, 4 till X and Y. So anything which is less than 1.5 is loss, and if it is anything which is gain more than 2.5 is gain. So this is how a normal euploid genome will look like. And to compensate that, we wanted to do a small pilot study wherein we started with 112 samples of product of conception. The first thing which we did was to eliminate any possible maternal cell contamination, because maternal cell contamination will definitely give you an NS result. So out of this 112 cases, almost roughly 11% of the cases showed maternal cell contamination, hence they were not processed. Remaining 100 cases were processed, and we found that out of this 100 cases, roughly 46% of the cases showed no chromosomal abnormality, whereas 54% of the cases showed some kind of abnormality. If you see, further segregate this, out of this 54, 47 cases were numerical abnormalities, almost 88%, and 7 cases showed structural abnormality. Most of the structural abnormalities were CNVs, which were in the range of 10 to 80 MB and were found to be pathogenic in nature. As far as numerical abnormalities are concerned, you can see almost 94% of the cases were aneuploidies. Aneuploidies are autosomal chromosomes or sex chromosomes, and 3 cases, interestingly, they also showed mosaics. And in terms of aneuploidy, 36 were trisomies and 8 were monosomies. So trisomic would be trisomy 20, 21 or T12, T1, any autosomal or sex chromosome aneuploidy, and monosomies were mostly of the X chromosomes. And in case of mosaics, we saw 1 autosomal mosaicism and 2 sex chromosome abnormality. So this is how this is a case wherein you can see the ploidy level of this is 3, and this corresponds to chromosome 21. So this is how we detect, and we give us this case was a trisomy 21. Similarly, if you see this one, this is clear cut or monosomy X, wherein you can see this X corresponds to the ploidy number, and the ploidy number is 1. So there is a clear cut deletion or loss of chromosome X over here, and that's why we call it as a monosomy X. This is what you can see in case of trisomy 22. You can see there is a clear cut ploidy gain, and the ploidy level is chromosome 3, so it is considered as trisomy 22. Trisomy 8, trisomy 2, trisomy 15. So these are some of the single aneuploidy examples which we have detected. Similarly, we also saw some complex aneuploidies. Like in this case, you can see, in this case, there was a ploidy level of chromosome 16, 17 and 19. All of them had only 1 ploidy. So it indicates there is a clear-cut loss of chromosome 16, chromosome 17 and chromosome 19. So like such cases, there were around, in some of the cases, there were more than 5 different abnormalities. So those are really considered as a complex aneuploidies which are being seen. Interestingly, you can, as I said, this NGS can also detect mosaicism. In this case, you can see there is a ploidy level of 1.7%, which corresponds to chromosome 19. So roughly, you can see there is a mosaicism of 30% of chromosome 19. Similarly, in this case, there is a sex chromosome abnormality along with because this ploidy level is 1.7, so this is mosaicism. But it was also associated with a complete ploidy of chromosome number 21. So this case had trisomy 21 along with mosaicism of sex chromosome X. Similarly, in this case, you can say there is a sex chromosome mosaicism, and the mosaicism level was found to be 40%. So with this, it is very clear that it becomes very easy and very fast track you can do, and there is not much of any skill is required once you set your assay and once you run the instrument on the platform. So to conclude, what we learned from this exercise is that NGS can also be used in other platforms, not only restricted to oncology or reproductive genetics. And this is one such example wherein it gives you all the flexibility, whether you want to scale the samples or whether you want to do a high resolution. Cost-wise, it is equally better, and of course, high success rate and low failure rate as compared to other existing technologies. With this, I would like to conclude, and thank you for your attention.
Any questions?
Yeah. Hi, Dr. Feroz. Thank you and a good job done. Just wanted to know one thing, NGS is a platform and there are various ways to do. So what did you do to do that whole genome sequencing?
Yeah.
What have you done? Low pass?
So it's a low pass whole genome amplification.
Okay. And at what depth it was?
This is roughly less than 1x also, because you're detecting aneuploidies. You're not detecting small micro like at a chromosomal level or nucleotide level. So you don't need to have so much of depth, because you're looking for bigger. Like most of them are CNVs and like entire chromosomes. That's why you don't need to have a deeper depth, like unlike in cancer wherein you have 500x or 1000x, or for germline you have 30x. So it is less than 1x only.
Yeah, okay, thank you. Great job, Dr. Feroz. Thank you.
Nice presentation. Next speaker is Dr. Pankaj Khanna, Manager Application Support, Gene2Me, Hyderabad. And the topic is role of role of NGS-based oncospecific gene panel to aid diagnosis, prognosis, and treatment.
Yes. Very good afternoon. Very good afternoon to all of you as well. So I am doing research and I am part of Genes2Me, and the idea here is how we can help the oncology and the other fields of genetics where people are doing NGS and getting the major bottleneck of the data points. So we are getting huge data. How do we handle, how do we translate it? That's what our aim is. The aim of our company is to make the genetics for the billions, and with that logics, we want to start with what the idea is. The main idea which we started is, okay, the first idea of doing the molecular diagnosis is get the things right, identify it, and so also characterize it at molecular levels. Everybody knows that's the aim of any test, but what is the differentiation factor is the breadth of the present tests which are available ranges from right from the morphological criteria, the classical way. You do it in the microscope, you do it in the fish, or you do it with the help of FACS. You want to see the morphological differences, and then came the other logics that you want to go for a single gene analysis. I have seen some of the slides where people were looking into expressions and then looking into the single gene levels and thinking monosomy was critically playing the role. And then lastly, people have gone to the NGS level and then to also to the level of single genomics, single cell genomics, where even we are targeting at a single cell level what changes are happening. But is it important to understand is that the screening processes, whatever we were seeing at, they were the gold standards. This is where we start, this is where we look into the data and we start guessing what next test should be taken up. In those lines, if you see, what are the major drawbacks? What we see onto the only looking into the morphologies are that we are seeing we cannot do the continuous test. As Dr. Ritu Gupta was showing in the data that there was a clonal subselection of the population. As the Dr. Suryavanshi was talking about, there is a change in the mutation over the period of time. How do we continuously test it only by morphological test or only by the single gene test? There is a shift of mutations which are happening either because of the age of the cancer what is happening or because of the resistance subpopulation of the cancer which are being developed up. So what are the next steps one can take? They can take the molecular approaches right from the levels, what we are thinking at the whole genome level. But one of the data which is being shown very recently that people have seen, if you just compare two same categorized cancers, there are more than 1 million SNPs variations between them. So as somebody has already said, one of the speakers, that there are something like variations of unknown regions that we don't understand what they do. And if you do the NGS levels, if you do at a higher clinical levels, you get a lot of data which we don't understand now at this stage. And moreover, we do require higher computing powers to understand what is happening with those and how do we understand maybe in the future, but at present what we thought is to take an intelligent design where we focus on different pathways, defined pathways, phenotypes, and so also a different complex disorder nature of the similar types that people have reported associations or important in the drug therapies to select for those, and we start thinking of how to create the panels. The idea came, okay, the idea is people have done a lot of research over the decades in these fields of oncology and other fields of genetics. How do we compile them and make it more sensible so that it is clinically easy to interpret and actionable for the patient? Because if you think of a WGS, wait for 1 month, the patient will no more be there to get the right treatment at that stage. So what next? So we started, okay, so we have two different methodologies to do enrichment. The first method is looking into the hybridization based, and the other one is looking into the amplification based. So amplification based technology, if you look into it, we have some of the advantages and some of the disadvantages of the both. If you look into the amplification based technology, there are a few chances that there will be a loss of alleles because of the allele dropouts, that the primers are not binding, there are changes in the primer binding sites, and you're not getting the amplification there. And you also have got the major limitation of how many targets you can do, because there the amplification multiplexing is the major limit, that how many genes and targets you can keep adding that to your assay, whereas hybridization based assay doesn't give you that limitation, that you can select the regions continuous, discontinuous in a different parts in a very effective manner. More so, if you look into the different regions of the larger genes where you need to cover larger exons, classically in the Michel's dystrophy, where we are seeing DMD gene, which is one of the largest gene known, it is hard to cover different regions. In the similar fashion, is there any genes which we want to cover at intervals of splice variants as well, so that we want to understand the different levels of spliceosome which is happening, so that we want to see that, okay, we took a route of taking the hybridization based approach so that it gives us a better leverage of selecting which pathways we want to go ahead with. And this is what Dr. Lalji Singh, my mentor, used to seek at that point of time in the early days when we started the research way back in 2002. What we want for a personal medicine. We want first all the chromosomes. This is Sirkos plots taken from the Cell paper, where they have shown at the edges all the chromosomes all the way from 1 to Y, followed by the kind of insertion and deletions. Then you are looking into the RNA up and down regulated, you are looking into the protein, and then also you are looking into the SNVs levels of all the things. So this gives you in a nutshell, if someone goes for a personalized medicine or in fact an intelligent panel design, you can go all the way from the chromosome right up to the protein and SNV levels. That gives you a complete picture what is happening within the body, not only at genomics, at expression levels of RNA, as well as protein, and of course what is happening with the interactome. With those approaches, we thought, okay, we will decide the panels, and the take-home idea of this was to see, number one, hybridization capture method, and we want to have the maximum efficiency of the capturing method, including AT and GC rich region. That was our main goal, that what we have seen in the most of the sequencing data, that we are seeing biases, the regions where it is high in AT or GC, we get less coverage, and the normal regions get very high coverage. The idea was to do the rebalancing in such way that we get the even distribution of across the sites, even at AT and GC rich regions. If you look into the panel content, if you look into some of the most important things which we have said, that number 3 is we want to be compatible with almost all the instruments that are available in the market, be it Illumina, be it MGI, be it Thermo Fisher. So we have created a kit which comes in a 2 ways, which is one way of hybridization is the major drawback, if you ask me what could be that, is the time what it takes for the hybridization. Then what we did is, we did 2 ways. Some people like, I start that work today, put for the hybridization in the overnight, and next day I continue my job. But few people want a faster results, so we have made another one called enhanced hybridization, where we will be able to do within 4 hours the hybridization and the process can be completed. And the idea is not only hybridization, we start with the sample and we do all the way library preparation, create a enrichment, and then the adapter ligated product will be ready for loading onto the machine. That was the idea of creating all these parameters to create it. And what are the major advantages? What we have now is ability to pick GC rich regions, ability to pick repetitive sequences, and without masking the homologous region. Normally people, what they do is they mask the homologous region to avoid, but really we want to pick it from the homologous regions, because most of the cancers we saw, they are having the important genes in the homologous regions which are repetitive by nature. And we have also performed some of the low amounts with the ctDNA, that we want to work with the lowest possible DNA, because in the ctDNA samples we get very, very low sample. How much low we can go, that is 20 nanograms, and normally for that 50 nanograms. And we also have got comprehensive panel for the disease as well as for looking into the disease specific panels as well. So if you look into the coverage, the data, what we have shown here, that we have got the GC regions which are covering normally into the areas which are normal. When it goes to the AT rich region, the area becomes others with lesser, and ours is looking into the same in the normal phase, that what we get in the same percentage manner. And looking into the depth of the coverage, looking into the AT-GC regions, we try to cover as much as possible where most of the other people lose onto the data. This is an example which I would like to show onto the pan-cancer panel, that what we have got is 524 genes which are covering the whole CDS, and what we have included is we have included 2 viruses, EBV and HPV virus, which are also important in knowing to the cancer, and we have also covered rearrangements, TMB, MSI into this particular part. The idea was to cover these viruses so that you can also understand the etiology in the different ones. Very quickly to see that we have covered very focused CT genes which are important into the lung, colorectal and breast cancer pattern as per the NCCN. And on the other lines, if you see that we were able to get to the 0.5% VAF, and we are claiming mostly 1% VAF for looking into those data sets. So looking into the other ones, we do have hereditary cancer, which we have worked out to differentiate between the somatic cancer, and we have also got some of the panels which are focused to the screening of the oncochecks, right from as low as 50 number of genes in a single panel to the cancer check of 100 panel, where we are focusing only the core set. So some people say we want very focused genes of the interest and we want to work on those. So for those applications we have developed that. Any other very important thing which we have developed is Metformin. This is the panel of genes which are important for drug responses to be seen. And not only that, we have covered only oncology, but most of the other diseases and the known targets of them, where we will be able to see even the infectious disorder, how we are going to respond to work with. The idea is we are also supporting these with the software, where you can just put the data and you can get the clinical output information which is important to you. And lastly, I would like to conclude with the fact that when you are looking into the clinical exome, there is a very lot of other products also available. We have differentiated that. We have included as many genes as possible we could, 7,513 genes, which are one of the maximum in the clinical exome presently. And we have also included the mitochondrial genome to cover it. So in these lines, we have covered not only the part that mostly people are doing, but we also want to see is there any ataxias, any energy differences which is happening at the level of electron transport chains, which is now crucially important. We have try to cover it with those. With that, I just want to touch with few quick slides that we are able to do CRPV panels for looking into not only the viruses which is coming, and with the other viruses as well to put for. This is another one which I want to put, that this is one of the most highest killer in India, with the number of people which are dying because of cardiovascular. We have created that as well. So with that, I would like to conclude, and I would like to take any questions if you have in those lines.
Yeah, any questions? Thank you.
Thank you very much.
So next I would like to invite Mr. Mohammad Zaid, Director Applications Shiva Scientific from Delhi, and the topic is NGS in a transplant diagnostic lab.
Dr. Zahid, just a minute, I have one announcement to be made. So I would like all the panel faculties who are involved in the panel discussion, which will be within 15-20 minutes after this session, to be present here so that we can, without a break, we can start the session. So Dr. Zahid, yes, you can go ahead.
Okay.
Should be there.
It's not here. Anyways, I'll manage. So good afternoon, everyone. My name is Mohammad Zahid. I'm Director Sales and Applications at Shiva Scientific. First of all, I'd like to thank the organizer and the chairperson for giving me this opportunity for presenting this presentation. So the topic is NGS in a transplant diagnostic lab. We have heard that so many stories about NGS. It's being used in oncology, reproductive genetics. So why not in transplant diagnostics? Thank you. So I'll give a short introduction about our company, Shiva Scientific. We've been founded in 1984 by Dr. Dinesh Upadhyay. He was the front runner and pioneer offering the transplant diagnostic products. As of now, we also deal in 3 more verticals, which are oncology, hereditary, and reproductive genetics. And these are some of our partners. So let's get to basics. NGS, next generation sequencing. It's also known as massively parallel sequencing. There are various platforms to choose from, but if we have to divide them into 2 broad categories, one would be short read sequencing, which is offered by Illumina, Ion, and MGI, and on the other hand, we have a long read sequencing, which is offered by PacBio and ONT. Yeah. Now, what happens in a transplant diagnostics lab? I think Dr. Varsha has already covered most of the parts. I will just divide into 2 broad categories. One is a pre-transplant workup, other is a post-transplant workup. So in a pre-transplant, you would be screening the patient and donor, trying to find the right match. It's more of histocompatibility testing. In the post-transplant, we'll be monitoring the patient if there is a relapse or there is a allograft rejection. So these are the 2 main areas. So what all can an NGS system offer for pre as well as for a post? So let's look at the NGS workflow to understand what can be done in a transplant diagnostic lab. So there are major 5 steps for any NGS workflow, starting with the target generation, library preparation, clonal amplification, sequencing, and data analysis. So target generation is targeting genes or loci of interest, and library preparation is actually adapting those genes so that it can be further clonally amplified, sequenced, and then eventually the data analysis. So clonal amplification and sequencing is platform dependent, whether you have a short read sequencer or a long read. Even in short read, you have Illumina, ION, or MGI. So let's look at the target generation. So in target generation, the most important target would be the HLA, which is the MHC. Here we can use a high resolution HLA typing. So we have different sorts of strategy for amplification, whether it's singleplex, multiplex, 6 gene, 11 gene, depending on the solid organ or onto the bone marrow transplantation, you can target HLA. Second target could be MYC genes. So what is MYC? These are MHC class 1 chain related genes A or B. They are also involved in NKG2 deactivation. You can do a high resolution MYC and MYC-B typing between a patient and donor so as to find whether they are a match or not. Even if they are a mismatch, in MYC-A genes there is a dimorphism at codon 129. It is highly recommended to have a methionine match at codon 129. And also, once you have the typing of the MYC-A or MYC-B, it can be used for anti-MYC antibodies. Third target we can use is KIR genes, which are killer immunoglobulin receptor, which are the receptors for activating or inhibitory receptor for NK cells. Their ligands are HLA class 1, which are already typed. And if you type the KIR genes on an allelic resolution, you can find whether they're activatory or inhibitory, because within the activatory genotype also there are inhibitory alleles. So once you do the allelic resolution, typing, you can actually define what will be the GVL effect, and then you can choose a best donor. So let's say if you have 3 donors which are full match or half match, out of these 3 donors, which ones to choose from, it can be captured from here through the KIR genes. Fourth target could be a indel. Indel is insertion and deletions. It's just like an STR or SNPs across the human genome. They're very polymorphic. We can target them in an insertion or deletion. So if you target them, there can be 2 kinds of amplicon, a short and a long. And we can actually differentiate a patient and donor. So this would be used mostly in chimerism or in engraftment monitoring, post-transplant, in hematopoietic stem cell transplantation. So what is the advantage of doing an indel? Because chimerism monitoring is usually covered with the STRs. The advantage is the sensitivity. So if you look here, this is an indel and this is a SNP, and STRs are not covered on the NGS. So this is a 5% sensitivity, and these are all noise. So if you're actually covering on the SNPs, they might get intermingled with the noise, where the indel can still be differentiated, and it can be easily traced up to 0.05%. So that's the advantage of having an indel-based monitoring on our NGS system. And the fifth target could be cell-free DNA. So why not? We can have a non-invasive test to look at the cellular mediated rejection of the allograft. So we can use the same indel markers, can also be used on SNPs, and we just have to target the cell-free DNA from the patient plasma. So what happens here? There can be an antibody-mediated rejection, which cannot be captured here, but there is a cellular mediated rejection which can be seen here. So this is the creatinine level across the different time points, and this is the donor-derived cell-free DNA. So this is cell-free DNA is actually more specific, coming from the allograft, which was from donor. Let's say this has also been verified by the biopsy at the day 14. You can start with the immunosuppressive treatment, and then you can see the donor-derived cell-free DNA has become stagnant. So this will be actually, again, monitoring. It will give you the early relapse onsets. Okay, this is going to happen. Let's start with the immunosuppression. You don't have to do a biopsy. So this is a very latest technology in transplant diagnosis, which can only be offered as of now on NGS. So these are the targets. So targets are done. It could be HLA, MYC-A, MYC-B, KIR, indels for chimerism, or for cell-free DNA-based donor-derived monitoring. Second step is library preparation. So library preparation majorly involves 4 steps depending on the platform. So I'm majorly targeting the Illumina, ION, and MGI platform. Fragmentation, adapter ligation, indexing, and cleanup. So what happens when we do the amplification of these genes? These are long-range amplifications starting from 3 kb to 20 kb. These are not short amplicons. And it needs to be fragmented because eventually you have to sequence them in short patches. So first is the NGS Go library kit, which will do the fragmentation of these amplicons, and it also do the end repair so that you have these A overhangs. Second step is adapter ligation. Adapters are platform specific, whether it's for Illumina, ION, or MGI. and you get these adapters. Now all of these samples need to be barcoded so that eventually they can be pulled along in the same run, and then they are provided with the proper or unique indices. Final step is a SPRI cleaner, which is a solid phase reverse immobilization cleaner with the magnetic beads, and eventually you have a clean DNA fragments which can be further sequenced. Before sequencing, you have a different kinds of library like we discussed, like HLA, MICA, MICB, CARE, chimerism, or donor-derived cell-free DNA. All of them can be pooled. So HLA, MICA, CARE would be more of pre-transplant, chimerism, and donor-derived cell-free DNA would be targeting the post-transplant, and they all can be pooled now. So this is about library preparation. We offer Library X and Index Kit, which can cover that. Clonal amplification and sequencing is platform dependent, so you don't have to be worried which system you're using. It will be handled by the instrument itself. And finally, you have the data. So once the data comes in, we offer our pipeline, which is an engine that will do the automated, just a play button analysis for all of the system. So it's hardly take 2 to 3 minutes. It can be installed on a local system or on a server or on a cloud. And eventually, when the data is done, you can review them. So this is the total workflow. Now imagine if I say this workflow usually takes like 3 to 4 days, then probably it may not be used for a transplant diagnostics. But the gimmick is I can perform it in less than 24 hours. It's not even a gimmick, it's actually possible. So we have collaborators like Unipath, MedGenome, Strand, various labs who are doing this. So if you start at 10 in the morning, by 5 you can load onto the sequencer, and next morning you can report. So in less than 24 hours, NGS is possible for transplant diagnostics. So now, what all is possible now? HLA typing, MICA, MICB typing, K typing in the pre-transplant scenario, and for post-transplant, monitoring for the chimerism, which will be for stem cell transplants, and donor-derived cell-free DNA monitoring for solid organ transplants. This is the summary. Less than 24 hours of workflow is possible, which would be need of the hour for the transplant diagnostics. NGS is important in transplant diagnostics. It is going to stay here, and probably next coming years you will see more and more tests which can be adopted to actually enhance the transplant outcomes. So these are our partners, Genetics and Devisor, from which we offer these transplant diagnostic solutions. We also offer some other NGS solutions which are targeted towards the cancer like BRCA, HBOC, hereditary like thalassemia, CFTR, familial hypercholesterolemia, some IVF-related panels, and if you're in need of any bioinformatics for any targeted panel, whether it's whole exome, whole genome, we offer Varson Clinical and Premium as well. Whatever it is, we support the full workflow, whether you just take the transplant or oncology. Thanks. Any questions?
Thank you. All.
Thank you.
Our next speaker is Dr. Anuj Gupta, Business Manager, India, New England Biolab. Topic is NGS library preparation from low input and precious sample from NEB.
Thank you, sir. Good afternoon, everyone. First of all, I must thank Dr. Neeraj sir and all organizers for providing this opportunity to speak here. All the wonderful presentations on the technologies, and in continuation to that, I wish to highlight here what New England Biolabs NEB is helping for preparation of libraries for NGS from samples which are a little difficult to process. There are those which are having very low input amount and precious samples which you cannot utilize again and again for preparation of libraries. So, founded 38 years back in 1974 by a Harvard professor, Donald Cohn. NEB is a company which is basically famous for enzymes, and yes, that is true. NEB has been developing beautiful enzymes and polymerases and all these things, and over the years it is driving innovations with introduction of FDA approved diagnostics and enzymes, and including providing, offering solutions and enzymes and polymerases to first NGS system, Solexa, in 2006, and later on NEB thought that when it is very having a mastery and expertise in enzyme production, polymerases, wanted to produce our own kits. So in 2009, we have introduced our NEB NAS portfolio with library preparations. And later on, we also came up for COVID-19 and other molecular diagnostics and vaccine manufacturing, providing some RNA, mRNA solutions, semi-synthesis solutions. And NEB has expanded its portfolio, diversifying its portfolio from the core DNA enzyme and cloning to amplification, RNA genome engineering, protein expression and glycomics, and later on for NGS reagent and kits. So if you see the typical NGS workflow, it involves nucleic acid extraction, library preparation, either targeted or whole genome sequencing in different ways, which involve adapter ligation, site selection, amplification, quality control, followed by sequencing and analysis. And if we see, library preparation is the most crucial step, as sequencing chemistries and technologies are fixed in a manner that whatever it is going into the sequencing, it will sequence. So the most important part is the library preparation. So when we thought of making different library preparation kits, the things come that it is yet another library preparation kit or something else. So we focused on some of the major challenges which people face in library preparation on daily basis, like very low input or minimal DNA sample, working on degraded sample, FFPE samples, or highly degraded, highly damaged ancient DNA, environment DNA samples, and also how to minimize library preparation steps in the workflow, how to get rid of adapter and primer artifacts, how to get rid of PCR wise, how to optimize our library preparation so that the GC bias, high and low GC bias can be taken care of. So NEB has come up with different library preparation kits, and with the inclusion of different modules, with fragmentation, without fragmentation, and many other things. But most important is the input amount which a successful library can pick up is very, very low. So a guaranteed input amount, it can work with 100 picogram to 1 microgram. So the dynamic range is very high. So we don't need different kind of separate kits for low input and high input library preparation for samples which are having different DNA amount. So if you see the sequencing, when we have more DNA, good quality, high quality DNA with good amount of input DNA, everything works well. The sequencing matrix works well. The things are messed up when we go up to the lower and lower DNA input amount. So if you compare the 100 nanogram starting material versus 1 nanogram starting material, you can see the things are going worse in terms of duplication rate, in terms of on-target rate. But here NEB has optimized the components and the enzymes and the workflow so that when you go up to 1 nanogram and even picogram, things are working as good as they are working with the high quality of high amount and quality of the starting material. So we have also reduced the steps of library preparation workflow and cleanup reactions so as the time duration of making a library should be minimized, the workflow should be minimized, and a few of the things like fragmentation and repair are included in a single tube. So end repair, detailing and enzymatic fragmentation, everything is happening in a single tube. So it reduces the workflow time, it reduces the hands-on time, it reduces the loss of DNA, and by this you are able to reduce the input amount requirement of the DNA. NEB also uses a specialized kind of adapter which is hairpin loop structure. So it binds to the DNA fragment to prepare a library, which helps in higher efficiency of ligation first of all, and then it also reduces the formation of adapter dimers and primers in the final DNA library. So what happens if you have more adapters? It will be getting sequence in each some of your flow cell positions, or we need to do another round of cleanup which is required for adapter removal, which is not in the case with the NEB library preparation. And this hairpin loop structure is having a uracil base which is cleaved by the user enzyme to prepare a final library. So it is very easy. And as it is more efficient, when we go from high nanogram to picogram of DNA amount, with the reducing DNA amount, the PCR cycles required are very low as compared to other library preparations. So this is important because if you are doing low PCR cycles to prepare a library, the percent duplicate rate is automatically reduced. So this is implicated in some of the good publications where people have detected, have taken good CNV calls with ultra low input DNA. So it is known that for CNV applications, high amount of DNA is mostly preferred, because if you're going to low input, you cannot have a very good and highly confident CNV calls. But with few nanogram and picogram level of DNA, CNV call can be properly actually managed. NEB kits have also been used in a lot of studies, including NIPT studies, and in this publication, the genetic profiling of Vietnamese population for a large-scale genomic analysis was done for, they have done nearly 2,600 samples for NIPT testing and identified nearly 8 million of SNP with only very low pass sequencing of 0.17x sequencing. Utilization of very low input DNA is also seen. If you are having a cfDNA or something, even it is producing very small amount of DNA, still it is having DNA in the range of nanogram. What if DNA is in the picogram and even less? So in this example, in this publication, basically, a Romanov family, which is a royal family, some of the people of this family were buried actually by some war or something, and they want to identify, they want to dig out the dead people and take on the sample to identify which dead body is belong to the royal family, and they got nothing but some of the rootless hair shafts found in the relics. So that is basically having a very low amount of DNA or nearly nil DNA. So, and they tried to prepare a library with NEBNext Ultra 2 DNA kits, and they were able to make successful library, able to make successful NGS run, and able to align with the known live persons and identify the family members actually which are really there. So this example shows that it is not 100 picogram or 50 picogram of library, but if you have very low amount of sample, virtually 1 or 2 copies or something like that, still it is possible to prepare DNA libraries for NGS. Similarly, if you have highly degraded DNA, in this example, this is one of the example of FBI's oldest case where they have found a 4,000-year-old mummy, and they want to analyze and identify the gender and other specifications for the sample, and this was most damage is highly degraded 4,000-year-old, and they prepared the library and could successfully identify the gender and everything. It's done. Okay. So one more thing is FFP samples. So in FFP sample, we usually see a lot of degradation, like deamination of bases, different nicks and gaps, bases are oxidized, ends are blocked, and along with DNA fragmentation and crosslinks. So NEB had developed a repair kit with which you can repair your FFP sample prior to make a library. So we are not joining the fragments because it is already fragmented, but we can repair all other things, which can significantly increase the yield of the library, and you can start with minimal amount of FFP sample as compared to unrepaired DNA. So this is one of the most important thing if you are working on FFP DNA, to repair before preparing the library. Similarly, for metagenomic sample, there is a challenge of isolating bacterial or viral DNA from the host DNA. So NEB has developed a novel method by which they utilize methylation binding domain of a protein, and this protein is selectively binding to methylated DNA. And this kit utilizes the methylation pattern differences between the prokaryotic and eukaryotic DNA. So eukaryotic DNA is highly methylated, while prokaryotic DNA is not. And with the help of protein A, which in turn binding to MBD, we are able to remove out most of the human or host DNA, and only the metagenic or bacterial DNAs remain. So it is highly successful in removing out the host DNA and enriching the microbial DNA, and the removed out host DNA can also be used in library preparation for the host DNA. Similarly, one more challenge is found when we are doing methylation sequencing. For methylation, we have to do bisulfite treatment, and it is known that bisulfite treatment damages the DNA, and there is a huge loss of DNA also occurring. So it is replaced by an enzymatic methylation, where we are doing the same thing. We are changing the unmethylated cytosine into uracil, but not with the bisulfite method, but the enzymatic method. So first the methylated cytosine are changed, oxidized by TET2, and with the ApoVec enzyme, they are again changed. So we are getting the same profile which we are getting with the bisulfite treatment, but it is enzymatic method. So there is no loss, no damage of the DNA. So if you see the unique SNPs and unique variants which you are finding is much more higher as compared to bisulfite treatment, and as you go with the higher depth of sequencing, the difference is much more visible. Similarly, for RNA library preparation, there are multiple RNA kits, and Ultra 2 RNA kits is basically amplifying a very low input amount of RNA as similar to DNA, and we have cells, we have kits which can prepare library even from small, even from single cell to 1 microgram input amount. So we have a huge range of kits which can work from the single cell to microgram input. So normally Ultra 2 RNA kits which are working with the 10 nanogram or 5 nanogram of starting material, based on what type of depletion pattern you are following, depletion kits you are following, we can prepare library as a directional library and non-directional libraries. And if you see and compare NEB kits with other kits, we have a similar pattern, similar profile. If you are working with the 10 nanogram, 100 nanogram or 1 picogram of starting material, there is no difference. That means it is working well with even with a very low amount of starting material. And 3' to 5' coverage is also very excellent, because there is no bias between where it is starting. Okay, so this single cell kit is able to work with a single cell in your tube, and there is no DNA and RNA isolation. It isolates RNA and prepare library from them. So it is producing. It is able to produce successful library from a single cell as well. So NEB, this is my last slide. NEB is providing you some DNA and RNA kits, which the same kit you can utilize in multiple applications, whether it's standard or low input sequencing. It is for whole genome amplification, whole genome sequencing, ChIP-seq, ancient FPDNA sequencing, and many more. So that's all I want to present. We have our contact details, and we have our booth number 9 in the exhibition hall. So if you have any more questions, we welcome you.
Nice to hear such archive material of 4,000 years, 400 years papers are already published doing the sequencing work. So you for the RNA-seq workflow, the input RNA is fine. You told it's a very low input RNA, you can prepare a very good library. What about the quality of RNA? So did your kit have any hybridization data like RIN index in the archive material or the FFP samples, and your ultimately the sequencing quality as well?
Yes, so basically RNA integrity number is very crucial for determining the quality of RNA. That is very true. So it basically depends upon the quality of RNA, or it depends basically if you are having some RNA which is of degraded quality, then it is important whether you can proceed with that for a library preparation, or you have to discard the sample. That is more important. We cannot say that if it is having a RNA rin number of 10 or rin number of.
So like you describe the quantity of input RNA, but is there anything like it should be the RNA quality should be more than like 7, then only to take, or what?
I'm answering the same question basically. There is no cutoff is made that you can proceed with this rin value, above rin value or the lower rin value, because it is not a diagnostic platform where you have to determine something, right? You can make library as most degraded sample. Maybe it is a rin value of 1, 2 or 3 as well, because in FFP RNA you can see rin value is never more than 3 or 3.5 or something like that, 6 or 7. But FFP sample is pretty good to prepare library. I have shown you example of highly degraded. Maybe your bioanalyzer cannot determine the rin value, even even rin value 1 or something, but still library can be prepared. So it's not point of a validation, but it is a matter of expectation. You have the sample. If you have the sample of rin value 1 or 2, or you are trying to make a library, you can expect that my library will not be as good as if I am making from rin value 7 or 8. That is an expectation. But the most important is you can prepare a library from with highly degraded DNA samples, and you can successfully utilize that in sequencing. Because sometimes if you have a choice and you can determine the rin value, if the rin value is very good, then you can go ahead. If the rin value is not good, then you can ask your user, or if you yourself are a user, that I will isolate again the RNA and do it. But sometimes you don't have a choice. Whatever DNA is available. So this is not the point of the validation. It is the point of, can you prepare a successful library which is sequenceable? You can get the data without much hindrance with highly degraded DNA or RNA. So rin value is important, but more rin value you are having, better will be the library. But there is no cutoff or validation for that.
Any more questions? So, thank you all. I would like to conclude this session and hand it over to Spring.
Thank you very much for all the speakers, and thank you very much these chairpersons. Now, ideally this should be a tea break, but we have arranged the tea and coffee at the back side of this hall itself, so it is there. But session will go on. So now there is a panel discussion which will be going on. So I hope all the panelists are here. So for this panel discussion basically on the pre-analytical errors, their impacts, and how to minimize in the era of precision medicine. So this panel discussion will be moderated by Dr. Mausmi Suryavanshi. She is head of Cancer for Molecular Diagnostics and Cell Biology at Rajiv Gandhi Cancer Institute and Research Center. Then I would like to call upon the stage all the panelists. So their names are Dr. Soumya Ranjan Malik, Associate Professor, Department of Pathology, AIIMS, New Delhi. Then Dr. Deepak Kumar Mishra, I think he is not available for this discussion. So we will move on to next panelist. So Dr. Rekha Pai, who is pathologist at Department of Pathology, CMC Vellore. Dr. Sudha Murthy, director at Datar Cancer Genetics, Nasik. And Dr. Bhavesh Parekh, who is senior medical oncologist at Selby Hospital, Ahmedabad. So may I ask all the panelists and moderator ma'am to come on the stage.
Because most of them, or you can announce that they should bring their tea.
Okay, so at the downstairs. Oh, no issues, no issues. So,
because it will benefit if somebody wants to listen.
Sure, I will do that. Yes. So, I will make that announcement and then we'll come back. So we will take 10 minutes of break here. So we are taking 10 minutes of break here right now for the settlement of the audience. I'm going to make announcement at the downstairs also, who all are going want to join this panel discussion, so they can come and sit over here, and then we will start. So we are right now it is 5:05. We will be starting this session by 5:15.
ये आयो सेम आयो हमारे ये माइक्रोवेव सेम ये दोनों है ना पर्सन फोन पर है दोनों का Bobo. बोल क्लिनिकल बोलो हाँ चालू આવી જશે 24 આપણે તો બધા દિવસ ચાલુ આવી હા થઈ જશે Uncle, follow. ઓકે હા જોઈ લઈશ આજે કોન્ફરન્સમાં જોઉં મોલિક્યુલર હજી હમણાં જઈશ એટલે જોઈ લઉં ઓકે
Hello everyone, good evening. So, so it's 5:15 and now we are starting the panel discussion. So I would request all the delegates to settle down. You can have your tea or coffee mugs with you. So we are starting the panel discussion on the topic of pre-analytical errors, their impacts and how to minimize in the era of precision medicine. This panel discussion will be moderated by Dr. Mausmi Suryavanshi. She is head at Center of Molecular Diagnostics and Cell Biology at Rajiv Gandhi Medical Sciences, New Delhi. Rajiv Gandhi Cancer Institute and Research Center. Panelists for the sessions are Dr. Soumya Ranjan Malik, Dr. Deepak Kumar Mishra, Dr. Rekha Pai, Dr. Sudha Murthy, and Dr. Bhavesh Parekh. So I would like to invite all the panelists to come on the stage and start the panel discussion.
I have changed my picture. So can you display mine? Okay, good evening everybody. So we will be discussing the pre-analytical errors in molecular diagnostics in the era of precision medicine. So my first slide is of a fairy tale. So this is the story of a dwarf, Rumpelstiltskin, and I'm sure all of you know him. Some of you may not be remembering. So I'll just tell you the story and you will remember. So this is a dwarf who is able to make gold from straw. And this makes an ordinary girl a queen, and then finally the queen is also able to save her son, and I think you will remember now. So these are fairy tales and we have grown up listening to them, but we live in real life and the real life is very different. No technology can spin straw into gold. You must begin with gold. So pre-analytical analytics are the gold you start with. So with that background, I will present 3 case scenarios. So these are real case scenarios and these are real things which happen very often in the lab. In fact, I selected the topmost 3. So what we go through and how we process and can we do something about it. Okay. So this is a first case scenario. This is a patient who is a case of non-small cell lung carcinoma. NGS was performed on tissue biopsy and the results were available. So I would like my panelists to please view the findings, and my first question is to the clinician, Dr. Bhavesh. This is a case of non-small cell lung cancer and you are finding lots and lots of mutations. There is NRAS, there is IDH, EGFR, not the usual one, BRAF, FGFR2, KRAS, HER2 and MAP2K2. So there are a lot of mutations. The coverage, if you see, is like in 2000. So my god, that's very good coverage. And even the allele ratio, like even the first 2 is like around 36% or 39%. So these are good variants. But for a case of non-small cell lung cancer, how comfortable are you reading this report when the lab gives you such a report? So are you okay with this? So what do you feel?
Totally confused. So looking at least...
Can we give him a mic?
At least looking that, at least end of this panel discussion, I got a gold coin from the organization. So really means even you had started a really good thing, but for a clinician to look this report, because we are just ordering a test, but when the report came, and then we have to face the patients and the relative to answer. And especially in today's era where Googleswar Mahadev is also there in Shravanvar, where we have to answer so many Googleswar questions also. So I think this type of report or this type of things, the importance of each and every finding, whether it is clinically relevant or not, and whether we had a clinical available drugs to offer the patient is very important.
Yes. So this is what I want the lab people to understand. Look, we are behind the scene. They are the guys who are facing the clinicians. So you have to understand their problem. So if he treats the patient based on such a report, which to him doesn't make sense, so it is a blast off every way for him, and then he will obviously blast you off. So you have to understand why they are angry, why they are not happy. So that is the reason. So this looks such a loaded report, but he's not happy. So then I want to ask you another question. So when you get a NGS report, so do you look at the quality parameters? Does the report give it? So do you look at those quality parameters? So
Ideally, madam, every clinician should also understand the quality report or ask for the quality report as well. It is a 15-page report of one NGS, and if we are not going to read one report, at least take 30 to 40 minutes. And in a busy clinical schedule, if we are not able to read properly and ask so many questions, even it's really havoc for us. And I think that's why I feel that to attend molecular pathology conference by clinician is really important.
Yes, this is another point, and this is for my scientist and my pathology students, colleagues, everybody. Look, I work with a lot of medical oncologists. They are reading really well. So if you don't keep pace with what they are reading, they will beat you at what you do. Because they are really read well, they have so many people to teach them also, and they do it for their own profession. And unfortunately, this is the truth about us, that we are not reading and we are not keeping in pace. So, but such reports do get out. And another thing he just mentioned is those 15-page reports. So those are 15-page reports. Yes, they are 15-page reports. So what is important, what he should not miss? So there are those things, and each report actually should carry quality parameters or a quality satisfactory, quality of DNA, quality of RNA, maybe uniformity, and what is the limit of detection. So that is a separate topic altogether. So now what I am trying to do is, so this is the problem that has occurred, this is the difficulty he is facing. So I will like that we all go backward. So what has happened? So what can we do about it? So Dr. Sudha, this is for you. So do you mention quality parameters in your NGS report?
Yes, thank you, Dr. Maushmi. Yes, we do mention. I think that is a basic requirement as per the guidelines as well. So the quality parameters in terms of depth of sequencing, coverage, and what is whether it is Q, the analytical sensitivity, specificity, and of course for each parameter, like if we are looking at a comprehensive panel for SNVs, perfusions, CNAs, each one we determine the during validation itself, and all these need to be mentioned. And of course the most important is the variant allele frequency. So these are the things, basic things. Probably I have not missed out anything.
Anybody who wants to add to something, Dr. Rekha?
I think it's also, if it's a somatic panel, you also need to declare upfront at what percentage you're cutting off all the variants. If it's 5% and over, and that becomes
limit of detection or analytical sensitivity, that's very important. In fact, anything, Dr. Malik, you'd like to add to?
One thing with uniformity, like apart from coverage and depth, you must see how uniformity is uniformity. That's also required, where you can get the errors. If it is a uniformity around more than 95%, so unlikely you are getting a false curve.
Yes, in fact that is very important, very good point. The quality checks are very important. Quality check at the DNA level, RNA level, and that should be mentioned in your report. So if you mention uniformity, that is very nice. Above 90% is good, but if you're not mentioning, at least you mentioned that quality checks have passed. At least write that, and DNA separate and RNA separate, because most of the samples actually RNA quality check is that which usually fails and DNA passes. So you can at least give the DNA report and not comment about the RNA, but make it very clear to the clinician, because tomorrow he's going to repeat the biopsy, then he should do the RNA which was missed last time, and it should be in bold, it should not be in those very small letters that nobody is going to read. So that is another point which I like to highlight. So I am not going into the bioinformatic details because that will be beyond this, but just to show that for this particular case the uniformity, as he mentioned, was low. Okay, but still, if I will still look at the sample, okay, it was 76%, it was low, and I'm not going into more details, but if you just look at the 500x coverage, so that was also, I think, 85%. So that is also not bad, but low. But just let's go back to the case. So if you just look at the mutations, there are a lot of C2T and G2A mutations. Most of them are C2T and G2A mutations. So that should raise a brow that something has gone wrong. And of course, this was clinically unexplainable. The first thing was, wow, what is it? There's some mix up. So many mutations. You usually don't see so many mutations in a targeted panel. So that is another thing, until unless it is a MSI deficient patient. It is that case where you might find a lot of mutation and the TMB is also usually high. So if the TMB is high, so you will be finding mutations. So that is another clue. So these are clinical things which should make you go back to the bioinformatics, or if you are yourself doing, so you look at your quality parameters. So these are very important. So, okay. So, the next question is, how would you investigate it? So you need to go backward. So where has things gone wrong and can it be corrected? So it is very difficult to get the sample again. So what do you do in such cases, Dr. Sudha? So okay, anybody can take it. So if you find something odd, okay, uniformity is not right here, and the mutations also, I am obviously not comfortable. So how do you go back? So what do you do? Simply reject it straight away? There may be something which is like, okay, true call, you can validate it by an orthogonal method and maybe report it.
That's
so,
exactly what I was thinking. To start with, there are two things you could possibly do. One is, apart from going backwards and looking at your quality parametrics, I think you could first open it, let's say, in an IGV, a viewer, and look at the sort of base calls that have arrived and how good the base calls were.
Yes.
Whether there's a strand bias, whether, you can first rule out all those and get some sort of an idea whether, this could be a true variant or not. Second is, even if you go down to looking at any of the somatic databases, you won't find many of these reported perhaps. That's yet another clue to you that you're possibly dealing with a set of them which...
You have that IDH 132, which
so
is like a...
R132H is being a very, very common one. So
yes.
These are sort of indicators to you that, yeah, you better look at it. And then the third point is, I don't know I thought you told me all the allele frequencies are high. So possibly that you could have another orthogonal system of testing. Yet again, there are problems. You're not going to have primers for every single one of them sitting with you. All those can cause delays. So these are some of the issues you have to consider.
Yes, and especially if you have something for which a drug is available. So you take that seriously. That particular mutation has to be taken seriously. So if you can offer something. So usually what we do is we go back, we look at how good was the library. So you can use the quant of the library if the quant is passed. Usually the samples which go for sequencing, they have more or less passed, or if there is space, something is borderline, they may like, okay, chalo isko bhi chala dete hain. So that attitude is there. But yes, you should go and look at the quant of the library and the tape station. So where your peaks should be, like in this case. So for this particular panel, usually the peaks are around 230 something. It was not very good, but not very bad also. Because at times these samples work. And then going back to the DNA quantity and quality. And of course, for the quantity, you can use Cuvette, and for the quality, you can use Nanopore, there are other things, even your PCR you can use.
And one little
yes,
addition
yes,
here,
please.
I think it's good to know where your sample originated from, and if it is from FFP, and also whether it's been a block which has been lying for 5 or 6 years.
Yes.
That can be a big clue to you
Yes,
that
yes,
you
yes.
have
We
a
are
problem.
coming. You are all bang on. We are coming to that, but of course it was a FFP sample. Just look at it. This was this particular patient sample. So what you are seeing here, and this is a normal genomic FFP sample. So just compare it is more fragmented. Most of the samples are these fragments are in this range, whereas normally it is in this range. So here you know that okay, this was a poor quality DNA. Okay, so from here the work starts. One is reject it, forget it. That you can do if you are in a private lab. Dr. Sudha, you may be lucky. But if you are in a hospital and it is your own sample, so what will you do? Yes, Dr. Malik. So if it is your own sample, so what are you going to do?
If you are doing a fragment analysis, you never get up to 600 base pair. More than 6 pair, you never expect. You will never, whatever may be your formalin, good fixative you are using, adequate fixative. It is never, you will expect like this 2500 highest.
But this is our sample, no. And look, the DIN is also not, 10 is the best one, it is 5. Okay, and that is 2, DIN number. So can you see that? That's 2 and that is 5. So this is our own sample, I'm telling you. So okay, 4 is also good, but 2.56 also at times work. And Dr. Anuj is there, he can tell better. So that also works at times.
Madam, for a clinician also at the end of 7 days or 10 days when the lab gives a call, sir कुछ नहीं हो सकता, sir एक दूसरा sample लेना पड़ेगा. Then again it's a very big problem to us also. So I think that type of giving an answer is also sometimes difficult, not that easy. Either is a private lab or in your own setup.
Yes. Actually, this is a very genuine problem and we face it so many days because at times it is after 3 weeks we say it doesn't work. And you know, at times you also know that this patient might not even get something, and we have a policy of refunding such patients. And they say, "Mujhe chahiye," as if you were going to give something out of magic or something, because liquid biopsy is maybe good for lung cancer, but other cancers you don't get anything. So, but the patient, yes, please.
So basically this is right that sometimes as you are saying, we don't have option for getting another sample of the DNA. Sometimes we degrade it. So in this library preparation protocol, there are different recommendations which are made for degraded DNA or high quality DNA. And based on that degradation also there are recommendations that if it is having a DIN value of 2 or less than 2 or more than 2 or between 5, you have to take that much amount of DNA, you have to do that kind of extra sequencing to get something good out of that.
I don't know, in this particular case, if I do extra sequencing, these 40% will only amplify. So, because these are pseudo mutations which are already there. Okay, so extra sequencing will not, this is my opinion, so it will not help.
I'm sorry, I may add in, I think in a real life setting, there are 2 things which can't be done. We can't keep repeating samples. One, because we don't have the money for it. Okay, the patient is not going to pay again for it. As it is, we have not been able to deliver their first report. And like she says, we have a system of refunding. So we've already lost money for what we've done. Okay, that's the first thing. And then again, the sample itself. See, many times when we're dealing with paraffin embedded material, it's not like we have lots of DNA to go back to. Correct? The sample itself is very, very limiting. So that again becomes another issue. So it's very complex. There's no one answer to it.
Yes. Is there anything which you can do in such cases? So, Dr. Malik, would you like to take anything you can do if there is?
Like you like to say this DNA.
This is what you have.
You can do an enrichment process so that at least purifying the DNA. Already you are doing purification by magnetic way to enrich and to extract out wash out the lower fragment DNAs. Other things like this CG mutation, CG if you are seeing in IGV repeatedly CG lens, so then you can treat your during the library preparation to prevent this deamination.
The treatment I actually differ is not, yes please.
Can we, am I audible? So can we do like deamination, because deamination can be a problem in such cases because of the storage of the FFP samples. So if it is the problem, then there is one treatment which has to be performed when you are preparing libraries with the FFP sample. Certain panels are available which are like costlier. They by default they add this step at the beginning. So I think the lab should use this step with all the panels.
When will you do deamination? So yes, I agree. If you don't have anything, you should try. So what step?
Ma'am, ideally, just before starting, the first step of the amplification of the DNA. At that point,
you have to start initially.
Yes,
only
initially. At that point of time, we have to do deamination, and it is not much costlier. Anybody can afford it, and in any panel, it should be. It can be added.
It is there.
So that should be followed.
Yes.
So you are advising all cases of FFP to do deamination?
Yes.
Yeah, actually, look, I'll give you an example of, actually that's my next question also. I am also partly answering the question. Panels like tumor mutation burden. So there, they are doing deamination, but if you have such mutations and somebody is, this patient will have a tumor mutation burden of maybe 15, 20, somebody might start immunotherapy based on your report, and that does happen. So if you don't treat this, so yes, it should be done, and especially if you find something and you don't have anything else. And otherwise, if you just let these reports go, so these can create a lot of problems, especially if it is a TMB panel of loss of heterozygosity panel. So that can be there. I think we covered all the questions. So this is a very good article on deamination. So how they do it. So you can read. This is for the viewers. And okay. So now the problem doesn't end here. Another thing which you can do is, like if their variant allele frequency is very low, so you can put a filter. So usually when it happens, it is less than 10%. Okay. So this was a rare case. Usually it doesn't happen like this. If it's less than 10%, you can put a filter. You know this is, and you can take help of bioinformatician, so they can give you confidence. Okay. And you can put a filter and you can remove all of them. So that if it is mildly going this, then going back to the histopathology lab department to see what can be done and what has gone wrong. Actually, in this case, this patient, this sample had come on Saturday evening. So the formalin, it was a formalin problem, and Monday was a holiday. So okay, so you can imagine what has happened. But this does happen in labs.
Just one thing, like in our setup, like at AIIMS, we receive approximately 30% referral things which diagnose outside, they come to AIIMS. And from the 30%, I can say more than 50% we send is like under fixative, partial autolytic changes. Even if, forget about this NGS, even if you can do IHC also. So those parts we are highlighting that most important for the private parts which are small labs which are doing, like you highlighted only Saturday to Monday, like over fixative. So many cases are under fixative, and I talked to few labs, they told their turnaround time. To decrease the turnaround time, they don't do the fixatives. And second is the 10% neutral of a formalin.
Yes.
That's more important. So people take formalin, make it 10%, so they never understand that it become a formic acid and it will degrade everything.
Yes, these are actually very genuine problems of our country, of our system. But what we can do is educate, I think. So that's what we are trying to do through this. We are showing the problem, we are showing what the clinician faces, and that's what we can all do. At least we are doing our bit.
But can be taken care by the clinicians, most of the part. Sir is here.
Yes, like but you have to educate the clinicians. If they are telling you their problem, you have to tell them your problem.
We are ready to learn.
Yes, they are. I am telling you, they are learning faster than most of us. So you should tell them that this is what I want. This is how it has to happen. If it doesn't happen, don't ask me. Come back.
But it is better, like I will suggest ki better use the commercial fixative which is already available, which is 10% neutral buffer formalin, which is a stable pH for long time. Rather than just cost cutting, you take the 40% and make it better.
It depends on the situation. Like if it is a place where nobody will be able to control these things, so it is better. Spend extra, get the quality. You cannot compromise on quality. So it is situation to situation, and yes, it is a very good idea. You should take a commercial this thing. So what are the parameters? So Dr. Sudha, can you answer this? What is ischemia time? So that is also important in processing. These things will affect the molecular testing. So what is ischemia time? Yes.
I don't need a mic probably. So the cold ischemia time is more important. Not that warm ischemia time is not important. It's a continuous exercise that we need to also educate our surgeons, because it's not just the lab fixing the tissue. The surgeon needs to keep, or the theatres. I have been to theatres wherein for frozens, we see that the surgeons are so engrossed, and in some hospitals the practices leave this tissue there, and then you continue with your surgery, come back to that. So there is no assign. Thanks to NABH, now at least we have those parameters which are getting ticked off.
Right.
So I think that is one thing which needs to be addressed. So the fixative needs to be put immediately, especially for small biopsies. If we can put it directly in the fixative, that would be the ideal situation. Now, in situations where Dr. Malik has identified in referral labs where you do not have an extensive support, I think it is prudent on our part to send a sample of fixative, which is 10% neutral buffer formalin, whosoever is our client.
Yes.
And the third thing, of course, cold ischemia time, there are standard guidelines. For small biopsies, 6 to 48 hours, and for larger biopsies, it's about 12 to 72 hours, if I remember right. And also, it's not just about putting the tissue, we need to section them and ensure that penetration happens uniformly. Oftentimes, even grossing is an issue when we cut thick sections. The prosector cuts thick tissue sections. We blame the histotechnician, but actually it's a problem on our side. So it is not getting fixed. That is one issue. For the smaller biopsies, it's actually the whole process needs to be driven by the pathologist. Looking at the tissue, we need to decide on, at that point in time, you need to take a decision what needs to be done with the tissue. And for that, we need to sensitize our own colleagues. So like for example, you have very tiny tissue, it's not always possible to have 2 blocks that like it is recommended. We have heard some lectures as well. So we have to ensure that we make our histotechnologist a part of our team, sensitize him that it cannot be cut or trimmed as much or refaced, so that we do not lose out tissue. Also, there are situations when we have to be judicious and be generous-hearted, limit our IHCs and leave the tissue for molecular. So that's an ongoing thing which needs to be.
At the heart of everything is a pathologist.
Yes, absolutely.
So, pathologists, you don't just have to look into the microscope.
This is the first time I've heard a pathologist say, don't do too many IHCs.
like you have to coordinate with the surgeon, with the medical oncologist, with the lab technician, with the molecular person, everybody. So your job is not just looking into the microscope, you have to talk. So actually I had 3 cases, and this could go on. So I will just show you this. These are very good papers. So if you want to know the details of pre-analytics, especially the histopathology part, so these are pre-analytics and precision pathology. This is the guidelines from CAP, and this is another good review articles, so you can read them. And Dr. Sudha has very nicely summarized most of the major issues. And I will skip the next 2 cases because I think we are short of time. He's already ringed the bell, but I will just tell you the second case is a hemolyzed sample of liquid biopsy. So you're taking, I'll just leave you with that. Kya hota? Huh? So when you are taking 18 ml of blood and then the sample is hemolyzed, okay, and you tell the patient or you tell the clinician, patient is in ICU, whatever, and dobara lena hai. I want it again. Okay, so what happens? So this is and the third case was this is a very common thing for, especially for lung carcinoma patients, insufficient tissue. So the tissue is not sufficient. Either the IHC depleted, as she was very generous of saying कि IHC, stop IHC. Most of the pathologists don't do it. Okay, so either it is that.
केस में कई बार आपको PET scan के साथ correlate करना चाहिए, और with the correlation of the PET scan where you get a more tissue. So here the role of interventional radiologist or who are doing the biopsy.
Yes.
The most of the times, even in other breast cancer or head and neck cancer, where also superadded bacterial infection or necrotic tissue is there. So to get a tissue in a proper from the core, from the center is also very important.
Yes.
Sometimes we are doing it from the periphery, and there is a more necrotic material rather than a proper malignant material.
So one more person has come into picture, that is the radiologist. So it is never ending. So I will leave it. So I think you can read, or if you want my slides. And ROSE, rapid on-site evaluation, is another very important thing. So if you are taking samples for NGS or something, so you should evaluate right there and then and see if the sample is adequate, and use the cytology samples for molecular analysis. So that's a very rich resource, and even the CAP ISLC has advocated that cytology samples should be used, even the stained cytology slides or FNAV blocks, or and of course tissue also. So okay, I am skipping all this and I'm coming to the last slide, and the conclusion is, I think there are no doubts about it, that's a shared ownership. It's a shared ownership, but at the center is the pathologist who's going to coordinate everything. So that's, with that, I would like to thank you, and thank you for the patience.
I would like to thank all the panelists for the beautiful, wonderful discussion over here. And now we will move on to our next, last session, I would say, that is on clinical perspective. Okay, sorry, this is the session 7 and this is in the technology part 2. I would like to invite the chairpersons for the session, Dr. Vivek Gupta, Associate Professor, Department of Pathology, in charge Molecular Diagnostics and Research Laboratory, Government Institute of Medical Sciences, and Dr. Apurva Shah, Histopathologist and Cytopathologist at Unipath Laboratory. So I think Dr. Apurva Shah is not here. So, ma'am, would you like to chair this session? No issue. Ma'am, please. So, can we start the session? So, I think you have the details over here for all the speakers.
Yeah, I would like to invite for the first as a speaker, Dr. Mike. He is from Thermo Fisher Scientific. He is working as Senior Manager, Medical Affairs, Asia, Pacific and Japan, and he is currently located in Singapore. So, Dr. Mike, over to you.
Thank you very much. I'd first like to thank the organizers for giving me the opportunity to come here and talk a little bit about what Thermo Fisher has to offer in the whole space. So, as our chairman said, I manage medical affairs in the Asia-Pacific region for Thermo Fisher, and that includes everything from India to New Zealand, stretching to Japan, all of Southeast Asia, Taiwan, and South Korea. And my role in this vast organization is actually not to sit here and sell you platforms and technologies. It's actually to generate real-world evidence that those platforms and technologies and the targeted therapies to which they point actually have some benefit to the patients, and in the long run actually benefit health economics in general. And that's what I want to talk a little bit about today. So I first have to put up a disclaimer. The company, lawyers insist on that. Everything I'm talking about, some of the platforms I will present as evidence and as things are available to be used in the clinic and for diagnosis, but some also are not. They're simply going to be platforms that let me drive home a point. So what I want to start with is we're trying to overcome an old paradigm in genetic testing in the oncology space, and that's simply using, where are we going here? How do you go backwards? And that's using single gene testing to point to a particular mutation or whatever. That's normally sequentially done, can take a very long time. So we want to overcome that because that simply can't keep pace with what's available today. And that's the fact that there are many, many hundreds of therapies now based on specific tumor types, specific mutations within those tumors, and the precision oncology, the precision drugs that are available for those. So we have to overcome the fact that it takes too long to get to the end result using single gene testing. And obviously the foundation for precision oncology that I want to talk about is NGS based. I don't think that comes as any surprise to anyone sitting here. So the challenges that are typically faced, we've already heard about many times today. That's simply the fact that there are a lot of biomarkers out there. We need to be able to assay those in a very efficient and timely manner. We need to do this within a budget, and that's very important in this particular region of the world. And we also need to do it in a very, very timely manner. Now, the general barriers that are perceived to NGS testing, obviously we all know about. It's perceived to be quite slow. Turnaround times can take up to a month. It can be very complex. We've just heard about that in the panel discussion. It's perceived to be very costly, and it's perceived to be limited in scope in terms of patient access. So I hope to convince you over the next 10 or 15 minutes that platforms are available to overcome many of these perceived barriers, and all of that revolves around next generation sequencing and the fact that you can actually test for multiple biomarkers simultaneously. I wouldn't be standing here if I didn't want to talk to you about this vast organization and everything that we have to offer in that space. Very fast, very efficient, easy to use. And I hope to present to you the information that we've generated from the real world evidence that we can overcome all of these barriers. So rapid comprehensive profiling is actually something that everybody's working on today. I wouldn't be, and I want to just present one particular site that decided to do something about this and see if they could do it at a point of care. And this study comes out of Canada, the Sheffield and his colleagues. They actually set up an automated NGS platform within an established pathology lab. So the lab routinely did immunohistochemistry, FISH, and things like that. They put an completely automated, complete workflow system within that pathology lab. It was then run by the pathology technicians, so no high-end technical expertise was required for this. What it allowed to happen was that at the same time as the samples were sent to the pathology lab for routine processing, they were also sent to the automated NGS facility, which was actually within the same room. And at the same time as the pathologist was given the standard pathology results, they were also presented in many cases with molecular profiling results at the same time. This empowered the oncologist very rapidly to make a very informed diagnostic decision. And the key take-home point here was they actually reduced the turnaround time from diagnosis to patient being put on a proper therapy down to around 3 days. That's very, very rapid. And quite often in this workflow system, the pathologist was presented with every piece of information they needed to make an informed decision at exactly the same time. So it can work in a real world setting. The study out of Canada here proves that beyond a shadow of a doubt. Now, the most important aspect of this, in my mind, is what happens to the patient. And what we're trying to do with targeted NGS and then directing therapies is overcoming the traditional therapy where one therapy for all patients, chemotherapy, immunotherapy, that can help a certain percentage of patients, but it can also be detrimental to some patients, or it can have absolutely no effect on other patients. What we're aiming to do with precision oncology, again on the right-hand side, is to simply identify what is the driving mechanism behind particular oncologic problems, and if a therapy is available, to get that patient on the therapy very rapidly. So the green patients get the green drug, the blue patients get the blue drugs. From there, I will present evidence to you to show you that that has a tremendous outcome on patient survivability. Now, this is a completely biomarker-driven a storyline. On the left-hand side here, sorry guys, on the left-hand side, you can see in the last 20 years or so, the number of oncologic studies that incorporate biomarkers has gone from below 20% to well over 60% now. So biomarkers are important for everything we're talking about. That is now driving the development of precision therapies and drugs in the world. You can see in the last 12 years, the number of precision therapies that are available in the world has gone from below 10 to now over 300. So a biomarker-driven field of study has now led to the development of many, many, many precise therapies in the oncology space. The most important aspect of this is the fact that it needs to be done very rapidly, as I've said. The reason for that, if we use non-small cell lung cancer as an example, is the fact that by the time the patient presents in the clinic, quite often their prognosis is very, very dire. Quite often within 6 months, many of those patients succumb to the disease. But with timely diagnosis, getting on the correct therapy very rapidly, we can improve on that. Now, part of the take-home story here is the closer you can get the NGS testing and therefore the targeted therapies identified to the point of care, the better the patient outcome is. So if you look at in the United States and NGS for outsourced versus in-house testing, as I showed you from the Canada study, we go from 1 month down to just 2 to 3 weeks. That now allows the oncologist to get the patient on the precise drug that's required at a much, much earlier stage. Now, again, the comprehensive genomic profiling is capable of looking at almost any type of driver mutation that happens in the DNA that results in an oncologic situation. Whether you're talking about small nucleotide variants, loss of heterozygosity, copy number variants, it covers it all. So it's a very comprehensive way of diagnosing what's going on in any patient and what is driving the therapies. Now, the key is there are multiple platforms for next generation sequencing. I point you to a Strata study from a couple of years ago now to the fact that they compared the feasibility of using an amplicon-based system to a hybrid capture-based system. About 22,000 samples were assayed for their suitability when processed to be analyzed by amplicon versus hybrid capture technologies. And what you can see on the left-hand side of this slide is that about 95% of those samples were suitable for amplicon-based diagnosis, whereas less than 50% of those samples could be used in a hybrid capture system. Now, what was the driving force behind this? It was simply the sensitivity of the amplicon-based system. It only took a very minimal amount of tumor tissue, therefore a very minimal amount of nucleic acid going into the system to make it a reliable readout compared to hybrid capture. And that's already down to around 15 to 20 nanograms of nucleic acid input, resulting in about 95% of the samples producing a workable result. So sensitivity is yet another aspect that must be considered when you're talking about next generation sequencing technologies. Again, this massive corporation that pays my salary has many, many assays in this space. This is kind of a follow-on from the workshop yesterday where Dr. Niraj was talking about a very comprehensive assay platform. Greater than 500 genes based on RNA and DNA can be tested. You can get very sensitive results without compromising on the testing success. The bioinformatics gives a very comprehensive readout of drugs that are available for any targeted, for any identified mutation, as well as pointing to clinical trials that the patient may be eligible to move into. It's very automated and hands-on, requires very little advanced technological training to be able to be used. And again, I've already highlighted the fact that input requirements are very, very low for an amplicon-based system. I won't go through this slide in detail. We have a booth here, but the platforms that are available on this technology are quite vast. They reach to many, many of the cancers. Very, very comprehensive is simply the word to use. Please stop by the Thermo Fisher booth if you want more details on all of the platforms that are available. Again, I want to point out one other thing. This particular platform that Thermo Fisher uses, it's called the Express Test platform, is actually not available here at the present moment. I hope at some stage it will be. But the driving message behind this, the technology needs to be available to target clinically actionable mutations. If you find an unknown mutation, an unknown fusion in your system, what do you do with that? Well, the unfortunate situation is you don't do anything about that. That might be the driving thing behind what's causing your patient to have the affliction, but you can't do anything about that. So the most important thing is testing for clinically actionable mutations. Our Express test covers all clinically relevant biomarkers in the lung cell space. By all, I mean 100% of what is known today. Aside from that, in other solid tumor-based systems, greater than 85% of all clinically actionable mutations are identified in a very rapid test. So platforms are there to do it. I'll just say the platforms are used worldwide. Here's 3 different studies from North America, Europe, and one from Japan, where the systems were used. That number of samples were tested. Success rates were 95% or so. The turnaround times average 1 week or less in all of these samples. Actionable mutations were detected in a great number of those samples, and the bottom line is Applikon-based systems work worldwide. It's not something that requires only a very limited thing. It can work anywhere. Now, all of this is well and good, but I told you my role in this vast organization is actually to generate real-world evidence that it makes a difference. And that's what I want to run through. Some very recent findings that Medical Affairs within Thermo Fisher has teamed up with some other organizations around, and we've generated some very nice data in terms of what it actually does for the patient. So here's a very simple study from 2020, the end of 2020, simply showing that patients that receive a biomarker-driven therapy have a better prognosis for survival than do patients on the left-hand lower curve, blue one compared to the purple one, that don't receive any kind of biomarker-driven therapy. Now, we've moved on from this study now. We know that if we get biomarker-driven therapies, they're beneficial for the patient. But if we move on, we overcome this problem. And the problem is that without biomarker-driven therapies, the patient simply goes on the left-hand side of one of my very early slides, any type of therapy, chemotherapy, immunotherapy, an autonomous situation where it's one treatment for all. That's the inner circle here. But if for some reason you can then do some type of molecular profiling, the hatched lines around the outside demonstrate that when that was available, the patients on the outside, 52, 29, 49% were then switched to a precise therapy. Now, this has a drawback that it takes too long. Patients start on chemotherapy or immunotherapy, a precise drug becomes available. They now must go through a recovery period before they can get on the precise drug. And what I'll show you in the next couple of slides is some of the most recent data that we've generated regarding what the outcome of actually getting that patient on the therapy rather than having to switch the therapy is all about. And that's diagrammed in simple phase here. So the study used about 500 patients on the left-hand side here. You've already seen the right-hand side multiple times. That's outsourcing versus insourcing. The blue graph shows patient outcomes, survival probabilities for patients that went straight to targeted therapies, the blue line. The pink or red line, as it may be, are patients who either went only on chemo or immunotherapy or went on chemo or immunotherapy and then switched to TKI type based therapies, precision therapies. What that leads to is fantastic patient outcomes. This is what makes me proud to stand here and talk. This is actually benefit patients in a very, very big way. Now, if we take a deeper dive into the data and simply separate the bottom graph into those that got only chemo and immuno, which is the green line, or the middle graph, which they were switched to a precision therapy after the initiation of chemotherapy, again compared to those that went straight to the precision therapies. What you can see is that immuno and chemo has very dire outcomes. It gets a little bit better if we switch them to the precision therapies, but again, getting the patient on a precise therapy as fast as possible has tremendous effects to help patient outlook. This all comes down to turnaround time. Very, very fast testing, getting the patient on the targeted therapy as fast as possible improves patient outcome without a doubt. Now, this goes beyond just patient benefits, and this is the last point I make. I only have one more slide. I've already shown you that patients benefit tremendously from getting on the proper therapy as fast as possible. It also has great economic benefits for the health system, for waste generation, for almost every aspect of the healthcare system. It actually costs less money in the long run to perform what is perceived to be a slightly more expensive NGS-based test initially, but in the long run it saves money. One NGS test is less than 1 month of chemotherapy, price-wise. So again, health economic benefits of doing the proper testing, getting the patient on the right therapy as fast as possible benefits everyone. So lastly, I just want to re-emphasize the fact that I get out of bed every morning trying to do what I call democratizing NGS testing. I want to make NGS testing available to every patient in the world. There should be no one who needs the test, therefore needs a precision therapy, that should not be able to get the test. We're driving the cost down as fast as we can. Some people question whether the platforms work very well. I can tell you straight up, we partner with every major pharmaceutical company in the world as companion diagnostic partners. There is no doubt that they believe the systems work, and they obviously are very keen to be pointed toward their drug when necessary. So with that, I'd just like to again thank everybody for the invitation to come here and let you know that some of the real world evidence out there does point to the fact that NGS is critical, and that the fastest turnaround time you can possibly generate is an in-house workflow system at the point of care, and that has great benefits for everybody. So with that, thank you very much.
Thank you, Dr. Mike, for a nice presentation on NGS and precision medicine, and that to the delivery in time of the results, and that can be benefit for the treatment. So if there are any questions, maybe 1 or 2 questions, you can take it. No questions, then we move on to our next speaker.
Thank you.
Yeah, thank you. Dr. Prashant Vagali from the Stars Life Science, Bangalore, and his topic is homologous recombinant deficiency testing, his experience from India.
Good evening, everyone. After a talk by the Mike, so my particular presentation will be more focused on the precision oncology, on the more focused approach of homologous recombination deficiency and Indian experience and all. So in the whole of this presentation, I will touch upon the science or the biology of the PARP inhibitors and what exactly we are doing as a more than we are the first company to start the HRD assay in India and all. So, and also with a more focused on the therapeutic angle, if any questions are there, so probably I'm interested to address those questions and all. So before that, the Strand is attending this after a quite long time, I should say, although we are in existence as a spin-off from Indian Institute of Science in Bangalore and all. So this is the one slide, 2 slides I just added to reintroduce the Strand, and I had the oncology section, and from the lab also we have more than 15 PhDs running the assay in Bangalore office and all. So we are into the different areas in the NGS and the different clinical conditions or the primaries and also like breast cancer, lung cancer, colorectal, ovarian and all. So the main focus of this is the genomic instability. So genomic instability, because you would have heard the MSI, the microsatellite instability and all, but here instability can be of genomic instability can be because of the different reasons and all. So one of the importance is what exactly is the genomic instability is. The reasons or the effect of that is it can be a incomplete DNA repair, or it can be a genomic instability which leads to the cancer, hereditary diseases and all. So because in the DNA, because if you go back to the central dogma of molecular biology, you will be having the DNA polymerase and all. So the body or each of the cell has the inherent ability to repair itself and all. But here in this case, the repair mechanism is incomplete, that leads to the instability, which is as a cellular response damage and loss of damage of DNA repair and all. So this leads to the two important mechanisms which have been very common for the past, I should say, although the recent earlier publications were there, but recently with the new drugs coming in the market, so for the past 2 to 3 years and all. So homologous recombination repair mechanisms or the pathways, and the homologous recombination deficiency and all. So in the repair mechanisms, basically is a DNA mechanism where the body or a cell has a mechanism to repair itself of the DSB, that means the double-stranded breaks in the DNA. So in the deficiency, there will be occurs when the genes regulating the recombination pathway are damaged and cells are unable to fix itself. So this is where the reason for the cancer, or efficient and error-free pathway for double strand breaks repair here, and here in this case is where the repair is error prone pathways. That means the one cell has this particular error, so it gone having an amitosis and uncontrolled mitotic divisions and all. Third important difference is they maintain the genome stability and tumor suppression function, because in the body we have the apoptosis, so that way wherever there is any incomplete kind of recombination and also the cells will get vanished by themselves and also over the process. But here in these cases, it will lead to the larger genetic aberrations and the genomic instability. So, and this provide the critical support for DNA replication and telomeric maintenance, and here there will be a cellular sensitivity, and later on I will able to share with you how the loss of heterozygosity, telomeric allelic imbalance will happen to them and all. So the main important point here, although there may be a greater innovations waiting, and the clinical trials, probably some of you are also participating in the clinical trials and all, at least in India also there are many. Throughout the world I have at least can list 21 clinical trials which are all have using the PAPA inhibitors for different clinical conditions and all. So here in this case, we are basically in India is having only for the breast cancer and ovarian one, and ovarian high grade serous cancers and all. So the testing strategy earlier was the full gene sequencing of BRCA1 and 2. So when it was there with the Pfizer tamoxifen and all, so this was what we used to recommend from the clinics and all. So mutation detected means, okay, provide the genetic counseling to them, and then either it is a hereditary one or they provide to the mutation specific tests and the counseling to the relatives and all. So no mutation detected means, okay, then go for the deletion duplication testing. It can be by the MLPA and all. So then again the mutation is detected, and then go for the germline panel and all. So looking at the more genes than the BRCA1 and 2, because reports started coming only 26% is having this, and there are maybe the other predisposition conditions and all. So and if that doesn't come also, then the whole genome sequencing and all. So this was the trend, and during this whole process they were missing some of the important conditions and all. So with that, then in the 2020 NCCN came out with the guidelines where they say that the important conditions, say the HRR and HRD can also be able to use, and those are also under the approved marker list or a diagnostic algorithm and all. So then from the different research institutes, if you all send the PARPI first paper came long ago, around 10 to 15 years ago and all, but the use of that as a drug targets came recently, I should say, so maybe like 2 to 3 years and all. So here basically, since it is for the somatic mutations and all, so FFP is the one which is very important for us, and testing is tumor BRCA mutations. So with the presence of mutations we are going to test and all, and genomic instability is these three main pathways or the phenomena which are responsible for causing the HRD. So it may be loss of heterozygosity, telomeric allelic imbalance, and the large scale state transitions and all. So later on I will just explain the comparison also. So the score is what we are analyzing at Strand is the GSS, genomic scar score. So later on I have a small one table showing the GIS score coming from the Myriad Genetics and the other place. So here we basically calculate on the three important concepts by using the artificial intelligence. So our computational, as all of you probably have used the services of Strand for its expertise in the bioinformatics and all. So we use the length of copy number, type of copy number, and the site of copy number, and together these details will have the GSS one. So the cutoff point is more than 50, whereas Myriad Genetics goes more than 42 and all. So this is the small slide which need to be understood, because we get a lot of samples from here, this region also. So detecting the SNBs, and also here people always ask whether it only detects the germline or the somatic mutations of the BRCA1 and 2. Since we are collecting the FFP tissue samples, so it collects both of the mutations in our calculations and all. So in the GSS score calculations and also how the clinical interpretation goes is, so pathogenic variant, of course, if the BRCA status alone is positive, one of that, it will be called as the HRD phenotype positive. And if the tumor BRCA is negative and GSS score is still less than 50, then it is considered as negative. So that means you will appreciate that there are two important aspects to that. That means the BRCA1 and 2 also play a very important role in calling a HRD phenotype. At the same time, the GSS score and all. So then you might ask, what are these GSS score calculations happens and all. So because in the whatever the kit we are using is, it takes into consideration how these copy number variations are making a genetic alterations in the other portions or other than the BRCA1 and 2. So that's where the computational power of the test increases its ability to detect more number of the patients in our sample and all. So in the HRD assay here, so this is just a one example I took for the discussion purpose and all. So being the first lab, we were, every lab from the AstraZeneca who sponsored the test and all were all looking at us for what exactly is happening in the Indian patient population and all. So we are the first lab to offer this test in India and also, and the only provider. So now there are 2-3 labs which are all doing this same assay and all. So here basically the accuracy was 100%, the positive predictive accuracy and all in terms of the single nucleotide variants and the indels for BRCA1 and 2, and also the deficiency and the GSS positivity concordance was 95.7%. And of course the specificity was very good, both of them are 100% in terms of the analytical concordance and all. But the important one from the clinical perspective, what I would like to show is, let's assume that you have taken the FFP tissues and done only the BRCA1 and 2. So here if you see the proportion of that, so I'll just explain. So sample reported and also I have not considered the sample which have been failed, so that I'll take it in the next slide. And also if it is only the tumor BRCA1 and or positive, then only the 10 samples would have been positive, or rather 55 plus 10 would have been positive. So, and if both only GSS is positive, means then only 69 would have been positive. And if both are positive, then this is 55. So what does it mean is, in terms of the total one, so 134 what we have is the 57.57% of the population is having the positivity rate of HRD phenotype and all. So this clearly indicates that if we go only by the tumor BRCA testing, we might miss out those who are all the patients who are all getting benefit out of the PARPA inhibitors and all. So earlier when we were doing in the last year, there were only olaparib from AstraZeneca. Now I have been seeing, and I've been, of course that's not the scope of this conference and all, the rucaparib and nicaparib and also are getting kind of the permissions to either a maintenance or a first line therapy drugs and all. So in this perspective, it's very important that we consider the GSS score as a part of our assay. So not to go by only the tumor or somatic BRCA1 and 2. So this is just a simple workflow for the purpose of discussion and all. So the sample what we are taking now is the FFP blocks and all, because it's a somatic mutations what we are identifying, and in that there are two important assay are happening. So in our kit, and the bioinformatics is where is a very critical one. It uses a specific proprietary pipeline for this, and the HRD report comes and all. So here the important one is the tumor tissue sample, so collection and shipping, because I was just seeing the panel discussions and all. Yes, it's very critical to have, like always we say in our computational, that the garbage in, garbage out and all, so we don't want that. So that's why I was very happy to see that the start with the gold kind of the statement from the Dr. Maushmi and the other panelists and saying that. So tumor genomic DNA extraction, then the HRD NGS assay will happen. Then these are the routine steps of the NGS, and then BRCA1 and 2 variant identification, classification and interpretation, and the GSS score classification and all. So, and also we have the panel of 4 board certified genetic counselors with Strand, and we made it a very mandatory, because some of these things need to be explained to the patients in local language. And however, we are also engaging the doctors with regard to this, because this is a new science and need to be understood for them before starting the therapy. And also of course the therapeutic treatment regimen is very expensive also. So this I highlighted because wherever I do the CMEs, I always get the question saying that why our sample got rejected and what is this. We have been doing the IHC, we have been doing the using that sample for other purposes and all. Yes, certainly these are very important. Of course the age of block is 18 months, so we are not saying more than that will be completely, I was just seeing the Dr. Anuj's presentation also and all. So these are the ideal sets. So what we exactly promise, the commitment, when we are taking it at time of TRF have been done and all. So this is the ideal one. So where there is a better chance, say 95% or of the getting the good DNA from that. Tumor content 30% also. So H&E report need to be there along with the FFP tissue blocks and all. And the DNA concentration is more than 15 nanograms and all. Amplifiable DNA has been, there are been only 3 earlier we were considering and all. Some labs now do that and all. But please remember, NGS, there has been amplifiable DNA has playing a very important role and all. So we have made sure that amplifiable DNA also is more than 20 and all. So we have considered like 10 and all. Then it has failed in the library to get a kind of a quantification, because this has a lot of, the type of copy number variations and all, which is very important and all. And of course the DIN score of 2.5 and the library concentrations and all. So this is the simple comparison, so that because there are so many samples here from I think Ahmedabad and Surat, Baroda and the other places are going to US also. So wherever they are non-critical, I am not really sure how they make the choices and all. But here what we are trying to compare is with regard to the TAT, 14 days is very critical, because although they say 14 working days, but when the sample goes and the logistics and all, we are all losing. And also see the genomic instability one, what they are doing calculations and all. So our GSS score exactly gives in terms of percentage, like 55, 60, 65, 70 and all. So there you will see only HRD phenotype, positivity yes or no kind of things and all. So I think that's the summary of it and all. So we are in the booth number 8. So if you have any questions, I'll be here tomorrow also. Thank you very much for your attention.
Thank you, Dr. Prashant, for sharing your experience and giving you a nice presentation.
Ma'am, I have one question. Sorry. Sir, I have been following this HRD for quite a last 2 months, and I came across the one question which is related to particular this test. So that is like when you are providing the GSS score, that's I know that which test is this. So some of the literature have reported like greater than 26 base pairs of deletion is not validated, like whether this test is properly detecting it or not. So what are your views on this, because you have already experienced this panel, means this particular kit and test. So whether it is detecting now or not, because maybe that is been reported like 1 year back or something like that. So what is the status now?
Yeah. So you mean the larger deletions,
yes,
right?
Larger
yeah.
deletions.
So larger deletions, yes. So if the larger deletions are making a changes to the copy number variations, then it is detecting. So because if you see the complexity of the assay, there are the deletions and the single nucleotide variants of the BRCA1 and 2. So if there is a larger deletions in the BRCA1 and 2, it is detecting. But if it is out of the BRCA1 and 2, then it is not detecting. So, but having said that, if it is impacting on the copy number variations, then it is used in the calculation or a computation purposes and all. Because there are two important aspects. That's the reason if you compare the Myriad Genetics only LOH, it comes out with that kind of a limitations and all, compared to this one, which is more of a using the AIML, so it computes other copy number variations like type of copy number variations or a site of copy number variations and all. So we have done that comparisons of those using the computational one. If there are less number of the copy numbers, what happens to impact on this and all. So that's why always they will ask that why your GSS score is more than the Myriad Genetics. So we always say GSS score is more is truth because we use that component of the copy number variation calculations, the summation of that in a multilinear regression, which adds on to the value. So that's the one important one. Yeah.
Thank you, sir.
Yeah.
Thank you, Dr. Oh, you have a question.
Yeah, I want to know the difference between somatic mutation and germline mutation. Whether there is sampling difference or procedural difference.
Yeah. So, sir, that's what I always say that a basic fundamental one, somatic mutations are coming from the somatic cells. Germline mutations are coming from the germline cells. Having said that, somatic cells also will have the actively dividing cells, which are all inherited from the germlines and all. So that is the reason when I say, if I extract from the FFPE tissues, it is the sum total of the representation coming from the germline mutations and the somatic mutations. But on the corollary, it is not correct. If I extract the blood DNA, and means DNA extracted from the blood, and do the DNA extraction, then I will only get the germline DNA and all. So when we say FFPE tissues, only our library kit or any library kit will not have the ability to separate that which is coming from the germline, which is coming from the somatic, because in that particular assay, depending upon the patient history and a clinical history, we need to make a decision whether I need to take the FFPE tissue because it's a metastatic breast cancer or any advanced carcinomas and all, or I need to take the blood because here there is only a counseling need to be done to the relatives or to the patient and all.
Means what I could understand that for somatic mutation I need tissue.
You need tissue, yes.
For
Soma basically is a body tissue.
For germline mutation I need blood sample.
Blood, yes.
Procedure is the same.
Procedure is the same. In extraction means extraction kit and all. So when and it on the contrary goes means a healthy woman walks in, you will always prefer a blood sample because there she wants to know the predisposition for the cancer and all. But when you find it in even in mammograph or any of them or in your ultrasound and all, you already know that there is a lesion or a kind of thing. So either you go for a liquid biopsy if it is not a non-resectable tumor and all. So if it is a tumor can be done by any of the FNAC and all, then we can then the tissue biopsy is the best one and all.
So for a patient who is having already a tumor or see a breast cancer, and for that I need germline mutation, I need say blood sample. For somatic mutation, I need tumor tissue.
Okay. So the choice of a test for a person who is already having a thing is two things. One is if she has any, means you want to advise her on the hereditary cancer related, HBOC and all, then you can take the blood sample and all. If you want to have that to do a treatment, the patient on the like a choice of the therapy or a patient management, then you require FFPE tissues. Yeah.
Any more question? No? Okay, thank you. So now the next presentation is by the Mr. Baswar, and the topic is a new generation proteogenomic tissue profiling for the improved molecular pathology.
Thank you so much, Chairman, Sir and Ma'am. I can see people are leaving, but for the people who are still here, I'll try to make this next 15 minutes interesting enough so that you learn something new. Since morning, we have been talking about next-gen sequencing, we're talking about DNA, we're talking about mutations. In the panel discussions, we have also talked about how exactly we can pinpoint what you're trying to look for so that we do not miss out information. So before the last talk comes in, I'll try to do something new. Encounter, DSP, Cosmic, these are the new names in the area of molecular pathology. And I represent a company called NanoString Technologies, and we come as a specialist in analyzing RNA, which is a little different than next-gen sequencing, DNA mutations, and everything else. So we are a company formed in 2008, and over a course of last decade or so, we have specialized in delivering technologies which can look at RNA and now protein with a new concept for the pathologist called digital spatial profiling, a much more improved area to look into, not by doing 3, 5, 10 markers, but you can actually look at hundreds of proteins and thousands of RNA together on a tissue structure. That's the power of the technology which comes in. So pathologists would always love to see something. For them, seeing is believing. What if you are only limited by multiplexing abilities? You cannot go on having multiple slides together to get as many markers as possible to analyze your tissue in the best, precise, and confident way. To do this, if you get an ability to look at hundreds of markers of your own choice, who can decipher the tissue biology into a very different dimension together. Genomics is called spatial molecular profiling technology. It enables you exactly pinpoint the area of tumor microenvironment, and the immune cells, everything together at one go. It just doesn't look into protein. It can also give you the choice to make a whole transcriptome analysis or a defined set of genes as you wish for. This is a fantastic review article. For the time being, I'll not elaborate on that, but the highlighted one is actually talking about what are the limitations of the current IHC-based methodology and what this NanoString's new offering called digital spatial profiling technology can offer. Again, I repeat, it can give you today around 147 protein marker of your choice and possibly a whole transcriptome or a cancer genome atlas as you wish to look for. The expression profiles can give you an extremely important amount of information. You are the gene of choice that you are looking at, and multiple different ways. Either you can apply this platform as a discovery module, or you can actually define gene expression profile, make a specific score, and I'll give you a couple of examples how they are directly relevant and are being used globally for this score to analyze specific disease molecular markers. Is this very different? Are there a lot of relearning process involved? Probably not. As an IHC specialist, you are probably going to do exactly the same, similar kind of treatment on your tissue types. You take your tissue, put it on a slide, fix them up, stain them with a morphology marker, and then additionally, you're giving profiling reagents to profile thousands of RNA and hundreds of protein together. So almost the first phase of this analysis is exactly similar. However, the technology allows you to pinpoint specific area of tumor microenvironment, immune cells, B cell, T cell, whatever you like to do that. So every rare cell, every demarcated microenvironment can be precisely looked into and figure out of your choice when you analyze them without having any ambiguity of choice. One quick example, this was a study done on which a drug of choice is being given and a progression-free survival was looked into. On the x-axis, they are looking at how much amount of immune cell is being infiltrating in the tumor environment, and on the y-axis, you get the PFS. The first part of the block, if you look at, a smaller amount of tumor infiltration, and the PFS is very sporadic. On the right-hand side is more confusing. You find more amount of infiltration cell with the x-axis higher one, but there are still the lower bottom part, which is a very narrow zone with a blue bar. They are having more tumor infiltrated cell, but the PFS is lower. This is very confusing because this was supposed to be the different one, because if there are more infiltration, the drug is supposed to work and there should be a more survival benefit. This cannot be resolved by a normal course of gene expression or a protein analysis. I'll show how we do, but before that, a quick table. We use different set of markers, morphology markers and protein markers. This was a bit old study published, so that time there was only 40 protein markers. As on today I speak over here, 147 different protein markers of your choice you can actually simultaneously analyze together. Yeah. This is how you can actually seeing and believing picture comes in place. You visualize them morphologically by staining them by staining markers or a morphology marker, and then you push across around 147, it could even 10 as your choice, but it gives you an absolute visualization. Coming back to the story which I was talking about, on that you find some of the cases whereby more amount of immune cell, but still the PFS was low. Look at the two distinct differentiation. In the first case, it actually represents why there are immune cell and the tumor cells together. In the next case, you find they're actually infiltrated. Here they are separate, they are more infiltrated over here. So the visualization gives you an absolute clue which cannot be resolved by looking into a normal gene expression analysis. That's the power of the technology for you to look at the tissue with all marker of your choice, whether you're talking about RNA or you're talking about proteins of your choice. Multiple such studies have been performed, but what's the beauty of this technology is that it not only allows you to visualize, but give you the complete control to look at those circular areas. Once you visualize, it can give you to control the pin and pick the areas. You can make different kind of geometrical sizes, like the rounds over here. As much as 12 different regions of interest, you can actually mark on the tissue based on the color-coded areas, and that will precisely land you on your immune cell, rare cell like B cell or specific T cell type based on the marker of your choice, or even a specific area which are more dense for a particular marker. So essentially it allows you to pick and choice of your area and then exactly extract either protein or whole transcriptome only for those cell types. You're not going to make it a diluted effect, you're not going to make an ambiguous choice. NGS is a great technology to cover up thousands and thousands of RNA, but we allow someone to visualize, identify, pinpoint, and then do the analysis, so that you never fail in terms of understanding it. This is a fantastic slide to actually recap what we learned so far. Bulk analysis is like a fruit smoothie. People jumped around single cell biology concept. This is like that fruit salad. And now we're actually talking about the technology whereby you can visualize, look at the picture. There may be scenario on which a specific choice is available in some part of the pizza, some of the pizza doesn't have. So you can now exactly pinpoint which slice to take up so that choice of your toppings are actually available over there, right? So the technology what we brought in, it brings down the ability of high plexity as well as visualization together so that you can pinpoint exactly where you have Dil mange more. You get exactly as much as you can. Whether you talk about protein, whether you talk about RNA marker, whole transcriptome or genes of your choice, that's the ability what it comes in. This is not something I need to decipher here because as a pathologist, you know how exactly to do that. What rather I will talk about is that once you infuse all the reagents ready to go, and one more thing before I forget, somebody talked about elaborately on green value, RNA quality. You all work with FFP sample. There is always a fear, will my sample work? The RNA quality is really bad. We have established numerous studies on which 25-year-old FFP blocks have been used to give more than 80% profiling data. Green value as low as 2, below 2, still give you more than 50% of data. That's where the uniqueness of this technology comes in. I'll not elaborate how do we achieve it. We have our partners both with BioInnovation available downstairs. You can meet them and they'll love to actually express that. But what we do essentially, either we attach an antibody tagged with a barcode or a RNA probe tagged with a barcode, which are clippable by UV to pinpoint and figure out location-specific barcode arrangement. That's why we call it spatial arrangement. It gives you exactly direction force to pick up the data and give you the proteogenomic approach to analyze together all marker of your choice. This is how the data look like. Again, I'll not go into elaboration, but as you can see on the right-hand side of the panel, there are different color coding for morphology to differentiate and visualize your tissue, and then you can have different choices to pinpoint an area, pull out the markers, whether they're protein or RNA. And this is the engineering how we can do. Just to worth mention over here, once you visualize, the AI-based technology software model can allow masking different colors together. One time you get tumor microenvironment, one time you get your tumor tissues, other time you get rare cell population. Exactly, distinctly, differently, stage by stage, so that you never get pull out, diluted and ambiguous data ever or so. It's a bad slide, but it allows to allows me to pull down the summary numbers, summary wordings, reliability, multiplexing, and absolute future direction. We are evolving every day with numerous studies. Two more quickly. This is, we talked about spatial biology, but as a RNA biology expert, we have a system called nCounter, which actually gives you digital gene expression data that can work on pretty bad sample, as I talked about, 25 years old FFP blocks, less than 2 hour in value, RNA quality will never bother you and never take your sleep away. You are still going to get that. We talked about a lot, even on the mainstream, people talked about Oncotype DX, Prosigna, and MemmaPrint. Prosigna is actually a prototype and commercially available test based on PAM50 gene signature, which was developed on nCounter gene expression portfolio. The backbone structure is called BC360. The part of this BC360 panel is PAM50, which is coded in red. But apart from that signature, which give you recurrent score, you get enormous amount of other information to look at: grouping, TNBC analysis, time series analysis, single sample analysis, and different kind of signature separately. It's a 770 gene panel. Apart from that PAM50 signature, you get a lot more data coming from there. You are open to evaluate that for your choice as well. One of the other examples, Dr. Agarwal was just talking before I actually came up over here for my talk. He elaborately talking about DLBCL-CO classification. It doesn't require more elaboration, but this is the most confusing area in DLBCL, that cell of origin is a prognostic marker, and gene expression signature, which is well differentiated now based on NanoString's platform, can give you the ability to well demarcate it. And this is just not claim. This has been published so far. Only 23 gene expression panel, very, very lower false positivity rate or misclassification rate compared to IHC, and it's a ready-made assay for you to directly pull out from your stock and analyze them. This is another published paper, and they are talking about that evaluated complete the clinical orientation of this assay, and I love to share this publication on you. They have shown a complete confidence that this goes pretty well in subtyping DLBCL, which is either by NGS or by other methodology like IHC, is not a very good assay so far. And the proof of that is the commercial evaluation being done with Celgene, and they have a drug of choice as a companion diagnostic based on the DLBCL subtype classification on nCounter itself. The last two slides before I stop, we talked about visualization on that scale. We talked about scale. Now we are going to talk about single cell biology and spatial biology together as a concept. At one go, you can analyze millions of cells to create billions of transcript of data. Amazing data analysis software is available to give you cell by cell analysis. As you can see, you visualize, you orient them into a spatial structure, and you can create billions of transcript data at one go. And remember, even the RNA quality will not matter. At this moment, what we offer a large 1000 RNA complex multiplexing analysis. We can do FFP, fresh frozen, TMA, organize multiple different variety of sample type, and again, it's a multi-omics approach. 1000 different RNA, as on today we talk, and we have a roadmap of around 3000 RNA which can be done together. We can do 100 different proteins, and as you can understand, it's a trade-off between what you can achieve at a single cell resolution and the higher level of transcriptomics. The previous one, which we call GeoMx, can do full transcriptomics analysis, but here you can actually achieve a single cell level with a spatial information, 1000 different protein and 100 different proteins. I can go on, but I might stop because the alarm is here, and I must honor the next speaker to come over here and end the day. Thank you so much for being a lovely audience. If you have one question at least, I'd love to do. Otherwise, thank you once again.
Is it related with nanotechnology, right? Its name is NanoString.
The company is called NanoString because you use nanoparticle and the fluorophore particle to do that. Yes, it's of course nano because you are talking about micron level resolution of million cells which can be visualized and analyzed at a multi-omics level. We can have a more elaborate discussions, but yes, it's definitely nano on that scale, but nothing to do with NanoString as the company name comes in.
Yeah. Name of the company is NanoString.
Yes, the name of the company is NanoString.
It is not absolutely not related with nanotechnology.
It is actually, because we are talking about million cells and infusing billions of probes inside, visualize them together, and it has enormous amount of application. You put a drug and you can visualize whether the drug has gone, bound to a particular protein or not. You can visualizing the protein at a cellular, subcellular level as well.
What happens in nanotechnology?
Will you allow me to catch up to a little later? That will be better, and leave us on the next speaker over here. Thank you so much, sir.
Yeah, that was an excellent presentation, and I think this spatial genomics is the need of an hour. Something beyond genomics is proteogenomics, which he has nicely elaborated in his talk. With this, I invite next speaker, Dr. Jayant Khandare. He will be there are basically two speakers for that, so I will invite Dr. Jayant Khandare. He is Chief Scientific Officer, Rectorius Innovations and Research, and he is the Managing Director. So, yeah, his topic is circulating tumor DNA and circulating tumor cells, the dynamics due and revolution of oncology.
Thank you very much. I think this is the last talk, very braveheart talk what we heard for the earlier speaker. I represent a company which is OncoDiscover and OneCellDx, and with me Dr. Gohar Shabi will be speaking half of this talk. CTC and DNA, circulating tumor DNA, the dynamic duo revolutionizing oncology. We are talking about precision medicine. I think precision oncology and the actionable drugs is the next future path. We are a company which believes in research, science, technology and translation in this country on phenotypes and genotype, which is accessible, affordable and available in India. OncoDiscover is a first-in-class, indigenously developed, DCGI-approved cancer patient monitoring blood test through circulating tumor cells in epithelial cancers. Who we are, we file patents, we innovate, we publish papers, and we, as I mentioned, do clinical trials according to the norms of Indian Drug Controller General of India. OncoDiscover has been developed with clinical mentors at Tata Memorial Hospital, Dr. Pankaj Chaturvedi and Dr. Kumar Prabhash, who gave a wonderful talk just below. And there is a lot of availability of data of circulating tumor cells in head and neck cancer patients in India, both at various stages as well as into the clinical interventions, and a lot of tutors around. We know that 90% of the patients, they succumb because of a metastasis. However, our focus is primary in the primary cancers. Therefore, the mortality index, especially in this country, is very dismal, and it is about 30% over to 60% in developed country. We know that circulating tumor cells has been spoken a lot, they have been evaluated, they have been limited, but circulating tumor cells 2.0, and we know that they are the fugitives on the run. And if we believe a seed and soil theory, that these are the circulating tumor cells in circulations, and they are the pesky fugitives which are on the run. And we do not know their transitions, we do not their EMT and MET transitions, but we can now isolate single cells and look for their metastatic signatures and possibility of disease progression far superior that we are aware 5 years before. So OncoDiscover, we are regulatory approved test in India. It is now MRP with 15,000 rupees. It is a patented technology. We just filed today, few hours before, our next technology in isolating circulating tumors as an extracorporeal device in eliminating all the disease burden through circulating tumor cells in cancer patients in the form of a dialysis. We have seen about 2,050 patients as on today in Indian systems, Indian phenotypes. We have seen 20,000 circulating tumor cells in clinical real-world evidences. We have done almost 1,000 samples in clinical studies, both in Tata Memorial Hospitals and AIIMS and other clinics in India. We give focus in head and neck squamous cell carcinomas with 500 patients, world's largest study, CTC as a biomarker. We have ongoing study in breast cancer 1 stage, ovarian cancer, that's an ongoing study, and we have a next proposed study with Dr. Pankaj Chaturvedi and Kumar Prabhash, who is sitting here with around 250 samples, and we are still proposing the clean objectives. Dr. Shabi is going to mention about what we do into ctDNA as well as multi-omics data. So the hypothesis one that always I like to mention, which is in the literature, that the presence of a circulating tumor cell is associated with worst clinical outcome, and that has been shown at various time points. and they play a very important role in extravasation, and we know that they have an extraordinary ability to survive into a peripheral blood. They have invasive ability and they can invade stromal surrounding very strongly, and they are a telltale signature of a metastasis, and there is no second hypothesis. So I think to make my story short, that we have to focus on an early med detections. We have to focus on MRD or minimal residual disease or molecular residual disease. Circulating tumor cells with all these properties which are mentioned on the board have been defined as the world's first liquid biopsy technology, which is now further extended to ctDNA or cell-free DNA. And we can predict actually clinical outcome or the prognostication in various cancers, especially in the colorectal cancers. And we're very happy that as an innovator company, we can today isolate single circulating tumor cells and look for omics data. I think these are the three major critical problems in oncology. As we mentioned, MDR, cancer dormancy, especially in the breast cancers. We know that the circulating tumor cells can be dormant for a decade, and minimal residual disease stands, I think, most prominent component of an occult disease or occult stage of a cancer. Dr. Pastun Kaji and many groups, including Klaus Pantel, have been actually showing minimal residual disease very strongly. So what is this technology? The ability of isolating circulating tumor cells is very, very expensive because you have to innovate materials, and we have innovated materials which are very specific, which are very evidently specific, and the tumor cells what we detect into our lab has an AI system, and we can scan patient samples and deliver a report to oncologists or patients within 48 hours. So this is one of an example of a multimillion cancer patient whom we monitored for 8 times. Unfortunately, the patient succumbed to the disease because the oncologist unfortunately did not took a very strong clinical decision. So sum up here for circulating tumor cells, we can monitor disease burden at real time, and we need to keep surveillancing these patients. We can now look for the CTC expression markers like PD-L1 when you do not have choices. We can look for the HER2, and these tests are under validations. Good tweet from Dr. Nirmal Raut with a good presentation of a colorectal or rectum cancer patient who was disease-free, did not have any PET scan positive signatures, sent a blood sample to us. We found 2 cells per 1.5 ml. And that's a panic actually both the patients and the oncologist would have. He took a decision to take a patient for MRI into the liver. There were 4 lesions. SIRT was done at Tata Memorial Hospital, and we monitored this patient from 3 circulating tumor cells to 0 cells. Dr. Colonel Amol Patel actually also posted these good questions very recently in a bladder cancer. It's not about just taking a blood sample, but it's taking a despair out of a cancer patient. This test is available in Pune at a very affordable cost. Thank you very much.
Now I invite next speaker, Dr. Gowhar Safi. He's Chief Informatic Officer in One Cell Diagnostics.
Thank you so much for giving us this opportunity. I'm the complimentary speaker of the day. So what we heard today just now was the CTC part, and what I'm going to talk about is ctDNA part. And it's very important, how we can use this circulating tumor DNA in precision oncology. And we have developed here 600 gene panel which can capture all type of genomic alterations which are very important in oncology practice, including fusions, indels, amplification, loss of heterozygosity, HRD, tumor mutation burden, MSI, and can be run on both tissue as well as liquid. This is typical NGS workflow where you have to design what genes you have to actually do the sequencing, then run through the routine NGS workflow. But what is the key question is when report is being generated, typically done by bioinformaticians and interpreted by oncologists. That's why the complex issue happens. So we have also built AI analytical tool which provides the combination of both analytical and chemical interpretation, which helps oncologists to understand and better interpret these results to make better decisions. The question is why we need comprehensive circulating tumor DNA. We know that tumor heterogeneity is the big issue, and what we have seen now, this is beyond actually, it's not just the local primary tumor, it's, now we know that intermetastatic heterogeneity, which becomes very important, and that's why that, tumor heterogeneity has impacted tissue NGS analysis, and that's why ctDNA based comprehensive analysis is very important. So for example, we had this 53-year-old ovarian cancer patient, and it has, the patient has done NGS by other lab, and what they have found is ERCC2 mutation and TP53 mutation. And they have found MSI high, which they tried to validate by, IHC, which came MSS stable, and treated with checkpoint inhibitors based on the previous NGS results, and patient has progressed within 4 months. And this patient was discussed in International Tumor Board, where I have offered them, we can help you to identify, some insights for your patients. So what we took is both tissue as well as, cell-free DNA. So in tissue, what we had the same 2 mutations, and tumor mutation burden was high, and MSI was stable in our case. And when we took this ctDNA, what we found, both those gene mutations suggests that, there's a concordance between tissue and the liquid biopsy, but very importantly, we found BRCA2 mutation, which suggests the patient could be benefited with PARP inhibitors, as it was an ovarian cancer patient. Also, we found STK11 mutation, which suggests why actually checkpoint inhibitors was not working. This is why it becomes very important. We always talk about financial toxicity, and we have to have an affordable testing. So affordable testing is not the question. Question is, you should have a comprehensive testing which can actually identify what treatment cannot work. Like in this case, 4 months immunotherapy, which costs about 4 lakh rupees. If they would have done it comprehensive testing, identified STK11, and they would have avoided that. That's what is actually financial toxicity. Second issue is tumor is evolving, and we know now that there's a lot of studies where they have done the comparison between tissue and liquid, where they have seen that concordance, discordance, and particularly this paper is talking about, if tissue is more than 6 months old, you miss 40% of information. This is because we know that tumor is evolving, and we know that we have to treat the cancer at the speed of cancer. And if we use an older tissue, and possibly we have old information and we are not treating patient right. And I have an example here. It was a 69-year-old female, metastatic infiltrated ductal carcinoma. And what we found here is, both tissue and the liquid. In tissue, we found ESR1 mutation and JAK2 mutation. If we look at variant refraction, which is in a, relatively looking at the load of the tumor DNA here, it was 1% for ESR1 gene, and it was 15.62% for JAK2. And when we did it on liquid biopsy, that ESR1 variant refraction was 2.59, which is 3-fold high, and JAK2 was 36.49. And this suggests that tumor is evolving. And if we see how old was tissue, it was just 5 months old tissue. And in addition to this, we found PIK3CA mutation, important biomarker for breast cancer, and MSH2, MSH6, CCND2. And tumor mutation burden both, in tissue 20 mutation per megabase, and in blood we had 27 mutation per megabase. So this is again telling us that liquid biopsy is better than tissue biopsy. And this also suggests that we are moving towards the longitudinal monitoring, because cancer is so complex, every day it's evolving, and we should longitudinally monitor the cancer using possibly at the baseline tissue biopsy and followed by liquid biopsy. What we believe maybe every 3 months should be a liquid biopsy done. And as we see the ESMO guidelines here, it's also suggesting that liquid biopsy should be done, and we know that now we are able to get the tissue tumor DNA from different stages, but if technology is available, if a small amount of ctDNA is available, you can do the NGS. So then second question is, most of the on-calls ask question, why you need a comprehensive panel? I'm comfortable with going for small gene panel or single gene panel. So one of the example I talk about BRCA beyond BRCA1 and BRCA2. So what we do mostly, if we have to go for a PARP inhibitors, we do BRCA1, BRCA2 mutations, and if we find there, and we start with PARP inhibitors. But what we know actually HRD, as in previous talks we were discussing also, it is BRCA1, BRCA2. It's beyond BRCA1, BRCA other HRR genes. It is LOH, we were just discussing here, large say, transitions, telomeric allelic imbalance, epigenetic modifications, and some unknown factors. So we took 177 ovarian cancer patients where we did a comprehensive analysis, but extracted first information on BRCA1, BRCA2. We found only 17% are positive, that means eligible for PARP inhibitors. When we included other HRR genes, including BRCA1, BRCA2, the percentage went up to 26%. But when we do the comprehensive way, where BRCA1, BRCA2, other HRR genes, loss of heterozygosity, LST and DAI included, that percentage went to 54%. So that means, actually 75 times you are actually missing the patients who could actually benefit with PARP inhibitors. So one of the example is here, how we have to go beyond BRCA1. So this is the, ovarian cancer patient, 55-year-old, which has a progression with metastasis BRCA1, BRCA2 negative, done by, single gene testing. When it came to us in both on tissue as well as liquid, what we found is KRAS G12 mutation, TMB high, MSI stable, LOH high, which is 18%, HRD high, which is 48%, and as also in the ctDNA, we found KRAS mutation, G12D, TMB high, and MSI low, LOH high, and HRD high. So what it suggests that, irrespective of, even BRCA1, BRCA2 or other HRR genes are absent, there is a possibility a patient could have an HRD high, homologous recombination deficiency could benefit with PARP inhibitors. If we look at it, the literature also, there is literature available, genomic LOH high greater than 16% in patients which are BRCA1, BRCA2 wild type, benefit with PARP inhibitors, and multiple studies are available. Another issue is, when like guidelines, which recommend retesting of HER2 amplification in ER/PR positive HER2 negative patients on every metastatic site. We know now literature says every HER2 negative patient turns HER2 positive, 25% of them, and out of them 20 to 30% are bone metastasis, and 50% of them do not have sufficient tissue to do the retesting. That's why the liquid biopsy plays an important role. One of the examples, we have found a breast cancer patient who was ER/PR positive, HER2 negative, treated with chemotherapy and AI, progressed with multiple bone metastasis, tissue was insufficient for doing the HER2 testing. When we got the liquid biopsy, we found T53, ESR1 mutation, very importantly, HER2 amplification, 8 copies. So, which was not possible because tissue was not sufficient. That's why liquid biopsy plays an important role. Also, liquid biopsy, now comprehensive liquid biopsy plays an important role in other cancer types, mostly, those difficult to treat, like one of the example, carcinoma, unknown primary origin. So what we have found here, KIT, KDR and PDGFR gene, 3 genes together having amplification 10, 12 and 12 respectively. And this suggests that, there is the possibility of identifying targeted therapies in the case where we do not know exactly how to deal with them, and there are a lot of literatures supporting such scenarios. To summarize, comprehensive profiling helps us to identify diagnosis, prognosis, help to identify driver mutations, capture intra and intertumoral and metastatic heterogeneity, help us to find the presence of biomarkers which can tell us response and resistance, and give the rationale for combination or sequential therapy. And we know it's a non-invasive procedure, help us to find the amount of ctDNA which can help us to even define or monitor the disease progression, monitor the treatment response, and most importantly, what we now move to molecular response. Possibly is the next wave for beyond PET CT scan, we may go for molecular monitoring. Thank you so much.
Any questions? Yeah.
Compared 5 months of difference between tissue and plasma. So how can it be presented? You showed 1:3 ratio.
So typically it's something more than in liquid biopsy, VFU don't get as much as you get in tissue biopsy. But if you look at that particular case, ESR having 1% fraction, possibly in the tissue it was a subclonal mutation, meaning small amount of.
जब difference duration का 5 months का duration का इधर उसमें थेरेपी हुई नहीं हुई या प्रोग्रेस किया, एक में टिश्यू में आपने 1 दिखाया, इधर में 3 दिखाया, तो this data is not correct.
Why is not correct?
Because 5 months के बाद में तो अलग status होगा ना पेशेंट का.
That's what I'm showing. That's what I'm showing. 1% change went up to 2.59%.
कंपेयर आप तब कर सकते हैं एक साथ आपने ट्यूमर भी लिया टिश्यू एक साथ आपने ब्लड लिया तब आप कंपेयर कर सकते हैं 5 मंथ के बाद में आप एक का 5 मंथ के बाद लिया एक बार 5 मंथ पहले तो उसमें कैसे आप कंपेयर करेंगे
So what I am saying is tissue biopsy was done approximately 5 months before.
वो डेटा करेक्ट नहीं होगा ना
Wait. It was done 5 months ago, and blood sample was taken today after 5 months. So what it suggests, 5 months before, the tumor, say for example ESR1, a small number of tumor cells having ESR1 mutation. Now after 5 months, tumor is growing faster, and when we do blood sample, what it indicates, there are more tumor cells having ESR1 mutation. Right?
लेकिन आपको उस समय का रहता तब आप कंपेयर कर सकते हैं प्लाज्मा से अगर उसी डेटा
Yeah, absolutely. That's what I'm telling. That's why I'm showing the study here. If it's more than 6 months old, you miss 40% of information. Okay? And now what I'm showing here.
That's the same thing I am saying that data is not correct.
No.
40% is a large amount.
No. Why is not correct?
What he has been stressing on, like both should be done simultaneously.
Yes,
That's what he is stressing.
definitely. और तब आप 5 month बाद आप correct कर उससे compare करें.
I'm not saying whether it
No.
both should be done simultaneously. I'm saying why circulating tumor DNA is possibly better solution than tissue biopsy.
He has
There are
just
two
shown a
reasons.
comparison that
There are
he is
two reasons.
circulating, yeah.
Tumor heterogeneity and tumor is evolving. Okay, tumor is evolving. See, I do not know about this patient whether it was treated with ESR, inhibitors. Okay, at least I can confidently say patient was not treated with JAK2 inhibitor. That's why he has that high-fold, VAR. Okay, and then tumor is evolving. We have additional
I
mutation.
think we
which
can
was
take the discussion later on.
So this-
If there are more questions, I think we can take it
One
later
more
on.
question that how much blood is required for liquid biopsy?
Sir,
So we can discuss it. We can discuss it.
This is very
later on.
interesting discussion going on, but can we please have this
There are
discussion
limitations
at
of
4:00
time.
because we are running short of time. It is already 1 hour late, so that's why we just want to end up.
Yeah.
Sir, you can have the discussion after this also.
Thank you so much.
हो गया खत्म हो गया.
So I would like to thank the chairpersons, Dr. Okay, and Dr. Varsha Trivedi, and again it is with me. So Dr. Vivek and Dr. Varsha Trivedi for being here for such a night. Ma'am, आपको मैंने spot पे बुलाया. So thank you very much. Thank you very much for being there. So now we will conclude for the day. And now the time for the annual general body meeting for MPI members. So we are concluding the session today. For all the members, you please note that all entire proceedings of the conference are being recorded, and it will be available for at least 1 month on the MPI website. Thank you very much.